Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research and affordable and accessible cancer care come together for a brighter, healthier future.
Am I audible?
Yeah, yes, sir. You are audible.
Okay. And am I visible also?
Yes, sir.
Okay. Thank you.
Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research and affordable and accessible cancer care come together for a brighter, healthier future.
A very good morning to one and all assembled here for the BMSECON 2023 conference. Sir, can we start the session, sir?
Should I go ahead?
Sir, if you're ready, then we will go ahead, sir.
Okay, all right.
Okay, sir. Just a moment.
Where are my slides? Okay.
Just a moment, sir.
All right.
A very good morning to all. Greetings to everyone from Arapadi Medical College and Hospital, Vinayaka Missions Research Foundation, deemed to be University Puducherry. I am Dr. Aarti, Assistant Professor, Department of Medical Biotechnology, AVMC. And on behalf of the organizing team of the 4th International E-Conference, BMSECON 2023, I extend a warm welcome to you. This year's theme is Beyond Boundaries, Exploring Excellence in Basic Medical Sciences. This is the 4th consecutive year that the Phase 1 MBBS departments of Anatomy, Physiology, and Biochemistry, along with the Center for Biomedical Research, are organizing this virtual conference. A heartfelt thanks to everyone who has joined us today. As we embark on this intellectual journey, we are privileged to begin the conference with a keynote talk. Our esteemed speaker, Dr. Deepak K. Sarkar, brings a wealth of knowledge, making this session one of anticipation and excitement. I request the moderators of this session, Dr. M. Gopinath, Professor and Head, Department of Physiology, and Dr. Lakshmi Jatia, Vice Principal, Student Affairs and Professor of Physiology, AVMC, to take over the session. Thank you.
Good morning, one and all. My greetings to you all from BMSECON 2023. We are very privileged to have doyens in medical sciences. Dr. Deepak Sarkar is there, Dr. William Stringer, and our chairpersons for this session, Dr. K.K. Deepak from Delhi, AIMS Delhi. Next slide, please. So our chairperson, Dr. William Stringer, I'm very privileged to introduce our chairperson, Dr. William Stringer. He's Professor of Medicine at David Geffen School of Medicine at UCLA. Currently, the Division Chief of the Respiratory and Critical Care Physiology and Medicine Division at the same institute. And he is a renowned pulmonary and critical care physician with research interests in improving function and quality of life in chronic respiratory illnesses. He has been a part of many research interventions, which include various devices, medications, behavioral therapy, and exercise rehabilitation in motto of exercise is the best medicine. Yes, very true. His clinical trial on long-haul COVID rehabilitation and recovery research program is the current project on process. So in 2021, Dr. William Stringer gave the same keynote address on long COVID. So that was very well appreciated by all the delegates. His research interests are in chronic obstructive lung disease, long-haul COVID, exercise physiology, pulmonary function testing, and rehabilitation sciences. Dr. Stringer received his undergraduate and medical degree from the University of California, San Diego, and his internal medicine residency and pulmonary and critical care training at Harbor UCLA Medical Center. He has published more than 23 international journals on the airway disease and effect of exercise on the airway. He has 38 years of experience.
Thank you. Now I feel privileged to welcome our next chairperson, Dr. K.K. Deepak. Sir is former professor and head in Department of Physiology, AIMS, New Delhi, and currently working as visiting professor, Center for Biomedical Engineering, Indian Institute of Technology, IIT, New Delhi. He has contributed to curriculum planning at national level at almost 6 workshops and contributed as resource faculty for workshops at the regional and international level. He has been awarded Dr. S. Radhakrishnan Memorial National Teacher Award in 2014 by All India Freelance Journalist and Writers Association, and 7th Dr. B.K. Anand Memorial Award by 2020 by Nutrition Society of India, NIN, Hyderabad. His Major General S.L. Bhatia Oration Award from APPA and many other awards to his credit from PSA and NAMS, National Academy of Medical Sciences. He has published more than 300 research abstracts and publications. He has contributed to biomedical innovation and device development in collaboration with IITs and NIT Jalandhar. Setting up a autonomic functional lab in 1989, a pioneer diagnostic facility for assessing autonomic dysfunctions and vascular function lab. And he had the responsibility to supervise and conduct the All India PG entrance examination, AIMS PG entrance examination, and other several postgraduate entrance examinations. He has served as a chairperson for the expert committee which prepared the Age Appropriate Fitness Protocol 2020, which were launched by Honorable PM Shri Narendra Modi. Welcome you, sir. We welcome you for this BMSECON to chair our session. Now I invite Dr. William Stringer to introduce our guest speaker, Dr. Deepak K. Sarkar. Over to you, sir.
Thank you very kindly for the invite and to get to introduce Dr. Sarkar. He was currently a distinguished professor of the endocrinology program in the Department of Animal Sciences at Rutgers University. He's also the chairman and the director of the Department of Animal Sciences. He got his bachelor's, master's, and PhD at Calcutta University in physiology and neuroendocrinology, and his doctor of philosophy at the University of Oxford. He did postgraduate work at Oxford, Yale, and Michigan State, and progressed through the assistant, associate, and full professor at Washington State prior to Rutgers. He and I actually were at UCSD in the early 80s, but I didn't know him at that time. But he's recognized for academic excellence in both Indian and American universities. He has well over 180 peer-reviewed publications. His interest is in neuroendocrine cell growth, differentiation, and secretion. Has done some very interesting work in addiction medicine, both alcoholism and opiate neurotoxicity, and also has an interest in fetal alcohol syndrome and some of the abnormalities of circadian rhythm. And finally, the effects of stress on infection, cancer, and also those alcohol-related diseases. So he'll be giving the keynote address to the 4th International E-Conference, BMSECON 2023, and he'll be talking about the identification of new mode of cell communication within the nervous system and focusing on neuroinflammation. So we're really quite anxious to hear your talk, and thank you for giving this, Dr. Sarkar. It's a real honor to be here with you.
It's my turn.
Yes, sir.
All right. Okay. Thank you very much for the lovely introduction, Dr. Stringer, and thank you for the invitation. I think this is very exciting. I have never been one of like this, but when I got a call and I looked at it, it really got me very interested, especially developing very modern biomedical sciences introducing into medicine is very exciting. So thank you again for your invitation. So what I really like to do today is, bringing it to you, giving you a description of something new going on in the neuroscience research. In the neuroscience research, we all know that brain is composed of all cell body, particularly neurons and glia cells and others. And this glia neuron, as you all know, they, people used to believe the brain is all under control by the electricity. Neurons generate electricity, it transmits from one to other cells, and then releases some chemicals, and then they modify the activity where it terminates.
Sir,
Um,
sorry to interrupt you. Please share your slide.
I'm on slide.
No, sir. Please share your slide.
Okay. Am I sharing my slides now?
Yes, sir, yes, sir.
Yes, sir. Please show in full screen mode.
Sir,
Okay.
please start a slideshow. Yes, sir. Now it's fine.
You're fine? Okay. All right. Thank you for telling me that. I couldn't see it. But anyway, what I was talking about, that we are dealing with, what I like to deal with today is some mode of new communication, which has been understood recently. And there's a lot of excitement going on with this communication. And this communication is within the brain and all over the body. But brain part is very exciting for me. And that's what I'm going to talk about. And this communication is really a little bit different from the traditional one. In a tradition in the brain, we have neurons which communicate by the electrical current and releases chemicals, activate the, where the neuron terminates, and induce the function. That's what traditionally has been known. And also there are, within the brain, there's not only neurons, but also glial cells, varieties type of glial cells, and they are microglia, astrocytes, dendrocytes, and all kinds of other glial cells. And they also participate within this communication. And unlike neurons, these glial cells, they don't really terminate, they don't make actions, terminals. So they communicate through the extracellular fluid. And this extracellular fluid, they releases peptides, they releases growth factors and others. But recently, the discovery was been made that every single cell now is not only endocrine cells secrete things that goes to the goes to the blood and travels all over. Neurons secretes and goes to the CSF fluid and goes all over. And these the glial cells and others, they also secrete, but they are predominantly use these chemicals called exosomes or microvesicles. So my talk is primarily on these microvesicles. And I also wanted to bring to the issue of how these microvesicles involved in neuroinflammation. Why is neuroinflammation? Neuroinflammation is very important in Alzheimer's disease, Parkinson's disease, and one of the diseases we deal with the fetal alcohol syndrome. So here, this cartoon picture showing that all these different cells in the brain, they're secreting the microvesicles and the extracellular vesicles, and they use this, and they have a target size. They have receptors in every cells. They can go in the target cells and act on those cells. So what I like to show you then, how do they act, what they do when there's a neuroinflammation goes on. So here again, let me bring to these extracellular vesicles I was talking about. And this is all cell types. But within these brain, these microglia and the other glia cells, they very much use these. And these extracellular vesicles are really comes from within the cells, and the cells make these vesicles, and these vesicles then get secreted out and as exosomes, and they are smaller size, or they bud out as a microvesicles. And these exosomes, or these microvesicles, they contain varieties of cargo materials. And these cargo materials are various hormone, proteins, genes, and also microRNA and many others. So what we are doing, we are trying to see how these microvesicle exosomes are involved in neuronal killing. And neuronal killing takes place, during the aging process. We get Alzheimer, during fetal alcohol exposures, during large amount of alcohol drinking. There's a lot of neuronal killing going on. And why do they get killed? And how did the exosomes participate in it? That was our objective. So that's what I'm going to tell you, how we dealt with it, how we understood it, and how we using this information to go getting into the treatment. So the system we are going to use is these the neurons within these part of the brain called hypothalamus, and this part of the brain get affected by large amount of alcohol or fetal alcohol, and not only this part of the brain get affected, but also various part of the brain like corpus callosum, cerebellum, cortex, hippocampus, and various other part of the brain also get affected. The neuron get killed. So the question then we started asking, why did they get killed? I mean, what are the signals? What are the molecular mechanisms involved in this killing process? And this is important not only for alcohol filled, but also important for Alzheimer and various other neuronal diseases. So the way we started looking at, how did they get killed and what are the molecular mechanisms? So what he did, we know that, in a human being where mother drank a sufficient amount of alcohol, causing these babies, which are called fetal alcohol syndrome babies, they have a lot of neuronal deficits. They have a lot of neural diet. So we used the animal model to create these fetal alcohol syndrome conditions. And this animal model is this rat particularly. They were given alcohol, the doses, this very moderate dose, and they were given for either in midterm of pregnancy or late term of pregnancy. If we do that, there's a lot of neuronal killing goes on. And the area we are interested is an area called hypothalamus. Why we are interested in hypothalamus? Because these babies which are born, they have a lot of anxiety, depression, hyper-stress response, and many other behavioral problems. So we did a lot of studies identifying the majority of this problem goes on because of the defect in the hypothalamus. And what is the defect in the hypothalamus? Within the hypothalamus, there is a group sets of neuron called pro-opiomelanocortin, or POMC. What are these neurons? This neuron, POMC, is very much involved in varieties of neuroendocrine function. What are the functions? What does POMC does? POMC cause activation of endorphin release. So when you exercise or you eat a chocolate or you eat ice cream, you feel good. And that good cause, because of the POMC releases endorphin, makes you happy. And also, these neurons, when they're not happy, the stress gets hyper. It's very stressful. Why is it stressful? Because this POMC neuron turn down the stress, makes it normal. So it controls stress response. It also is very important for metabolic function. When you eat, the POMC neuron gets activated, tells don't eat anymore, because you might get obese. So POMC reduces the food intake behavior. So abnormality of POMC makes you obese. And POMC neuron also are involved in insulin actions. So it also controls the glucose imbalance. So there are varieties of function the POMC neuron does. So that's got us interested in these neurons, how this fetal alcohol or alcohol exposure kill this neuron, what are the mechanisms involved in it. All right, so let's see what we could do and how we can study these. So we have these animal rat models. We gave them the first 5 days of birth from postnatal day 2 to postnatal day 6. And when you do that, we find these neurons in the babies are significantly reduced. So we have a control AD, pair-fed control, and alcohol fed. And then when you do that, we find this alcohol fed has a reduced number. So why is that reduced then? We anticipate that this reduction is caused by neuroinflammation. So one of the cell bodies within the brain causing neuroinflammation is called microglia. And this microglia can be prevented by a drug called minocycline. So when you give this alcohol-fed animal with minocycline, we see this neuronal death is reduced. So that kind of gave us the idea the microglia is involved in this neuronal killing. All right. So if that is the case, let's see how does microglia do that. So when you do this, study this in confocal microscopy, what you find with this microglia are connected with all these beta endorphin neuron in the normal animals with the control pair-fed control animal, they are all normal. But when you get the alcohol fed, this microglia is chewing up this neuron, engulfing it. So this confocal microscopy picture showing that microglia is interacting and killing this neuron. And this killing could be blocked by minocycline. So again, proving that microglia is killing these endorphin neuron or POMC neurons. So how does the microglia do that? So we started anticipating that it may be using exosomes. So how do you study this? We have a in vivo exosomes is everywhere. How do you know what is the microglia exosome? How do you know it's not coming from the astroglia or from neuron? So we need to figure out the technique how to do it. So what we did then, we take these babies, which are alcohol fed or control fed, and then get the microglia out and cultured them and made a pure microglia culture. Or we keep them in vivo and get the microglia separated out and before we treat them, we did the microglia culture and then put alcohol and get the exosome out. Or after we treat them, we collect the hypothalamus out and extract and dissociate the microglia, break the microglia rupture, and got the exosomes out. And how do you know we got the exosomes out? We looked at the varieties of marker of exosomes, and we found there is a significant number of exosomes. We looked at the other molecule within the cells, like mitochondria, the Golgi and extracellular vesicles. They are all absent. And when you look at the purity of these, we found there's no neuron. There is a very slightly astrocytes, but primary microglia. And when we looked at these nanoparticle, and we found that there are really very pure exosome molecule in it. So now we got pure exosomes from microglia, and we verified with varieties of marker, with a nanoparticle, with the TM microscopy, and we identify that we have really exosomes from microglia. So let's see how does microglia exosomes is causing this signal which is killing these POMC neurons. So now we have the exosomes, and we started looking at from control and from alcohol fatty diet. We find indeed the exosome number is high, both in MBAs in vivo and or in vitro. And when you look at their activity, killing activity, which is used the aminopeptidase activity, or with metalloprotease activity, we find indeed alcohol feeding increasing the killing activity of microglial exosomes. So how do you know those are really exosomes or they're not others? So what we did, we used the blocker GW4869, which blocked the exosomes transmission. When you do this, we prevent this killing activity of beta endorphin by both in vivo and in vitro. So showing that these exosomes are involved in this killing, and exosomes coming from microglia killing these neurons. All right, so what are the molecules then in exosomes causing this killing? So we took varieties of approaches. One of the approaches is called proteomic approaches. What it does, you get the protein out and put in the proteomics and identify all the proteins got changed by the treatment. So we took the alcohol-treated microglia and then put in the proteomic, and what we find separate molecules are upregulated, increased by alcohol, and several molecules are downregulated by alcohol. One of the molecules got us very excited about, called complements. I will tell you why they are very exciting. And other molecule, also chemokines, apoptotic molecule, RNA molecules. So we started looking at what are these molecules and how they are involved in this killing process. So microRNA is one of the molecules we found in the proteomic, indication of proteomic. Then we used the tilde RA, which detect all the different microRNA. And we found a large number of microRNA are elevated, and some of them are also suppressed. And we are currently ongoing all this stuff, and I'm not going to present all this, then it will be 2 hours lecture. So microRNA, I'm not going to talk. I'm going to talk some of the findings which are already been proven and very exciting. So one of the molecules I'm going to talk about is called complement. Complement in the brain? Complement is supposed to be in the periphery. Complement is supposed to be working as an immune substance within the periphery. When the peripheral immune system, complement is very important. How is it important? When there's a viral or bacteria comes and you get antigens, and immune antibody comes and binds the antigen. When it binds, complement get activated. And then what complement does, it have a varieties of protein of complements. They then activate the antigen bound antibody and goes to the bacteria and make a hole in it and kills the bacteria or virus. So that's the way complement function in the periphery. But how it could be this antibody doesn't go to the brain? So why is the complement in the brain? The complement in the brain does a different role. They are nowadays they have been found that they affect the neurogenesis, they affect neuronal migration, they affect synaptic remodeling, varieties of function. For us, we found complement is also involved in microglia killing of neuronal function, or microglia killing of neuron. So this paper, this work is recently published in Journal of Neuroscience. And so let me go through some of these, what he found and how did he establish this complement is involved in the neuronal killing. So the complement, as I already showed you, complement is composed of varieties of complement protein, C1, C2, C3, C1q, MAC and various others. I showed you the chart. I'm going to go through these again, showing you how we prove that complement is involved, is neuronal killing. First thing we did, we stained the hypothalamic beta endorphin neuron, and also stained the complement. We find this, look at the complement, the C1q, these all are covered with this beta endorphin neuron. So it's binding with the beta endorphin neuron when you stimulate with the alcohol. So alcohol stimulates complement and it binds to the beta endorphin neuron. Microglia secrete this complement protein, which goes to the beta endorphin, it binds on it. So it's showing this alcohol-fed animal, the brain C1q level is high. Then also it increases caspase-3, which is marker of the killing. The C3 is another bound of complement. So all this thing is activated when the alcohol is there, or we use alcohol activated microglia exosomes put in, we found a similar effect. So here is saying that all this activation could be blocked by minocycline. The minocycline is related to the microglia. So it's the microglia which is secreting complement and which is causing this neuronal death via the complement system. So, so we did then complement with both in vivo and in vitro, and with the exosomes, without activated exosome and inactivated exosome. What we did find that when the alcohol is there, is complement C1q, C3, C4, they all get activated. And this both in vivo and in vitro system. So that means that the complement are involved. So C1, C2, C3 proteins are involved. We also then start looking at their interaction directly with the neuronal death. And when you do that, we found is also activate the MAC or C5b9, which is the large part of the molecule which makes the hole in the cells. So when the microglia secrete exosomes, which then activate C1, C2, C3, and then C3 get activate the MAC protein, and that caused the destruction of beta endorphin neuron and activate the caspase-3. So we also find that with the in vitro, using the exosomes from the microglia, and again we find that C1, C3, C4, C5b all get activated, and they could be all blocked by the inhibitor of the complementary inhibitor, anti-C1q or other complementary inhibitors. So the take-home message then, when ethanol comes, ethanol is given, is activated microglia, which produce exosomes. Exosomes then have complement. Complement then get activated. Then it comes to the beta POMC neurons, and then using the cascade of complement reaction, it makes activate the MAC on the neuron, which makes a pore and kills the neuron. In addition to that, it also activates the mitochondrial ROS. ROS is an antioxidant, and it also activates caspase-3. So in addition to this MAC-induced caspase-3, ROS also induces caspase-3. This is all part of the microglia induce the exosomes, which uses this pathway to kill the beta endorphin POMC neuron. So this is one of the processes. In biology, there is no such thing as a single thing or does everything. In biology, there are hundreds of things work in concert or in sometimes it works, some other time it doesn't work, and that's why it is very complex and it has a lot of the homeostasis maintenance not only caused by one system. So complement system is not the only system. There are many other systems involved in exosome-induced neuroinflammation and healing. So the next system we started looking at is called chemokines. The chemokines are varieties of chemokines, and chemokines are also involved. So when you look at the chemokine array, we find there are two chemokines, MIP1 alpha and MCP1 are very much activated in the exosomes. And these are both in vitro and in vivo. And so the ethanol activate these. And again, minocycline, which is a microglia blocker, prevents this activation of MIP1 alpha and MCP1 and MIP1 alpha. So we started looking at the role of these MIP1 and MCP alpha, MCP1, MIP1 and MCP alpha. And when you look at, we see these, when we use the chemokine blocker, when you use exosomes, and it increases from ethanol-treated exosomes, it increases the killing of the endorphin neuron. But when you put the chemokine blocker, it reverses it. So showing that chemokine are also involved in killing microglia-induced killing of beta endorphin or POMC neurons when you give alcohol. And furthermore, we see the apoptotic death of these neurons could be blocked by the chemokine blockers. And MCP1, when you give them, it increases killing. But when you use the chemokine blocker with alcohol, it suppresses it. Or chemokine blocker with MCP1, it suppresses it. So showing that chemokines are also involved in this killing. So how does those chemokines do that? So we then looked at the PCR array of the genes taken out from the exosomes, alcohol-activated exosome-treated POMC neurons, and studied all different kind of genes to understand the mechanism involved within the cells. We found there are varieties of apoptotic genes that are elevated, like caspase-3, caspase-9, BAX, BCL2. There's all kinds of apoptotic genes that are activated. We also find varieties of inflammatory genes that are activated. IL-1 beta, IL-6, CCL2, TNF alpha. So all these inflammatory genes are also activated. In addition to these, the glutamate receptor genes we found activated. And also varieties of transcription signaling genes are activated, like GSK3 beta, MCP-IP, STAT3. So these are all genes are involved in this chemokine activated pathway. So when you put them together, what we predict that alcohol, when through the chemokines, it secretes the exosomes, and exosomes have chemokines, MCP1 or MIP1 alpha, and which is also called CCL2. And this MCP1 alpha act on this receptor CCR2 and activate the GSK3bA or MCP-IP, which then activate the gene expression related to the apoptotic genes like caspase-3, caspase-9, PARP1, and then they go and cause activation of mitochondrial BCL2 and other genes, and also it caused the oxidative stress by NMD receptors and also varieties of inflammatory cytokines and causes the death of this neuron. So this is the molecular mechanism we identify from these chemokine-related the involvement in exosomes activated killing of endorphin neurons. And so that's good. So can he make a therapy out of it? Could he use something to prevent some of these? So we used some blocker of these chemokines and treated those animals which were given alcohol, and we found that this blocker indeed reverses some of the actions when you take them in the in different ages, the postnatal period or adult period, we do find indeed both male and female, we can reverse some of this healing. This is the adult, the previous one is the neonate. The adult, we also find this effect is, we treat them during the puberty, during the neonatal period, and we alcohol as well as this drug, and we prevent some of the killing. So that gives a lot of excitement that we could, we could bring some drugs in the market perhaps and help for preventing these alcohol-induced killing. And so why do we need this drug? Can this drug reverse some of the actions also with respect to the behavioral aspect, like anxiety or hyper stress response? And we find indeed we could also do this by these chemokine blockers. We could reverse some of the effect on the open field and elevated plasmas, which is the behavioral test one use for studying anxiety behavior. And when we use this drug, we find that these effects are suppressed, normalized in many ways, both in the anxiety behavior as well as the stress response, both male and female, very similar way. So these are exciting, and we are making some genetic modification of these exosomes. We are modifying these chemokines or complements, and then trying to use them in the animal, trying to see whether we can reverse some of them. So that work is currently in progress. Also the microRNA stuff in progress. So anyway, this is the summary of the thing I presented today. I just showed you that the exosomes or extravesicular pathway is one of the new exciting pathway, is causing lots of excitement within the field, and not only in the brain, also in the periphery, in the cancer field and others. And the reason behind that exosomes molecules are natural molecules, and you can modify the exosomes molecule by putting new genes or putting modified proteins and treat them. Unlike the drugs, which when you add the drug have a lots of side effects and others, but exosome is a natural molecule, and you can load them with the altered the genes or altered protein and give them to the patients or give them to the subject, and perhaps and exosomes has the target receptors, then you can drag the exosomes to the target site and cause the reversal of the effect. So that's where the excitement comes. Exosomes gives a new venue of the therapeutic use of the drugs, therapeutics of the gene therapy, which might really advance the field of many diseases, including fetal alcohol syndrome. With that, I want to acknowledge the contributions of the people who worked on these, the Natka, Dr. Vajiva, Omkaram, and Benedict Russo, Tina Franklin, Miguel Sahani, Saista, Prashant, Pallavi, and Lucy. These are fellows and graduate students, and also the National Institute of Alcohol Abuse and Alcoholism with multiple grants for this project. Thank you for listening. I'm open for questionings.
Thank you, sir. That was wonderful talk and wonderful research which you have done, sir. Over to chairpersons for their queries. Thank you, sir.
Yeah. So can I speak? Yeah.
Yes.
So thank you so much, Deepak Sarkar. It's wonderful listening to you. And very nicely you have elucidated how alcohol-induced activated microglia can, and then further the block by which you can reverse that. So it's a very important piece of research with the alcohol, etc. And we have seen in the worldwide alcohol dependence of the pregnant women is on rise. So this is another issue which has been addressed even in India. But I was wondering, you told that POMC, when stimulated, might induce endorphin, and normally this is related to activity like physical activity and exercise.
Yes.
So you think in a fetal state, when it's not doing activity exercise, it may be activity. Even in that activity, will be releasing endorphins. That's my question.
Yes, I think there are some study going on with the children with the fetal alcohol syndrome. They use all kinds of exercise phenomena, and they help something. But the problem is the exercise and others giving it to the kids are often difficult, especially if you have anxiety and depression. They don't listen to you, they don't do stuff. So it's very difficult to do that. But I think there's also, it's quite beneficial. And believe it or not, this endorphin is not only doing this, but also does a lot of things with prevention of cancer and others, which we have done a lot of work with.
Yes.
So the exercise is very important for health, better health.
Yeah. Thank you so much. Thank you for the nice question. Congratulations for delivering a wonderful talk. Thank you. Over to other chairpersons. Thank you.
Thank you, Dr. Sakar. It was a really nice talk. I just wondered a little bit about if you've examined the exosomes and microvesicles outside the blood-brain barrier, and whether there's communication with other organs with some of those, messenger RNA or inflammatory mediators.
Yes, I think exosomes, there's the field is making a big difference with the study of exosomes. Exosomes are small molecules. They can pass the blood-brain barrier easily. So nowadays, even we are doing some work with the saliva, human saliva, and we are finding a lot of brain-derived molecule in the saliva. So the only way we can explain it must be coming through the exosomes.
Well, and also the question sort of with COVID these days and spike protein and all those things, have you actually looked into whether some of that's being transferred in the microvesicles and maybe associated with some of these neurologic abnormalities we see in long COVID?
I really haven't done any work with long COVID or COVID. So that's a field. I think there are several paper I saw. People are working with the exosomes also, because obviously would be a good target.
Yeah, there's definitely some literature out there that shows that either the protein or some of the proteolytic fragments actually may be pro-inflammatory to some of the toll-like inhibitors.
Sure.
Well, thank you. Nice talk.
Thank you, sir. Thank you. sir,
Okay.
and chairpersons for honoring the session. We would like to give a token of appreciation and felicitate our speaker with a e-certificate and a memento. Thank you, Dr. Sarkar, sir.
Thank you.
And we'd like to give a felicitation to our chairpersons, Dr. William Stringer. Thank you, sir. Now we'd like to provide a e-certificate and virtual memento for our first chairperson, Dr. William Stringer. Thank you, sir. Now we'd like to give our e-certificate and a virtual memento to our next chairperson, Dr. Professor K.K. Deepak, sir. Thank you, sir. Thank you, everyone. Another 5 minutes, we'll start our inauguration program. Dear panelists and resource persons, please be online, sir. We'll resume another 5 minutes for the inaugural program. Thank you.
Thank you, sir. Thank you, ma'am. Please stay longer as we will commence shortly. Meanwhile, I request the delegates to sit back and enjoy the videos of BMRF and BMSECON.
Dr. Aarti, do you hear me? Hello? Manju
Yes.
ma'am?
Yes, sir.
Do you hear me now?
Yeah, I can hear you, sir.
Okay. I just checked my voice, ma'am.
Okay, sir. Actually, all are inside, sir. Only we are waiting for Mahalakshmi madam to enter, sir. Vice Chancellor, sir, Kotur, sir, Dean, sir, Vishnuvat, sir, all are there, sir.
Just to remind her.
Yeah, I just called, sir. She told she'll be entering. When she enters, we'll start, sir.
Yeah, we can start. Ask Dean, sir, and VC, sir.
Yeah, I'll ask, sir. I'll ask.
Am I audible, ma'am?
Yes, Langadhar, you are audible.
Thank you.
Dear delegates, we are about to start the inauguration program. We are just adding all the panelists for the inauguration.
Ma'am. Sir, Sudhir sir, shall we start, sir?
Yes, yes. What the participants only 38?
No, sir. That is only in our link, sir. There are more than that. We have 2 links, sir. We cannot
Okay.
see the number of participants from this link, sir. This is only for
Okay.
VIPs, sir. Okay.
Okay.
There are a lot of people, sir, around 300, sir. Thank
Okay.
you. Shall I start, sir? Okay.
Yes. Professor Sekel has not joined?
I have joined.
Okay, okay. Good morning.
Thank you, sir. A very good morning to everyone. On behalf of the management, faculty, and students of Arupadi Veedu Medical College and Hospital, Vinayaka Missions Research Foundation, deemed to be University Puducherry, with immense pleasure, I welcome you all to BMSECON2023, the 4th International E-Conference on Beyond Boundaries, Exploring Excellence in Basic Medical Sciences, organized by the Phase 1 MBBS Departments of Anatomy, Physiology, and Biochemistry, along with the Center for Biomedical Research, ABMC. I, Dr. Aarti, will be emceeing this inaugural program. The purpose of this conference is to bring together clinicians, educationists, diagnostic consultants, and biomedical researchers on a single platform to share their knowledge towards the advancement of basic medical sciences. To commence our proceedings, let us seek the divine blessings of Goddess Saraswati through a solemn invocation song.
Saraswati Namastubhyam Varade Kamarupini Vidyarambham Karishyami Siddhir Bhavatu Me Sada Siddhir Bhavatu Me Sada Siddhir Bhavatu Me Sada.
Thank you. I request the organizing secretary, Dr. M. Manju, Professor and Head, Department of Biochemistry, to welcome the gathering.
Thank you, Aarti. It's a very, very good morning and a warm welcome to one and all. With the divine blessings of God Almighty, our founder chancellor, Dr. Shanmugasundaram sir, and the benevolence of our beloved trustee, Mrs. Annapurni Shanmugasundaram ma'am, our beloved chancellor, Dr. A.S. Ganeshan sir, Madam Chancellor, Dr. Anuradha Ganeshan ma'am, and as the organizing secretary, I, on behalf of the organizing team, take pride in welcoming you all to the inaugural ceremony of BMSECON 23, organized by the Departments of Anatomy, Physiology, Biochemistry, and Center for Biomedical Research. We at Arubadi Medical College and Hospital, we are honored and delighted to host this international conference for the fourth consecutive year. And the theme that we have selected for this conference is Beyond Boundaries, Exploring Excellence in Basic Medical Sciences. This conference has the distinction of bringing together such a large number of resource faculty and delegates across the globe for a vibrant and an intense exchange of views, knowledge, and experience on the emerging trends in research, diagnosis, and innovative TL methods. And we are very happy to announce that this year too, we had an overwhelming response with 850 registrations and 240 plus abstracts for scientific presentations. On this momentous occasion, I wholeheartedly welcome our Honorable Chancellor, Dr. A.S. Ganeshan sir, though in absentia, a visionary who has been constantly supporting us in all our endeavors. His wishes and blessings are always with us. Now it's my honor to welcome our Honorable Madam Chancellor, Dr. Anuradha Ganeshan, Vice President, BMRF, whose invaluable guidance is cherished by all of us. We warmly welcome you, ma'am, in absentia. The organizing committee is extremely delighted to welcome the chief guest for today's inaugural program, Dr. Deepak Sarkar. He is the director of the endocrine program in the Rutgers University, United States of America. We just heard his enlightening keynote address. We extend a warm welcome to you, sir. We are delighted to have the illustrious presence of our Honorable Vice Chancellor, Dr. P.K. Sudhir, who is a man of wisdom, vast experience, and intellectual acumen, and has always been a support for us. We wholeheartedly welcome you, sir. The organizing team also takes pride in welcoming Mr. Suresh Samuel, the member board of management, BMRF, who is a man of great insight and the one who has always encouraged us to come up with more innovative ideas and support us. And welcome, sir, in absentia. Now it's my pleasure to extend a very warm welcome to our beloved dean, Dr. Rakesh Sehgal, sir. He is actually known as an embodiment of simplicity and efficiency. Sir was there as a strong pillar of support in each and every step we took from the planning stage to the execution today. We bow before you and welcome you, sir. Now I would like to warmly welcome our Tamil Nadu Medical Council observer, Dr. T. Prasad, who is already logged into it. Welcome, sir. We cordially invite our most respected provost, Dr. P.F. Kotur, who is a man with unabated energy and enthusiasm. He was a great pillar of support for BMSECON team in the last 3 years with his encouraging words and unexplainable support. Welcome, sir. I welcome our super energetic and dynamic Dean Health Professionals Education, Dr. V.N. Mahalakshmi ma'am, who was very supportive and was kind enough to offer help at any time we have approached her. And even without approaching her, she has offered help, especially regarding the technical support. We wholeheartedly welcome you, ma'am. We also welcome all the honored guests present here today, Dr. Vishnu Bhatt, Advisor Research and Publications, Dr. Satish Kora Kuruvilla, Medical Superintendent, Dr. Jay Singh, Director, Health, Hospital Growth and Outreach Services, Dr. Lakshmi Jatia, VP Academics, Dr. Rajan, VP Admin, and Dr. Lata, VP Academics, Dr. Arunachalam sir and Dr. Shankar, who are the DMS, and our outspoken and eloquent resource persons and chairpersons who have joined us, deans of other medical colleges under VMRF, head of the institutions of other colleges in AVMC campus, university officials, members of the STB, HODs of different various departments, faculty members of AVMC, and friends from the present media on this auspicious occasion. We welcome our, before that, a very special welcome to our two vice principals, VP Admin Dr. Rajan and VP Student Affairs Dr. Lakshmi Jatia ma'am, who are actually my predecessors as organizing secretaries for BMSECON for the last 3 years. Both of your guidance and support was really appreciable. I really welcome you both to this conference. Now, I would like to extend my warm welcome to our most enthusiastic delegates who are joining us from far and wide. Without your support and encouragement, this conference would not have been possible. We sincerely hope you will enjoy today and the next 2 days of deliberations and networking. I once again welcome you all to the inaugural ceremony of BMSECON 2023 and a special welcome to our chairpersons of the previous session, Dr. William Stringer sir and Deepak sir, who sir has given the keynote address last year also. A special welcome to you, sir, for joining our inaugural program. And I wish all of you a great learning. Thank you.
Thank you, ma'am. As we pursue knowledge, the radiance of illumination serves as a beacon, guiding our endeavors. I humbly request our esteemed dignitaries to partake in the virtual illumination of the lamp. This ceremony solemnizes the special occasion, embodies shared wisdom and collective spirit.
शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्नमोऽस्तु ते दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपज्योतिर्परब्रह्म दीपज्योतिर्जनार्दनः दीपो हरतु मे पापम् सन्ध्यादीपनमोऽस्तु ते ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः ओ दीपब्रह्मरूपस्त्वं ज्योतिषां प्रभुरव्ययः आरोग्यं देहि पुत्रांश्च मतिं स्वच्छां हि मे सदा
Thank you. I now request Dr. T. Rajan, Vice Principal Admin AVMC, to enlighten the gathering on the history and growth of BMSECON.
Thank you, Dr. Aarti. Do you able to hear me?
Yes, sir.
Okay. Respected dignitaries in main hall and delegates at auditorium, very pleasant morning to all. I, Dr. Rajan, Professor of Anatomy, Vice Principal Admin, Arupadi Medical College, Puducherry, affiliated to Vinayaga Missions Research Foundation, Salem. Thanks to the organizing team for giving me the wonderful chance to talk about this history and growth of BMSECON in this wonderful academic event. BMSECON, Basic Medical Science Conference, is a modified PonCon, that is Pondicherry Conference. Let me share the slide for a better view on this. Can you give me access to share?
Yes, sir, please share.
Okay, thank you, sir. Do you see the slide, Dr. Aarti?
No, sir, it's only the back.
Yes, sir.
Okay, right. In the year 2003, within the 2 years of establishment of our institute, Dr. K.R. Srinivasan, who was the head of the Department of Anatomy, organized state-level conference jointly with Association of Anatomists. That was the first conference organized at AVMC. Dr. K.R. Srinivasan organized again a state-level conference jointly with Association of Anatomists in 2007. Both were a grand success with support of our management. Our management provided free accommodation for all delegates at the time because the conference was physically organized. That was the talk at the time when I was assistant professor in AVMC. There was a long gap after that conference. In 2018, myself and a new energetic team decided to organize international conference and organized the anatomy conference. It was a grand success. It was possible because of our management.
Yours was not visible, sir.
You are not able to see the slide? Okay.
Yes, sir.
Again, share it. Do you see the slide?
Sir, into full
The
screen.
first slide is there, sir.
Okay,
It's the first slide. Yes.
right.
Yes, sir. Yes.
Yes. Okay. So this is the conference which we organized physically. It's a first international conference. Till this, the conference named as PonCon, that's Pondicherry Conference. By seeing this success of all these conferences, we have decided to extend this conference with other department, anatomy, physiology, and biochemistry, as well as Center of Biomedical Research. So we have planned for a physical international conference named as a BMSCON. Due to COVID, we could not do it as a physical conference. So we have organized it as a e-conference. So it was named as a BMSECON. That's the first e-conference organized by the all the 3 basic medical science department, which was organized by myself. Then in 2021, we planned again. Due to second wave, again it was organized as a e-conference. It was organized by myself and the team. Later, by seeing the request from the delegates to continue this conference as an e-conference, so we have decided to continue to organize this conference as an e-conference. Dr. Lakshmi Chatia, who is the professor and the head of the department that time, and now VP Students Affairs, organized this third conference.
Again, your slide went off, sir.
Okay.
It went to the first slide, sir.
So I think there is I'll come for that.
Yes, sir. Yes, sir.
Yeah. Okay. So in 2022, Dr. Lakshmi Sati organized the conference. It's e-conference. Now we are the 4th e-conference, which is organized by the same team, but the organizing secretaries keep on changing. Now the organizing secretary is Dr. Manju, professor in the data department. This is about the PonCon, how it has become BMSCon, and how it's become a BMSECon. Now here you can see some of the glimpses of our conferences, our chancellor address, our provost address. These are the organizers. Souvenir release and our volunteers, students culturals, our resource persons, panelists, award to the resource persons and chairpersons, group photo virtual, chit chat by our faculties, post-event celebrations. Thank you. Thank you.
Thank you, sir, for taking us through the years of BMSECON. I now request Dr. Lakshmi Chatia, the Vice Principal, Student Affairs and Professor of Physiology, to address this gathering on the theme of the conference.
Thank you, Dr. Aarti. The topic, the theme which was aptly chosen as crossing boundaries, excellence in basic medical sciences. Excellence in basic medical sciences refers to achieving high standards and significant advancements in the fundamental scientific disciplines like anatomy, physiology, biochemistry, microbiology, and pharmacology that form the basis of medical knowledge. Scientists and researchers across the globe are striving for excellence in conducting rigorous studies, making groundbreaking discoveries, and contributing to the foundational understanding of the biological processes essential to medicine. Indicators for this quest of excellence include publications in high index journals, innovative discoveries that showcase a commitment to advancing knowledge, and also mentoring the next generation of scientists and medical professionals, and collaborations. Engaging in meaningful collaborations with other experts and institutions can enhance the overall impact and quality of research. Success in securing research grants and fundings, and that also indicates trust and support from the scientific community and the funding agencies. Receiving awards, accolades from peers and professional bodies is another testament to one's contribution and impact in research. Translating these basic medical sciences discoveries into practical application for medical diagnosis, treatment, and prevention demonstrates the relevance and excellence of this research. And this also comes through continuing education, continuous learning that we do through our continuing medical education programs and conferences of this kind. So I summarize, overall, excellence in basic medical science transcends geographical boundaries and contribute to global healthcare advancements. Thank you.
Thank you, ma'am. May I now request our Honorable Vice Chancellor of VMRN, Dr. P.K. Sudhir, the epitome of excellent leadership, to declare this conference open and address the esteemed gathering.
Can you hear me?
Yes, sir.
Can.
Yes, sir.
I declare this conference, Basic Medical Science Conference 2023, open. Respected Dr. Deepak Sarkar, the distinguished chief guest of today's function, Professor Rakesh Sehgal, the Dean, Professor P.F. Kotur, our Provost, Dr. Mahalakshmi, Dean HPE, Dr. Rajan, Dr. Lakshmi Jetia, the Organizing Committee Chairperson, Dr. Ilankathir, the Secretary, Dr. Manju, distinguished resource persons from India and abroad, the delegates from all over the world, chairpersons of the sessions, ladies and gentlemen, a very good morning to you all. I'm extremely happy to note that this fourth International E-Conference on Basic Medical Science 2023, organized by the Department of Anatomy, Physiology, Biochemistry, and Medical Biotechnology, attracted 800 plus delegates and around 240 scientific presentations. At the onset, I congratulate the organizing committee, the entire organizing committee, and the dean for this terrific effort. Medical education, since last 1 decade in India, is undergoing a series of transformations. In 2019, National Medical Commission has come out with a competency-based medical education, and the first batch is undergoing the final-year training now. The best attraction of the new regulation is early clinical exposure, the gelling the departments of the basic medical sciences and the paraclinical departments and the clinical departments. The Department of Anatomy, Physiology, and Biochemistry has a greater role in shaping our physicians and surgeons in medical education. I am sure this type of conferences, both physical mode and online mode, have a serious impact among the faculty members and students. Vinayaga Mission Research Foundation deemed university always encourage our constituent colleges and schools to organize conferences and workshops and focusing on creation of knowledge, dissemination of knowledge, and application of knowledge. I am sure these 3 days deliberations on various topics, especially on the theme Beyond Boundaries, Exploring Excellence in Basic Medical Sciences, will have a serious takeaways and a very extensive and elaborative deliberations on various topics. Thank you. Thank you very much.
Thank you, sir. It is with great enthusiasm that we have prepared the souvenir, a conglomeration of scientific knowledge shared in this conference, blessings and messages from our patrons, administrators, and university officials. It's my privilege to invite our beloved Dean, AVMC, Dr. Rakesh Segal, who is our pillar of strength and support, to release the souvenir.
Good morning, everybody. As our Honorable Vice Chancellor has said, that this is we have got a gathering of a number of scientists, and a number of 800 plus is a huge gathering, I could say, even for an econ. And I congratulate the organizers for having such a scientific gathering today in this conference. And this is all with the blessings of our Chancellor and Madam Chancellor and Mr. Samuel, who have been really encouraging. And I found that it is really encouraging to see them and our Honorable Vice Chancellor, that they are encouraging even conferences, research, and other activities in the college. So this is really wonderful thing which I have seen in this VMRF. And I would congratulate the VMRF that for this, because I have come from PJ Chandigarh, and I see that the activities are almost as par at what we will see in our institute also. And they are also giving a lot of research funding at their level, which is also very encouraging. So today's conference, I think, as you all know, that it is basic medical scientists who are organizing this conference. And this includes, well, 3 departments mainly, but it is all a teamwork of AVMC, and all people have really pitched in. I am really grateful to all the help that has been given to organize this conference from all areas of AVMC and all staff of AVMC. I'm really grateful to them also. Now, as you know, that they have announced that there are around 240 abstracts which are to be published, and the abstract book is to be released. And I think I had released this abstract book on behalf of the organizers and on behalf of AVMC and on the behalf of VMRF. Thank you very much.
Thank you, sir, for the honors. May I now kindly request our beloved Provost, Professor P.F. Kotu, an unwavering pillar of strength and steadfast support, to share a few words with us and impart insights and inspiration to mark this special occasion.
Sir,
Sir, please
So you
unmute
Please
yourself.
unmute.
Yeah, sorry. A very good morning to all the distinguished personalities over here on the virtual dais, the Chief Guest, Dr. Deepak Sarkar, our Honorable Vice Chancellor of our prestigious university, Professor Sudhir, the respected Dean of ABMC, and my colleagues, the Vice Principals, Heads of Departments, and the resource persons and the participants. I am the first-hand witness of the evolution of this BMSECON for the past, almost this is the 4th year. I distinctly remember in the post-COVID era when we all were feeling claustrophobic, the trio of 3 departments along with the our research center conceived this idea. Professor Rajan, Professor Lakshmi Jatia, and I distinctly remember Dr. Mishra, Sashmita Mishra, who is of course not with us, and now her place is taken by Manju. We conceived the idea, and initially I remember very well we were waiting, finger crossed, whether how will be the response. And when the registration crossed 4 figures, I remember we were overwhelmed. And it was such a grand success, it was and first experience as well, which laid down the foundation and for the conduct of consecutive 4th conference. Now I'll say BMSECON has become the institutional character of AVMC. And certainly we all know that research and academics are not, cannot be separated. They are the 2 sides of the same coin. One will complement each other. Keeping this in mind, the impetus that is being given by our university under the leadership of our Honorable Vice Chancellor, our Chancellor, and the research along with academics is encouraged in all possible ways and at all possible levels. Apart from creating the infrastructure for research, etc., the capacity building in research, how to use the research, not doing is only not enough. All those things will be possible if we conduct such a platforms wherein the knowledge, expertise, skill of the persons who have developed expertise in the field will share, and that will be a very much guiding force for all the participants, especially the younger generation. And I take this opportunity of welcoming all the respected resource persons who have spared their time. I very well understand the difficulty of participation by the overseas resource persons. It might be sometimes an odd time for as well. So ultimately, it is for the growth and development of this one. And our again, I used to say that the basic medical science department and our research are the pillars of our AVMC, which lay down a strong foundation for the product which we are trying to bring out at the conclusion of the course, the medical doctor. So with these few words, I'm very, very happy to be a part of this wonderful, excellent function. And I'm sure the memories of this BMSECON will be cherished forever in the years to come as well. Thank you very much.
Thank you, sir. Thank you so much. May I now request our dynamic and inspired Dean HPE and Dean Faculty of Interdisciplinary Studies, Dr. V.N. Mahalakshmi, to share her thoughts with us. Her leadership and vision have been instrumental in shaping our academic landscape.
Thank you, Aarti. Good morning, respected speakers and the dignitaries who are sharing this platform with us. It's indeed a very proud and nice moment to be with you all for the 4th consecutive edition of the BMSECON. And this particular platform has attracted PhD scholars, UG students, and postgraduate students to come and share their research opportunities, not only the esteemed research persons. So I think that is the USP of this particular program, that we are able to attract people from the specialty who would come and be part of this event and share their own research work. the number of papers which we receive every year for the scientific session, that they bears a witness to this particular fact. That is the outcome we have received. And this year I was told they have more than 400 scientific paper presentations, which is a like mind-blowing number. That's all I could say. And the departments have put in a lot of effort to ensure that this program goes in a smooth fashion. They have contacted research persons of repute. They have got eminent chairpersons to be in the session, and they have taken care of the technical and participant side also. So with all these efforts, I think this program, no, like Lord Krishna keeps telling, that if your process is all right, outcome will come automatically. The process has been all right because it is going on for some time. So the outcome also should be very good. I wish them all success, and thank you for making us being a part of this event. All the best. Thank you.
Thank you very much, ma'am. We are very honored to have with us today our chief guest, Professor Deepak K. Sarkar. Right before the inauguration, Professor Deepak K. Sarkar graced us with his insights in the much anticipated keynote talk and set the tone for this enlightening event. Dr. Deepak K. Sarkar is Distinguished Professor, Director, Endocrine Program, Department of Animal Sciences, Rutgers University, USA. He is Director, Alcohol and Drug Abuse Program, Washington State University. He has more than 300 publications. His exceptional contributions have earned him numerous awards and accolades, both from the Indian government and American universities. So we are very happy to have you with us, and we request you to address the gathering.
Sir, unmute your mic, please.
Okay. I'm okay, right? Can you hear me now?
Yes, sir.
Thank you so much, so very much for inviting me here. I think this is the first time I attended this, and I've been very delighted to see the group of devoted educationists and scientists are putting a gathering which could really bring the young mind and open up the young mind to build a new era in the scientific field in India. So I really like whatever you're doing, and I think the reward is already coming because you already have more than 800 participants, close to 300 abstract. I mean, that's a good number, very good number for being a e-Congress. So this shows that the organization team is putting an outstanding kind of contribution here, outstanding kind of group building. So I really appreciate for giving me a chance to come and talk and learn these things, and in future probably we'll interact, and I wish you best of luck. Thank you for the opportunity.
Thank you so much, sir. Thank you for gracing this occasion with your presence. I now request Dr. S. Ilankadar, Professor and Head, Department of Anatomy, to propose the vote of thanks.
Good morning, one and all. Myself, Dr. Ilankadar, Organizing Chairperson, feel humble and privileged for being given this opportunity to present the vote of thanks. Thank you, team BMSECON2023. I thank the Almighty for the blessings showered upon us for the ever success of our conference. My sincere and heartfelt gratitude to our founder chancellor, late Dr. A. Shanmugasundaram sir and Annapurni Shanmugasundaram madam, who has given us this platform for conducting this mega conference. On behalf of our university, institute, and the organizing team, I would like to thank the Honorable Chief Guest for today, Dr. Deepak Sarkar sir, Distinguished Professor, Director of Endocrine Program, Department of Animal Science, Rutgers University, USA, for his guest lecture. Sir, you have been readily accepted our invitation and granted us your valuable time amidst your busy schedule to enlighten us in identification of a new mode of cell-to-cell communication within the nervous system, focusing on neuroinflammation. It was a wonderful session, sir. Thank you. I also would like to thank Dr. William Stringer and Dr. K. Deepak, sir, for chairing our guest lecture for today. You have given your valuable time for us. Thank you, sir. I would like to thank our beloved Honorable Chancellor, Dr. A.S. Ganesan, sir, in absentia, and Dr. Anuradha Ganeshan, Madam, Vice President, VMRF, in absentia. Together, they have been behind the success of all the academic programs conducted in our institute. Thank you, sir. Thank you, ma'am. Though you are far, your best wishes are always with us for the success of this conference. My sincere thanks to our beloved Vice Chancellor, Dr. P.K. Sudhir, sir, who have always showed interest and supported us in conducting our conference. Thank you, sir, for declaring our conference open. I also like to thank the other VMRF administrators for supporting us in organizing this event. On behalf of our organizing team, I would like to extend my sincere thanks to Dr. J. Suresh Samuel, sir, member, Board of Management in absentia, the drive force behind all the academic events in our institute, for his constant encouragement and support in organizing this conference. We thank you, sir. My sincere thanks to our beloved Dean, Dr. Rakesh Sehel, sir, for his constant support in organizing this conference and for his presidential address and releasing our E7 year. Your support has given us a framework upon which we have planned and executed this program. Thank you, sir. Sincere thanks to our Provost, Dr. P.F. Kotur, sir, and our Dean HPE, Dr. V.N. Mahalakshmi, Madam, for their felicitational address and for their constant support starting from the preliminary stage until this date. Thank you, sir. Thank you, ma'am. My sincere thanks to Dr. T. Rajan, sir, Vice Principal Admin, AVMC, for making us to recollect the path we walked through to reach where we are today through history and growth of our conference. Thank you, sir. On behalf of the team, I extend my thanks to Dr. Lakshmi Jatia, Vice Principal, Student Affairs, on enlightening and briefing us on the theme of our conference. Thank you, ma'am. My sincere thanks to all our advisory members, Advisor Research and Publication, Dr. Vishnu Bhatt, sir, Dr. Jai Singh, sir, Dr. Satish Kora Kurivilla, our Medical Superintendent, and Vice Principal Academic, Dr. Lata, our Deputy Medical Superintendent, Dr. Arunachalam, and Shankar, sir, and other administrators for their constant support. I also would like to thank the heads and staff of our other colleges under VMRF for their valuable support. My sincere thanks and heartfelt gratitude to our esteemed speakers, chairpersons, and judges who have consented to support us in this venture. Thank you all. My sincere thanks to our TNMC observer, Dr. T. Prasad, sir, for being with us today. I thank all our enthusiastic delegates who have readily spared their valuable time to show interest, register, and to participate live today and make our conference a benchmark in history. Thank you all. Thank you, one and all.
Thank you, sir. That brings us to the end of the inaugural session. And we thank the dignitaries and everyone who's joined us. So shall we conclude with our national anthem? I request everyone to raise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जगति तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
We thank everyone once again, and we request everyone to stay logged in for the scientific sessions, which will begin right away. Thank you.
Team care. Am I audible?
Yes, ma'am.
Okay. Please check whether Sucheta ma'am is inside. She's a panelist. Sucheta, Dr. Sucheta is there. Has she been made a panelist? Okay, ma'am. Ma'am, can you switch on the video, ma'am?
Switch on the, yes. Switch on the video. Yes, madam. Good morning.
Good morning, ma'am.
Good morning.
Okay. We'll start, ma'am, now.
Yeah. Shall we start sharing the slides?
Wait, ma'am. We'll just introduce you, then we'll.
Okay.
Okay, so yeah. So we are starting our next session, scientific session. It is by Dr. Sucheta ma'am. She'll be introduced by our chairperson. First, let me take the honors of introducing our chairperson for this session, Dr. R. Shashikala. She's a gynecologist by profession. She's a professor of obstetrics and gynecology in Arbade Video Medical College and Hospital, Pondicherry. She's retired as a professor and HOD of OG in Government Deni Medical College, and she has worked as professor and HOD in Sri Manakula Vinayagar Medical College Hospital, and at present she's in our hospital. She's a past Senate member of the Tamil Nadu Dr. NGR University. She has published papers in national and international journals, many. She's a very well-known teacher and a UG and PG examiner in Tamil Nadu, Kerala, Andhra Pradesh, as well as in Karnataka. She's a past member of the PG Board of Studies of the Pondicherry University too. With this small introduction, I hand over the stage to Dr. Shashikala ma'am. Ma'am, over to you.
Thank you, Manju. I'm privileged, honored, and feel proud to introduce the speaker of today's session, who is an eminent professor, a renowned academician, and a research scholar, Professor Suchitra Kumari, Professor, Department of Biochemistry, Hegde Medical Academy, Bangalore, Karnataka. She is also the Principal Scientific Officer, Central Research Lab. She has 3 decades of teaching experience and highly acclaimed researcher for the past 2 decades in the subjects of biochemistry, molecular genetics, cytogenetics, immunology. She has published in many national and international index journals, and she has high index more than 42 plus H index. She is a dynamic leader with extensive research expertise and experience as principal investigator and co-investigator in managing several international and national funded projects. She has 10 ICMR STS projects. She has authored a book chapter of multidisciplinary research methods. She has patented pomegranate herbal mouthwash for managing radiation-induced oral mucositis in patients with head and neck cancer. Her areas of interest include biochemistry, molecular genetics, cytogenetic, biodosimetry, immunology, radiation biology, nutrigenetics. She has been an invited speaker to 3 international universities like Ajman University, United Arab Emirates, Indonesia, Bali, and University of Miyazaki, Japan, and many national universities like SRM Chennai and Reva University, Bangalore. She is a visiting scientist in Wake Forest University, Johns Hopkins University, and Penn State University, USA, and University of Miyazaki, Japan. She has received many prestigious awards like Women Researcher Award, Most Cited Paper Award, Fellow Lifetime Achievement Award, Best Oral and Paper Presentation, Best Researcher Award, and All India Women Achievers Award of 2022. When I go through her CV, I feel she has set her bars higher and higher. Today, She is going to sensitize us regarding the impact of genetic, diet, and environmental factors on polycystic ovary syndrome. This polycystic ovary syndrome, which starts from the intrauterine life, a fetal origin of an adult disease. It is caused by many factors and it is controlled by various genes. Because of the altered lifestyle, the dietary habits, and the adolescent obesity, and the gut microbiomes, dysbiosis occurs there. And because of that, there is an increasing incidence globally of the incidence of PCOS. This is the only condition which involves many branches of medicine. The lady will come to us with AUB, abnormal uterine bleeding, infertility, and failure of reproduction. And for the metabolic syndrome associated with it, such as diabetes, hypertension, dyslipidemia, and coronary heart disease, and the fatty liver disease, she seeks the opinion of the physician and endocrinologist. To reduce her weight, she goes to the dietitian, and because of the frustrating acne, hirsutism, and loss of hair, she seeks the dermatologist's opinion. Because of depression, anxiety, because of these factors, she seeks the psychologist's opinion. So, left untreated, she may land in endometrial hyperplasia and adenocarcinoma of the endometrium. Finally, the oncologist also they come to play. So, it is a disease which the syndrome starts right from the fetal life, spans through the adolescent period, the reproductive age group, and even beyond the age of menopause. And as a gynecologist, I feel excited to listen to this lecture. Now, this screen is yours, madam.
Cheta ma'am, you can share, ma'am. Ma'am. Sucheta ma'am. Sucheta ma'am, am I audible?
Hello.
Yes, ma'am. You can
Yeah,
share the
yes,
screen,
yes.
ma'am.
Yeah, you are audible. Thank you very much. Yeah, we are sharing. Good morning to all of you. I am very grateful to all of you to given me the opportunity to present some of my work on PCOD. First of all, I thank the chairman. It is my pleasure to sit in front of the renowned gynecologist who knows everything about PCOD. We are researchers. We have taken this opportunity to screen out just what are the other factors affect on the PCOD. So especially Manju madam, who really given me opportunity to talk about this particular topic. This is one of the very, very important topic nowadays. So my more than 5 students are working on PCOD. Some are completed their work. So I thought environment and the genetic factor effect on the PCOD. So coming to introduction, I think not much introduction is required because madam already mentioned there are so many factors affecting on PCOD. It is a complex, common metabolic and heterogeneous condition affected women of childbearing age. So it starts from 18 to 40 years. There are a lot of people are suffering from PCOD due to so many reasons. One of the common reason is diet. Second reason is environment. We are not giving much importance for diet and environment, which we land with the PCOD problem. once we get affected by the PCOD, then different types of metabolism is changed. So we'll come to the PCOS develops when the hormone, that is LH from pituitary gland, or the levels of insulin from pancreas are high, which in turn causes the various to make extra amount of testosterone. Testosterone is a male hormone. So male hormone increases here and female hormone decreases, and they also develop insulin resistant. But the major cause here for
Hello, ma'am. Hello, ma'am. She's
differ. So I'm all of you hearing me properly?
Yeah,
Yes,
ma'am.
ma'am.
Please share your slide again,
sir,
ma'am.
your slides are yes.
Yeah, we are sharing. Some problem with the internet. Okay.
Please
Go ahead,
put in
ma'am.
slides on.
Okay. So PCOS affect 1 in 10 women, incidence of, because for example, those who are near the seashore cities, the exposure of various pollution is less compared to when we go away from the Mangalore. For example, Bangalore city, it is away from the seashore. One of my students is working from Bangalore. He gets PCOD sample like anything, but Mangalore we find difficult to get that level of PCOD patients. So that when I thought why can't we think about various toxins exposure to the women? So we got a very good result associated with this work. So as factors affecting, the genetics, epigenetics, when we exposed, when our lifestyle changes, when food pattern changes, when exposed to environment changes, there is a epigenetic changes. When there is epigenetic changes, it may alter the genetic expression for various production of the hormones, or the requirement of various proteins or enzymes, all that. Then in addition to that, when the carbohydrate accumulation increases, immediately they increase a lot of inflammatory markers. So the inflammatory markers is one of the factors which is related to all types of metabolic syndromes. So when we see in PCOD subjects, they express insulin resistant one side, hyperandrogenism another side, and another side genetic defect because of epigenetic changes. So all these factors we observed in our studies. So these are the various polycystic syndrome, etiology, clinical features, diagnosis, and risk factor associated PCOD, already hormonal, everybody studied very well. Genetic, also there are a lot of work is going on. And non-genetic is environmental toxins, obesity, and diet. And then PCOS, if you see the environmental exposures in the life, that is hyperandrogenism, you see ovarian dysfunction, genetic changes, transgenerational effect. That is also, we can know, obesity, environmental factors, especially how it affect on the endocrine disrupting chemicals, miscellaneous like diet, you can say all these types of pharmaceutical agents, vitamin D, psychological, all that changes. Epigenetic, we can observe with the microRNA assessment, DNA methylation, abnormal gonadotrophic hormone, all these changes we can observe in a PCOD patients. So environmental, bisphenol, this is one of the very important compound is exposed in the air due to the vehicle smoke and other smokes. See, that will affect all the bisphenol is catabolism decreases, it increases in the circulation. And this is the one factor which block the hormonal production. So it increases the testosterone level, estrogen decreases. So this if you observe NAFLD, non-alcoholic liver disease, this is another very important factor. When the carbohydrate intake or the sweet intake or junk food intake increases, it will accumulate in the liver. The carbohydrate convert into fat and accumulate non-essential fat. So the sex hormone binding globulin, SHBG level decreases. This is another one of my students did SHB level in the PCOD samples. So there also we observed all the factors, how it impact on the metabolism of various diet patterns. So this is the dietary intake and environment pollutants, sedentary lifestyle, stress on the here what we have to observe, everybody knows, doctors knows, scientists, that is the insulin resistant. It's a common problem in now in everywhere in whole world. Insulin resistant, usually we never bother to know the resistant problem. The major problem is we are blocking entering the glucose inside the cell for metabolism. So the glucose is not entered, in the insulin is there, but is not used for the entry of glucose. Maybe the cell membrane is filled with the, that is the bilayers of lipids, maybe the non-essential fatty acids, not the essential PUFA. So the fluidity of the membrane decreases. So sugar level increases in the circulation. Automatically insulin production increases in the circulation. So this insulin level is high, sugar is high, they will convert into fat and accumulate in the liver, and the fluidity of the cell membrane decreases. So that is the one of the very major problem develops in the PCOD or diabetic, which we call insulin resistance. So modification of diet is very important. We have to supplement with short chain fatty acids, omega-3, and decrease the sugar, especially free sugar, decrease completely carbohydrate content. You have to reduce, increase the diet, and we have to maintain the normal requirement for the various metabolism. So these are the various problems with the hormonal imbalance, which already we know, the hypothalamic pituitary ovarian axis, the important pathophysiology underlying the PCOS because of all these reasons. And it is estimated that between 50 to 90% women PCOS manifest insulin resistant. This is the insulin resistant is one of the major problem in the PCOD. And these are about the hormonal connection synthesis. Then we also have the cytochrome P450 level. Cytochrome 450 is a generic term for a group of oxidative enzymes, all of which have about 500 amino acid and contain a single heme group. Human genomic includes genes for 57 cytochrome P450. These genes are now formally termed as CYP genes, and encoded proteins may be given the same name without the use of italics. So CYP11A1 gene encodes P450, where the suffix, this is one of the gene which is essential for the control of the PCOS. So there are two genes we have analyzed here, genetic variations. One is a CYP11A1 gene, cytochrome P450, side chain cleavage of the enzyme of the cholesterol synthesis, and CYP19 gene, it is aromatase enzyme. So these two genes we analyzed in the PCOD samples. Yes. So these two genes we have identified where which chromosome it is located and all that in detail. So we are study conducted with the all these different. One is CYP11A1 gene variation and compare with the sex hormone and insulin resistant. Association sex hormones binding globulin and abdominal obesity in patients with polycystic syndrome we studied. Non-pharmacological strategy in development awareness and intervention of polycystic ovarian syndrome we studied. Association of vitamin D gene receptor gene polymorphism in polycystic ovarian we studied. And now our ongoing project is gut microbiota derived short chain fatty acid as a regulator of hypothalamic pituitary ovarian axis in polycystic ovarian syndrome. This is the one of the very good result we are getting. Etiological role of toxic chemicals. This is another very important work is going on. And heavy metals on aromatase and its expression in polycystic ovarian syndrome. Effect of ghee on letrozole-induced polycystic ovarian syndrome model in Western rats we studied. Association between gut microbiota dysbiosis and polycystic ovarian syndrome. Effect of polyamines. This is another very, we never given importance that the polyamines production is very important. Polyamines on the expression anti-Mullerian hormone expressed by granulosa cells. Screening for putrescine in women with polycystic ovarian. These are the work is going on now. And where I just give you a gist, in microbiota short chain fatty acid like butyric acid is one of the short chain fatty acid which crosses the blood-brain barrier. And this is the one which regulates the hormone production and also enhances the lifestyle changes in the PCOD patients. And second one, we have toxic chemicals. You may be surprised to listen which toxic agents are effect on PCOD. One is the moisturizer. Most of Bangalore, it is a slightly cold weather. They use a lot of moisturizer. So PCOD patients are very high in Bangalore. One is the moisturizer contain chemicals. Second one, exposed to the bisphenol. Third one is the aluminium. What we found is when we analyze for the toxic metal ions, lead, arsenic, cadmium, chromium, aluminium is the one of the mineral which is very high in PCOD samples. So which we are ignore so many things, but we are not concentrated on that part. Then we have given the ghee. This is, I think nowadays it's a known factor. All gynecologists and also in Ayurveda, they will supplement ghee, which will reduce the PCOD. And microbiota, the intestinal microbiota, this is another very important factor. Actually, if you take a lot of dietary fiber in your diet, it is the base for the growth of the probiotic in the intestine. So we have to increase our dietary fiber so the probiotic bacteria increases in the intestine, and they are the one which gives all the micronutrients, mainly the short chain fatty acids, vitamin B12, folic acid, vitamin K, E, so many very, very important micronutrients are produced from the probiotic. So that is one which controls, which by taking all the junk food, all the fried items, or the lot of carbohydrates, the whole probiotic is completely vanished, and toxin bacteria are producers, 1001 toxins, which leads to the various type metabolic syndromes. Then one of our group, we were planned to, by mistake, we have estimated some of the polyamines like putrescine, spermidine. We observed in PCOD, it is completely decreases. When we accelerate, when we see the changes by modification of lifestyle, polyamine production increases. So this is also one of the outcome of our project. So these are the, we have taken ethical consideration, sample calculation, study design, study setting, duration, all that we have practiced, inclusion, exclusion criteria we have taken. So then we have done the experimental workflow for the collection of DNA, analysis, the sex hormone. We have a fluorescent ELISA method. We used all the hormonal analysis, and we estimated blood glucose, lipid levels, and molecular genotyping. We did separate DNA, and we screened, and Sanger sequencing, we found the SNPs. So many work our PhD students are doing on PCOD. So these are the basic baseline characteristic among study group. Also, we observed their BMI is very high compared to the control we have observed, and our marital status, married women showed most of the PCOD compared to the normal. So then when we clinical characteristic also we observed, there are 32% weight gain observed, and 54 oligoamnuria patients are we observed, subjects, sorry, and 46 primary secondary infertilities out of the 400 samples, and some combined effect also we observed. So in this current study, we observed that PCOS was more common among the reproductive age women, especially between 25 to 30 years. Most of the PCOS women in the present study were overweight according to the characterization of BMI. This highlights the prominence of obese females in Karnataka, in South Asian females have early manifestation with more severe symptoms, including diabetic, hypertension, cardiovascular disease, and other related metabolic disorders. When we compare with the articles. When we compare BMI also, we observed the same. Since abdominal obesity considered to be related to women having reproductive issues, we have the waist-hip ratio also increased. So then when you see the sugar level with the compared to, it is not very high, but compared to the control, it is high. Total cholesterol is almost not very changed, but triglyceride level significantly high. So these are our observation on lipid profiles and sugar profiles. Then plasma insulin. If you see the plasma insulin level, we observed the HOMO-IR is in the PCOS is high. Significant increase of insulin and HOMO-IR we observed. And when we compared with the other article also, we found the same report. Then when, as usually, we know the different hormonal changes with the gonadotropin and sex hormone among women, we observed significantly low levels in the PCOS patients. So these are the articles we compared, and then we also analyzed the CYP11A1 allele frequency. We observed the changes in the genetic expression of the SNPs. So here the CC has more observed that genetics have consistence of PCOD, and the TT, the recessive alleles are not much showing the differences. So this is what we observed in the PCOD samples. Then these are compared with the various parameters with the different genetic variations. So our study, predominant pattern, mutant carrier allele type, both in case and control, but in the northern Indian region, Punjab, the predominant polymorphism pattern observed for the RS 4077582 of CYP11A1 in PCOS was the mutant carrier allele type, and for control pattern observed was wild type. But our report, when we see the association of another CYP11A1, we compared with the lipid profile BMI. So these alleles are associated with the hip ratio significantly. You find in all the allele it increases. Triglyceride level is significantly associated with the these particular allele. So like that, even it affects on the hormonal changes, FSH, LH, and LH-FSH ratio. So these are our observation. We compared with the various dominant model, also recessive model. We compared the various parameters, lipid profiles, BMI, and hip ratio. So inadequate secretion of gonadotropin are an important characteristic feature of PCOS. One of the most common reason for PCOS that is the LH level. Individuals with the different genotypes may react differently to LH surges due to differences in LH level in different genotype. Levels of LH in the current study were significantly different among the three genotypes of the PCOS, with a higher LH level in the CT genotype when compared to the CC and TT genotype. So here, real clarification we need. Still, we have to work with the PCOS sample with the different varieties of parameters affecting how epigenetic changes takes place, how the genotype effect on the PCOS. A lot of study we can do further. So as the study conducted on Chinese population reported similar findings, but with the control population stating that they were more likely to develop PCOS due to the abnormal internal secretion of LH, particularly those women of CT heterogeneous genotype. So various changes we observed in different populations. Interesting, in our population, control women showed no significant difference in LH level among the three genotypes. But genotype is there in both, but we are not observed in the control. Maybe it is no epigenetic changes appeared in that particular control. In the present study, significant difference were observed in the pattern of FHS. Women with PCOS where a lower FH level was observed in the TTM. So overall, what I'm going to try to tell you is the genetic variation also very, very important. We are not further studied. We just observed in the allelic changes and the mutation observation compared with the hormonal changes. But further we can control by doing the epigenetic changes. This is one of my student was doing heavy metal toxicity on PCOS at Bangalore. What he observed is, in his study, he observed data exposure to toxic environments such as traffic, cosmetics, food, etc. obtained through the valid questionnaires, and levels of heavy metals such as aluminium, cadmium, lead, arsenic, nickel were measured. And they are observed the aluminium level is very, very high in the PCOD samples. So interesting, it was observed that aluminium was significantly high. So which we are not bothered much. What are the reason, whether due to water, whether due to the vessels, all that. In addition, this age group using lot of moisturizer were significantly high in the PCOS. So the chemicals which present in the moisturizer is one of the reason to develop the PCOS. So the third point, our diet, whether does diet affect polycystic ovarian syndrome? So high sugar, especially children age from 8 to 13 or 14, 15, they take a lot of ice cream, sweets, and different types of chocolates and so many types of sugar. Actually, we are born like a ape where we used to eat only nuts. We never used to eat the carbohydrate or cereals. When the ape become men's, the human beings, that time we, from the trees, we came to the ground, and easiest available is the cereals. We are like addicted to the cereals. So because it's just like a drug addict, any addict, we are so addicted to the cereal. So the cereal intake increased, so that our life, our cell started with the cell membrane bilayers of lipids. Our brain is full of lipids. Like lipids are the major requirement for the maintenance of the body function or the cellular level. We are replaced with the carbohydrates, where it convert into the saturated fatty acid, and it decreases the mobility of the membrane or fluid mosaic, fluidity of the membrane. So we are prone to get all different types of metabolic syndrome. So this is my conclusion on PCOD. That changes in the CYP gene changes is one of the factors, maybe epigenetics, maybe exposed to the various toxins or any other exposures. So these are the findings in our lab. I'm very happy that we should know a lot about the PCOD, because the later generation, this is the reproductive age, they only become the diabetic patients or any other metabolic syndrome patients, because it will continue like that. So we have to improve by giving a lot of dietary fibers, omega-3, and then the micronutrients, increase the probiotic level in the intestine, and cut down the sugar, dietary sugar in our diet. These are the ways we can improve the PCOD and also decrease the epigenetic changes. So this is my take-home message. All of us has to really bother about our diet. So management of PCOS is diagnostic PCOS, mainly improved communication. The person's subject should explain properly and improve the compliances and improve the self-care and quality of life. Regular monitoring is required for the PCOD patients. So many references. We had many publications on PCOD. Thank you very much.
Thank you very much, madam. It was an interesting, inspiring, and informative talk. The clarity of presentation and the depth of the knowledge must be appreciated. To be frank,
thank you, madam. Thank you so much.
I got some input for giving the thesis for my postgraduates. I should accept this, madam, from your thesis. And only one question I want to ask, madam, regarding the supplementation of ghee for induction of labor on patients on letrozole. What was your inference, madam?
It's my face. Yes, madam. It is very, very interesting. Very interesting part we observed. Even we have some naturopathy doctors. They are treating PCOD. And here my colleagues, I'm here last 39 years. Some of the reproductive problem, infertility problem, they used to go to the Ayurvedic doctors. They will tell 1 week, 1 month, 3 months, 6 months. They ask them to take only ghee, and then afterwards they are carrying and giving birth to the baby. So when I started working on that, short chain fatty acids are very important. And I had one NIH project where in the NIH project, it is associated with Wake Forest University and John Hopkins University. So they are given me the project thinking that the vegetarian Brahmins only take ghee more in their diet. Their function is what is the different fatty acid level in that particular subject, in healthy subject with all the best diet. Especially we are taken the food frequency chart and we analyze, and they compared with their brain and eye function. So what we observed is they take a lot of ghee. When you see omega-3, omega-6 ratio, it is 1:4. Omega-6 is inflammatory marker. Omega-3 is pro-inflammatory. But still, they don't develop any inflammation. And then when we compare, they were telling the ghee contain lot of short chain fatty acids. Short chain fat directly absorb into the circulation, not necessary for the transporters. So they have found, and crossing blood-brain barrier also easy. So these are the things the vegetarian Brahmin category are very intelligent, very brain, and all that they compared and found it very good. So that made me to think, why can't we study? So we got a very good result on this also, madam.
Thank you, madam. Thank you for the clarification. Ma'am,
yes.
Sucheta ma'am, it's me, Manju ma'am.
Yes, yes, madam.
First
Yes.
of all,
Thank
let me
you so
thank you,
much.
ma'am. Yeah, no, let me thank you, ma'am, for such a overwhelming and such an insightful talk, madam. Though we all know too much of, we all speak and we all know the details of PCOD as insulin resistance, hyperandrogenism, etc. But today, when you were sharing your real, what is it, ma'am, the study or the project results with us, it really increases our knowledge, ma'am. The sharing your study results, like giving importance to the that it varies from SNPs. Your SNP studies showed us that from person to person, if it's a mutant allele, so person to person, genetic variation also influences. You have even proved by your studies by doing your Sanger sequencing and SNP genotyping. Then you also, yeah, and the heavy metal aluminum level thing that you've told, it is really a new thing, madam, for us. We have just heard that heavy metals can influence, but giving importance on your study results showing the importance of aluminum in causing PCOD is really eye opener for all of us, ma'am. And actually, like what Shashikala madam was telling, your study has actually gave us lots of insight about doing lot of other studies with our PG students and our PhDs, ma'am.
Yes,
and
madam. Yes.
a role of all these genetics, environmental factors, as well as the diet. You have not only just put it as a thing, you have proved with your study results in this talk. And thanks a lot, ma'am, for accepting our invitation, sharing your personal project details with all of us in this conference. Thanks a lot, madam.
Thank you, madam. Thank you so much for giving me opportunity. Thank you.
Okay, ma'am. Okay, ma'am. Now I would we would like to, though it is a virtual conference, we would like to honor our resource person first with an with a certificate and a memento online on a virtual mode, ma'am.
Okay, madam.
1
Thank
minute, ma'am. Have you stopped sharing, madam? Sujeta, ma'am?
Yes, that's stop sharing. No, 1 minute.
Please stop sharing, ma'am. That's
Stop.
बात नहीं करना.
Can you please?
Yes.
1 minute, not
1 minute.
They have to give access.
I think you have to give access.
Yes.
Yes. Thank you.
Next, I think I should really thank our chairperson, Dr. Shashikala madam. As soon as I told, she accepted and she prepared and she was very enthusiastic. Thank you, both of you, ma'am, for sharing your experiences here. Thank you, ma'am. Thank you.
Thank you, madam.
Thank you, ma'am.
Thank you.
Good morning, everyone. I welcome you all to the BMR presentation. And today's presentation is about the single cell RNA sequencing. So I feel extremely happy to introduce today's chairperson. And the details are early. Chat. Today's chairperson, Dr. Samandam Varikum. He has been working in the ABMC for the past 7 years. He has joined the ABMC in 2017 as a principal scientist. Now he is the associate professor and assistant director of research and head department of medical biotechnology, ABMC. His research interests are the molecular diagnosis, gene polymorphism, molecular markers, extremophiles, and synthetic biology. And I invite Dr. Ravi Kumar to introduce our today's speaker. Over to Dr. Ravi.
Thank you, Dr. Ganesh. I'm very happy to introduce Dr. Aribalan Perisamy, PhD. He is Associate Professor, Center for Creative and Convergence Education. He's from the Research Institute of Next Generation Material Design from Anyang University, Seoul, from South Korea. His previous experiences as a Research Associate Professor of Cancer Biology and Molecular Biology at Anyang University, and Research Associate Professor position at Yanggi University and Seoul National University, focusing on the cancer biology and microbiology and natural product chemistry. And he's also worked as a postdoctoral fellow in Seoul National University. Dr. Aribalan Perisamy has also received several grants and awards. In much noteworthy is that he has received a special research grants from the National Research Foundation Korea for a project related to Alzheimer's disease and nanotoxicity models. Dr. Aribalan has also published nearly 67 peer-reviewed articles with 30 as a first or corresponding author, with a Google citation score of around 1711 with an H-index 25. He has numerous articles in a reputed journal covering the topics such as cancer, research, nanotechnology, drug delivery, and the research spans a wide range of areas from the development of nanomaterial drugs for Alzheimer's disease to the study of mass cytometry and multi-omics analytical approaches for the nanotoxicity models. He has a strong publication record, has actively contributed to various interdisciplinary fields with the biotechnology and cancer research. I hereby request Dr. Aribalan Perisamy to give today's sessions on transcriptomic profiling of heterogeneous immune cells by high-dimension cells RNA sequencing analysis.
Good afternoon, everyone. My name is Dr. Harival Perumalsamy. I'm working as associate professor in Hanyang University, Republic of Korea. First of all, I would like to apologize not having the live online presentation. I'm sending my recorded video for this one. Due to my important meeting, I am unavailable for the live presentation, and I'm very sorry for that. And also, before beginning my presentation, I would like to congratulate for the organizing committee for such a wonderful e-conference on the biomedical or basic medical sciences field. So it's a great opportunity for me to present one of my research topic. And also I would like to thank organizing committee member to give me such a great honor to present my topics. And thank you everyone. So today I'm going to present about the single cell RNA sequencing analysis method, one of the advanced high dimensional method, which is very useful for the heterogeneous immune cell profiling. So when we are exposing some of the nanomaterial or drug, how these immune cells are responding individually at single cell level. So we need to have proper comprehensive profiling so that we could have to avoid misconception of the homogeneous type of cellular detection. So this is the main important topic I would like to present today. So I hope you may understand my presentation. However, if you have any doubt or comments, so please leave me in you can drop your comment or questions in the chat box. So I will try to reply as much as I can. So yeah, so let me explain more detailedly one by one about my presentation. Yes. Yeah, so today I'm going to discuss about the different type of nanomaterials. So particularly, I'm just selecting the silver nanoparticle. So because we are keep on working on the nanomaterial exposure to the immunological immune cells, and also we are profiling the how immune cell responses in the pathogenic sample, like a deceased patient sample as well as healthy donors, and also we are profiling the COVID-19 samples when we are having the booster shot and when we are having the we don't like COVID patient sample as well as the booster shot the sample. We can do compare the immune profiling because it's very important for the vaccine development. It's very important for to understand the how immune response or defense systems are working. So we need to have these kind of analytical method so that it will be very useful to make such a decision. So that is the main objective for our thing. So nanomaterial exposure is one of the objective for our laboratory. So in here, I'm going to explain about the silver nanoparticle. Since silver nanoparticle, there are so many biomedical application in case of viral, antiviral activity or anti-cancer, antibacterial. There are so many application so far in the biomedical field. So, however, in case of frequent exposure of the silver nanomaterial, and we need to have proper in vitro toxicity assessment. So, for example, so far we do have short-term in vitro toxicity assessment in homogeneous immune cells, for example, epithelial cell or cancer cell or different type of immune cell or like raw cell or many things. And in case of the in vivo, we do have exposure to the silver nanoparticle infrequently, and we don't have proper ex vivo toxicity evaluation in case of the PBMC because PBMC have the different type of leukocyte like NK cell, B cell, monocyte. There are so many type of different type of NK cells or like different type of cells. They can respond differently so that we can call it as a heterogeneous cellular environment. So for these heterogeneous cell types, we don't have proper assessment at a single cell level profiling. So we need to have proper high dimensional profiling approach so that we could avoid misconception of homogeneous in vitro as well as in vivo analytical method or toxicological approaches. So that is the main objective. We do have single cell RNA sequencing analysis. So that is the main objective. So before going into deeper, let me explain about how immune cells are responding when you are exposing some kind of drug or nanomaterial to the immune cells. I think biomedical students, they do have understanding about the immune cell response. So in case of low immunogenicity, for example, if we are exposing some nanomaterial or drugs, so they do recognize this will be the antigen or harmful to the cell, then they may respond or activate T cell mediated adaptive immune response or innate immune response by activating the MSC1, major histocompatibility complex 1, or MSC2. They will be stopped in case of they think there will be no harmful to that particular cell, that will be the low immunogenicity. So in case of they recognize that nanoparticle as a antigen, so they do have respond quickly. So they activated the antigen presenting cell which can recognize the antigen, and then they do activate the T cell which can into adapt immune response, or they can produce some antibody mediated humoral immune response by B cell activation. That will be the nanoparticle antigen specificity. And third one will be the antigenic modulation. So some nanomaterial, they can modulate the antibody mediated cell surface. They do have some kind of, they can lose the variant. So there will be lost the specificity when we are exposing some kind of nanomaterial. And some nanomaterial induced or suppress the immune system by showing the toxicity to the cell. So they may be upregulated or downregulated, or they may be suppressed completely. And then another one is the privileged site. They will be target specific. So they can be as a physical barrier to protect that immune cell when we are exposing the cellular nanomaterial. So these kind of heterogeneous responses will be profiled or will be studied at a single cell level at the same experimental condition. So we don't have such a proper analytical approaches to analyze these heterogeneous differences at the human PBMC. So there will be so for homogeneous single cell type, like I said, is insufficient and may cause the misconception of the nanoparticle response. So we need to have comprehensive toxicity assessment so that it can be contribute to the understanding precisely in case of toxicity or in case of immunological response, either will be hyper or hypoinflammatory response. So, and similarly, after we identified the cellular response, we do have some kind of molecular targeting way. Those are the commonly available, like ROS mediated oxidative stress or inflammatory response, or apoptosis or autophagy mediated response. So when we exposing the cells, for example, B cell or T cell or monocyte or hydrogranulocyte, so they may respond individually. They may enter different type of pathway by exposing inflammatory or anti-inflammatory responses. So that will be profiled individually at the same experimental condition at one by one at single cell level. So that is also our second objective, to expose everything. So before going further, let me explain the differences between most commonly available, we can call it as a bulk RNA sequencing analysis, as a single cell RNA sequencing analysis. There will be the main important to understand what is the difference between these and where we can apply for single cell RNA sequencing analysis. Since the bulk RNA sequencing is the most commonly used RNA sequencing analysis method, it will be like there will be RNA isolation in all together as a one bulk RNA input. Then we can go for the sequencing. There will be the homogeneous expression, and it failed to expose the cellular heterogeneity. It will be masked together, and there will be no differences what type of cell and how they responded when we are exposing some kind of drug or nanomaterial or some kind of disease related exposure when compared to the healthy individual. So these are the commonly available method which is not applicable for the heterogeneous immunocellular environment. So we need to have, or we need to profile cell one by one individually, so that we can also have different type of unique specific expression, and also we can reveal based on the cell type, we can reveal the heterogeneity, and also reveal the subcellular population, so that we could precisely expose all the type of phenotype as well as DAG, differently expressed gene expression. So in case of healthy or pathological tissue sample, when we have the tissue samples, we do have profiling using single cell RNA sequencing. It can expose the differences between the cell types based on the morphological changes or expression. And we can also identify what type of cells can be disease associated, what kind of cells can be profiled differently between the normal unaffected cell. Then it's easy to prepare the drug, or we easy to target it that those particular cells for the further treatment process. So these things will be explored or unrevealed. These things, it will be very, very important when we go for the single cell analysis. And the main objective for this one, I already explained, the immunotoxicity response. In here, we just expose silver nanomaterial and also compare with the untreated control. So we have cell type identification, phenotype identification, and we also expose the heterogeneity responses individually. And also we also express the differentially expressed genes from all individual responses. And finally, we have interpreted the gene network and the molecular level pathway. So this is our main objective. Like I said before, both identification as well as molecular target for each individual. This will be help us to overcome the limitation whatever we have approached so far, like in vitro or in vivo analysis in the homogeneous as well as another expose. So we do have overcome all these limitations by using or performing single cell RNA sequencing analysis. And in our study, the experiment design will be the human PBMC will be isolated from the human blood from the volunteers. And then we have treated with the silver nanomaterial for the 3 hours into microgram per ml. After that, we have barcoding the cell individually, and then we do perform the 10x chromium analysis. It will be performing by the 3 different steps, like a bead attachment and cell individually separate, and it coated by the oil layer, so that that one oil droplet will having the barcoding and one cell, and we can have some kind of primer which can be attached with this one. So after we have isolated individually with using the 10s chromium analysis, and then we can go for the further amplification and library construction and PCO analysis, and then we go for the data analysis, and finally we get the result, and we can do for the data analysis in the another part, huge part. So this is the main thing already I explained for this objective. So when we getting the data, like a pre-processed data, so we do have the like a different type of processing method to have the whole sequencing read, and we remove the unread, and we remove the unwanted thing. And then we do have many different type of like files to have the UMA code or cell barcode or transcriptomic, and then we can go for the pre-processing one by one to get all the expressed gene. And we performed many different type of program like R to visualize high dimensional 2D plot to expose the phenotyping using the marker gene expression. And further, we do the differently expressed gene, and also we do the pathway analysis to expose all the isolated things. That is our main objective. One part will be like experiment, another part will be like a data analysis. There will be 2 huge different parts. which can work on this one. So based on the cellular expression, initially we do have some kind of all the exposed cell which can be identified in two different parts like control and treated groups, so that we can confirm all the cells are alive or exposed well, even though when we are exposing the treated silver nanoparticle treated group. And then we do have the cluster identification for the unique expression. So in here, we got more than 11 clusters with unique identification clusters, and each clusters will be expressed based on the marker gene references. For example, in T cells, we can have the three different type of marker gene which can express uniquely in here, which will be considered as a T cell. And for monocyte, that green color, like number 5 cluster, they can express uniquely for these genes. Similarly, for NK cell and B cell, platelet, and neutrophil, all these cells are expressed uniquely for individual. Then we can phenotyping individually one by one. And similarly, we have finalized the all the immune cell type like control and treated group. And we do have naming like B cell, NK cell, T cell, and all the thing like control and treated group. And when we compare the immune cell population individually for the both the individuals like T cell, monocyte, both control and treated group, there will be no further toxicity in case of the cellular nanoparticle. So this will be a huge interesting. So when we are exposing the nanoparticle, there will be no further toxicity. Similarly, for the expression individually for all different cell type by showing the different type of cellular marker expression. So in here, there will be not much huge differences. However, we do have the further deprofiling for the individually like B cell, T cell, or NK cell. Then only we can expose for the subset identification, so that we plan to profiling one by one. So initially, we profile the monocyte because monocyte will be the primary target for the any type of nanomaterial or antigen. They will be captured and then they get differentiated into either phagocyte, or they will be differentiated into dendritic cell, antigen presenting cell. Then they will be activated for the B cell or T cell mediated adaptive immune response. So that is the main important thing. So we profile monocyte to explore the monocyte subset. We isolated two different type of subset like CD16 positive, CD16 minus. We can call it as a classical or non-classical monocyte. So these things will be like when we do the initially, there will be no differences in the monocyte also. But when we go for the further deprofiling individually, so we do have some kind of slight changes in the population, and also slight huge differences in the up and down regulated genes. So interestingly, so monocyte, particularly the metallothionein related gene, which is called it as a cellular metal ion response gene, is upregulated completely. It's indicated that monocytes are primary target. They can bind very well with silver nanoparticle. So, however, they can closely upregulated with the metal ion related thing. But however, they don't have further inflammatory response. All the inflammatory response genes like interleukin beta or TNF alpha and chemokine genes are down regulated. This is a very interesting finding and novel finding for us because nanomaterials we always consider or thinking that there will be always will be they produce the toxicity to the cell. But this indicated that even though they are closely associated with the monocyte, there will be no further inflammatory mediated cytotoxicity. So that's why there will be no differences in the population differences. So this is an interesting finding. So there will be no further inflammatory response was activated because, like I said before, monocyte can be activated either macrophage mediated engulfing or B or T cell mediated adaptive immune response because monocyte can differentiate into the dendritic cell or antigen presenting cells. That is the important function of the monocyte. So there will be no further inflammatory response to initiate either innate or adaptive immune response. And similarly, this is the gene ontology of molecular pathway detection for the monocyte. The down-regulated genes, like I said, there will be no further inflammatory response or confirmed again by gene ontology as well as gene enrichment analysis. The blue color all down-regulated. All the thing can also show the enrichment score. It will be the minus score. It is like an immune response or defense response, all are down regulated. This right side image, it's indicated that all the cellular metal response association was highly upregulated. Even though it was closely associated or attached into the monocyte, there will be no further immune defense response was initiated. So that is what concluded in the monocyte. In summary, whatever I said, it is a confirmed that beta-glucan associated genes were strongly associated when compared to the control gene. This is the result we have got in the monocyte. It will be helpful to maintain the hemophytosis even though they initiate the pro-inflammatory response. When they are initiate like acute inflammatory response, there will be no further continuous inflammatory responses. In case of B cell subset, similarly, we do have isolated two different type of B cell subset. One will be like naive B cell, another will be memory B cell. Either both B cells will be significantly activated either with the down-regulated or up-regulated. However, the down-regulated gene, the most important gene from SOD or MSN or thing will be like down-regulating the oxidative immune response. It will be avoid the ROS-mediated inflammatory cytotoxicity. In case of MS1 or FLT, this will be like helpful to impair the T cell or B cell mediated proliferation or immune responses. Similarly, for other one, it also like helpful to downregulate all lysosomal mediator, which can suppress the B cell activation. So these all downregulated genes are clearly notice that there will be no cytotoxicity was produced when we are exposing the cellular cell nanoparticle in case of the both naive and memory B cells. And this is the gene ontology result. This indicated that both naive and memory B cell, the naive B cell may be initiated initially to be activated the RNA related mechanism like cytoplasmic mediated translation process or protein process. However, the defense process will be down regulated in either memory or memory B cell. This is the memory B cell source, all the inflammatory response will be down regulated. So this indicated that both naive and memory could help researchers better understand how the immune systems react when the metal ions are attached. So that is that inhibition of oxidative stress indicated there will be no further toxicity, and inhibiting the memory B cell, this indicated that there is will be no immediate early response when we are having the external stimuli. So like all the silver nanoparticle when we are exposing particularly to the naive or memory B cell, the inflammatory response will be suppressed or inhibited. This is what we have found in our thing. And finally, the T cell. So when we having the T cells, when we go further deep profiling, the time the T cell will be further divided into either T helper cell, which includes CD4, naive, memory, and effector, and T killer cell or cytotoxic killer cell will be like CD8 mediated because this will be activated by MHC2 complex and this will be activated by MHC1 complex. So there will be no differences in case of the population differences. However, the significant genes from the either naive memory or naive T cell will be upregulated or downregulated significantly. And like I said, all the genes from the T cells, like all this one and this one, will be downregulated because this will be the very important gene for like T cell differentiation was downregulated. Enough kappa B genes, which is very important for initiate the T cell mediated adaptive immune response, also downregulated. And particularly the cytotoxic killer T cell, which is very important for toxic producing the T cell, it will be no significant. So none of them are showed any significance. This also indicated there will be no cytotoxic response to the T cell. And also finally, all the different expressed from the both naive and T cell, they prevent further initiation of the adaptive immune response. This is what we have noticed in case of defense or response to cytokine or response to cytokine similar, all will be down regulated. So that's what we have noticed. And finally, the conclusion, when we are exposing all kind of nanomaterial or all kind of drug, the initial target will be the blood. So when they are enter into the blood, so they will be encaptured by the different type of heterogeneous immune cell. They will be activated either by the NK cell or monocyte or T cell or granulocyte or anything. In our study, these three cells are significantly responded into the silver nanoparticle. In case of NK cell or granulocyte, there will be no further significant response. So we have selected those three for the further deep profiling. So in case of monocyte, so like I said, the metallothionein genes was upregulated, which indicated that monocytes are closely associated with the metal cellular response or closely associated with the cellular nanoparticle. It can be activated metallothionein related protein. So it indicated that there will be this slight oxidative degradation. However, there is no further inflammatory response. This is the pro-inflammatory or cytokine response or chemokine response. This indicated that so there will be no hyper mediated or inflammatory mediated toxicity response when you are exposed to the cellular. This will be observed both classical or non-classical monocyte subset. And similarly, in case of B cell, we divide into naive B cell and memory B cell subset. In case of naive B cell, like we already said, there will be inhibition of the oxidative stress of the ROS generation and also impairment of the T and B cell lymphocyte. It will be helpful for the adaptive mediator or humoral mediated immune response will be stopped due to the impairment. Similarly, for the memory B cell, inhibition of growth factor or cytokine release will be stopped. There will be no further memory-related inflammatory response in case of silver nanoparticle exposure. So finally, the T cells also divided into two different things like cytotoxic killer cell or T helper cell. So in case of T helper cells, that all the T cell differentiation, all the T cell activation cells are downregulated when we are exposing the silver nanoparticle. It denotes that there will be no further adaptive immune response was noticed. In case of CD8 T cell, there will be no further significant differences were observed between control and treated group. It's indicated there will be no further cytotoxic killer T cell activation when we are exposing the T cell. So finally, silver nanoparticle can closely associated to the monocyte. Even though they have associated with the monocyte, there will be no further monocyte differentiation into to activate T cell mediated or B cell mediated humoral responses. So that's why monocytes are, even though they have closely associated, they have detected as a antigen, but there will be no further inflammatory response which can activate into B cell or T cell. So that indicates that silver nanoparticle are not that much toxic or not produce any toxicological response or hyperinflammatory response when we are exposing to the human blood PBMC cells. So that is the overall story for today's presentation. And also in our Center for Next Generation Cytometry, we do have two different type of analytical approach. That will be the mass cytometry analysis. This is the CyTOF, and we are analyzing or deprofiling the immune cell based on the surface protein markers for the mass cytometry analysis. In case of single cell RNA sequencing, we do have analyzed cellular phenotyping based on the barcoding as well as gene expression. We do have identified all the cell at the single cell level and identify the molecular pathway by using the single cell. And the only different thing is this will be the biased, or it will be the selective protein markers to identify target immune cells, and it will be unbiased one. So we can identify all the cell type with the huge data analysis. That is the two different things we are doing. And also, I would like to thank Professor Yoon, also one of the colleague and co-worker of my as the postdoc and my students. Those were supported and helped me for this thing. So, and also this is the project that we have received on the lab. So once again, thank you so much for the organizing committee for giving me opportunity to present our work. And also, like I said, and please drop your comment, and I hope you can understand about today's presentation. If you have any doubt or further clarification, please
So I thank Dr. Haribalan Perumalsamy for his wonderful presentation on transcript profiling of immune cells by single cell RNA analysis. As he has an important meeting now, he could not present on live. But anyway, we have shared his mail ID. If you have any queries, you can mail him. He assured that he would definitely reply to all your mails. So as a token of appreciation, we would like to share the e-certificate and the memento to our speaker, Dr. Haribalan Perumalsamy. Also, we would like to share the certificate of appreciation, a memento to our chairperson, Dr. S. Ravi.
Thank you, Dr. Ganesh. I want to say a few things about this study, that I want to express my sincere appreciation for outstanding presentation during this conference. Your talk on silver, the Haribalan talk on silver nanoparticles and the impact on transcriptome profiling of heterogeneous immune cells through high-dimensional single-cell RNA sequencing analysis was truly especially. So here are the few specific aspects that I found particularly commendable. So I have two more questions for him that I will mail to him. One is that what is a specific immune cell types shows a most significant alteration in the transcriptome following the exposure to silver nanoparticles. And second question is that can this single cell RNA sequencing analysis distinguish between a direct effect of silver nanoparticles on immune cells and the potential secondary effect mediated through intracellular communications on signaling pathways? So these are the two questions I just want, because we also work on this pathway analysis. So these questions, if he can able to answer us, it will be very nice. I found the particularly commendable about his work is that his presentation was very especially clear and well structured. His ability to engage the audience was impressive. Since the talk was related to the next level, next generation sequencing, and all the research labs are moving towards that area, I think this study will help the young generation how the future of the mass spec analysis as well as the single cell sequencing of transcriptome profiling is very important to the understand the what do you say, the challenging molecular mechanisms. Congratulations, Dr. Haribalan. Thank you for sharing the groundbreaking work with us. Thank you.
Thank you, Dr. Ravi. I hope Dr. Haribalan will answer all his queries. I thank each and everyone for joining. Thank you. Very good morning. We'll continue with our next scientific talk presented by Dr. N. Mukundan sir over translational research in anatomy. I'm happy to invite our chairperson for today, Dr. Sulokshana Saktivel, Additional Professor, Department of Anatomy, JIPMER, Puducherry. Madam has done her undergraduate from Tirunelveli Medical College in 1990 and post-graduation from Tanjavur Medical College in 2006. She has added numerous publications in both national as well as international level, and Madam has got an experience of more than 17 years on the field of anatomy. We welcome you, ma'am. I also welcome our next chairperson, Dr. Shantini Arul Selvi, Professor and Head, Department of Anatomy, Vinayaga Missions Medical College from Karaikal. Madam has done her undergraduation and post-graduation from Annamalai University, and she has got more than 15 years of teaching experience for both undergraduate as well as post-graduate session. We welcome you, ma'am. I also welcome our resource person for today, Dr. N. Mukundan sir from Sri Mookambika Institute of Medical Science. May I request our chairperson for today, Dr. Sulokshana Saktivel, to introduce the speaker today. Over to you, ma'am. There is a slight interruption with the connectivity. I'll introduce the speaker. We have with us Dr. N. Mukundan, Professor of Anatomy, currently working at Sri Mookambika Institute of Medical Science, Kulasekharam, Tamil Nadu. Sir has got a wide experience. Sir has been a former Professor of Anatomy at Sri Balaji Vidyapith University, Puducherry. Sir has completed his undergraduation in Government Tirunelveli Medical College in 2009 and from his post-graduation from Madras Medical College. And he took his DNB in 2012. Sir has completed his PhD in anatomy in 2016 from Sri Balaji Vidyapith University. I think Madam has joined. Ma'am, we are happy if you continue.
Okay. Dr. Mukundan made an impact with his excellence as a teacher, research worker, and research guide for MBBS, MD, and PhD students in anatomy. He has published a series of research papers on the effect of mobile phone radiation on animal issues in various national and international journals. He is a member of National Academy of Medical Sciences, New Delhi, and a member of various national and anatomical societies. He is also a critical reviewer in many national and international anatomy journals. He is an American Heart Association certified instructor for BLS and ACLS courses since 2013. We welcome you, sir, and we are honored to have you here, and we are eagerly waiting for your speech. Dr. Ilankathir?
Sir will join within 5 minutes.
Okay, sir. We'll wait for sir to join the session.
NICA Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future.
Sir is joined.
Yes, sir. Extremely sorry for the delay in the start. It's because of the connectivity issue. We welcome Dr. N. Mukundan, sir, from Sri Mukambika Institute of Medical Science, Kolasegaram, Tamil Nadu. Sir.
Yes, sir. Good morning, sir.
Good morning, sir.
Good
And
morning.
may I request Dr. N. Mukundan, sir, to present his talk on translational research in anatomy. Over to you, sir.
Oh, thank you, sir. Thank you, sir. Very good morning to everyone. At the outset, I would like to thank the organizer of the 4th International Conference, the BMSECON, especially the Professor and HOD of Anatomy, Dr. Ilankathir, sir. And sir, very thank you very much, sir, for sharing that regarding the knowledge of translational research in anatomy. Just a few things I would like to share regarding the translational research in anatomy. Am I audible to everyone?
Yes, sir, you are loud and clear.
Can everyone see my screen?
Sir, if can share your slides.
Is it okay? That everyone can see in the slides?
No, sir. No, sir.
No. No. Okay. One second. One second.
Your screen is visible, sir. You can go for the slideshow, sir.
Okay. Visible, sir?
Yes, sir. Clear, sir.
Yeah. Can I proceed, sir?
Yes, sir.
Yes, sir. Very good. Okay. So that is these are the learning objectives in the translational research. First, I would like to explain what is translational research and what is translational research in anatomy. And I will give a few examples about that, the translational research in anatomy. Okay. So what is translational research? The definition is very simple. The process of turning observation in the laboratory, the clinic, and the community into intervention that improve the health of the individuals and the public. It can be a diagnostic and therapeutics to the or the medical procedure and the behavioral changes. So simply that is, we are turning the observations, what we are seeing in the either in the dissection hall or in the clinic or even in the community into interventions. That will be helpful to that public. That is translational research. The goal of the translational research is very simple. That is to translate or move the basic science discoveries quickly into the practice, quickly and efficiently into the practice. That is translational research. Just this, I'll just explain this one with a small example. So in our college during that, the family adoption program, FAP program, the students are divided into, that is the 5 villages. They visited to the villages about, that is each family, and taking the data and everything, when their health status and everything, in one particular village, they found that the people, they all are having, there is a same complaint, that is the running nose and the sneezing and the nasal congestion due to, that is a sinusitis, that problem. So particularly that is nearly around, that is the nearly 30 to 40 families, they have, everybody having that problem. They reported to that is the faculty, community medicine faculties. These families are there having such a problem like that. Fortunately, there one student, he already completed the Siddha BSMS. And when he visited that near the house, he found a plant. There are a lot of plant that is called the Parthenium plant, and he understood that this plant is the culprit creating, that is the allergic response. And he informed to the concerned faculty, sir, these all the houses, the houses are surrounded by the Parthenium plant, and this plant is the one maybe the cause for their the nasal congestion and the allergic rhinitis and the sinusitis. Then the faculty, okay, understood, maybe a reason. Okay, the next day when they are coming to the visit, they requested the family that all the villages, can you remove all this plant and this Parthenium? It may be it is not good for your health. That's why you are having this allergic problem and such a thing. They agreed happily. Okay, sir, we will remove it. So within 2-3 days, they have removed everything. And the next week, they have visited. Nearly, that is the 50% of the people, they were free from the symptom. They followed up again after 2 weeks. They were completely free from the symptoms. So it indicates that these students' family, we simply say that is some family adoption program, and they are going and in the first year. So they found that is they observed that in the community, they found the problem. They identified the problem. What is the problem? What is the cause for the problem? So they identified the plant and they removed the cause. Now they are free. Now it is. So this is simply like a translational research. So that is the observation, what you are seeing in the community, they are putting into that, turning into that intervention. So that is the translational research. There are 5 steps in the translational research. I will just give a brief outline of it. So usually in the anatomy, the preclinical and the paraclinical department, that the research will take place, that is at the level of the T0, that is translational research at the 0 level. So this we are calling it is the basic science research. It is done in the preclinical and the paraclinical department. Especially this is an animal study. Not only it is not restricted to the animal studies, but for example, we are formulating a device to diagnose a particular disease or particular things in the blood. Like simply like a glucometer, like that, a new device or such a thing there. So this is starting in the pre and the paraclinical department. Especially if we are formulating a molecule, drug molecule to prevent a particular disease and such a things, it will start with an animal study. So here in the preclinical and paraclinical, we are defining the mechanisms and the targets, or it may be a molecule. We are leading to molecules. Okay, so that is the T0 level. Now that is the T1 level. Now we have designed a study, that mechanism we have designed, or a drug molecule or a device, whatever it may be. We have designed and we tested in the animals. Now we are translating into the human. This stage that is in the phase 1 clinical trial. Here the T1 stage. That's what we applied in the animal studies, we are applying into the human. It will be the human volunteers. Okay, so that is, we are giving the proof of concept that is in the phase 1 clinical trial. So now we are adapting a new methods for a new molecule or a diagnostic device or such. So this is the T1 stage. Next is that is the T2 stage here, that is actually originally that is the phase 2 and the phase 3 clinical trial will be happen in the patients. We are administering the molecule or we are administering the device from the human volunteer to now it is the patients. So the translation to patient. So we are translating from the animal studies to human volunteers and to that is the patients. So this is where the controlled studies. We have a control and we are applying that. So now from this phase 2 and phase 3 to we are moving to the phase 4 clinical trial, that is we are calling it is the translation to the that is into the practice. So here that is the molecule is in use, or say that is the diagnostic device what you developed is in use. So here that is now we are testing that is its outcome of this drug. What is the main outcome of this procedure or the drug or the device? So here the delivery of recommended and timely care to the right patient where we have to give. So that is the T3 stage. Now T4 stage is now that is the translation to the community. So already the drug molecule used by the patients already is in the use for say that is 3 or 4 years or 5 years. Now what is the effect of that the research or that is a particular device or particular drug molecule we developed? What is the outcome in the community? So this is that whether that drug or the device is actually truly benefit to the society or not. What is already existing molecule, we will compare that, whether it is any new benefit to the community. So that is in the translation stage 4. But when we will consider in the preclinical and the paraclinical, we are mainly that is focusing on that is the animal studies. So we are the one that is putting a foundation. The pre and the paraclinical department are putting a foundation for the translational research. We alone cannot complete that because that our observation should reach to the community to get the benefit of our research. So we are the initial part. We are the foundations to provide that is the basic for the translational research through the animal studies. Okay. Now come to the translational research in anatomy. What is translational research in anatomy? So the process of turning observation in the dissection or the procedures in the cadaveric lab, not new endoscopic procedures, so many new procedures we are doing in the cadaveric lab and also in the not only cadaveric lab, the histology and also in the genetic lab. into the interventions. That intervention should improve the health of the individuals and the public. So that is translational research in anatomy. Okay. So simply that is whatever we are observing, either in the dissection lab or in the cadaveric lab in the histology or genetic, or whatever in division, either in osteology class, whatever that we are observing in the lab, that is we are turning into the interventions. So that is that intervention will helpful in the health of the individuals and the public. That is sometimes you may think that what we can do in anatomy, like simply observing the histology and the cadaver, and how we are going to help into the community, especially when that is the STS project in ICMR comes, the students used to come into the anatomy, because they are very close with the anatomy, physiology, and biochemistry, the first year student. After completing the first year, they used to come to the preclinical department. Then most of the I saw that most of the faculties say, STS project in anatomy, what you can do? We have only cadaver and the histology lab. We can't do anything like that. No, don't say like that. So we can do, that is a many thing in the translational research. See, that is the what we are observing. For example, the students are doing a dissection. For example, any variation in the nerve in the upper limb or in the abdominal organs, anything, we are putting a publication. We are finding out, present it, and publish it, bring it to the knowledge to the surgeons and the clinicians. Through the clinicians, it will reach to the community. Who will get the benefit? The public. The community is going to get the benefit through the clinicians. Who is the foundation? We are the one foundation. We are the one founding the variation. So I will give the brief examples about that, what are the translation research in anatomy, and we will go for a few examples in that also. Okay, so again, so the translational research in anatomy is scientific knowledge gathered via anatomical research being used to create workable solution for that is the human health and the medical procedures. Okay, the integration of activities from the bench to the bedside. Bench is that is in the lab, okay, cadaveric or in the histology lab, to the bedside, to the clinics, to the patient. Okay, so bridging the gap between the basic anatomical research and its real world application is the translational research in anatomy. Okay. So these are some of examples of the translational research in anatomy. In the clinical anatomy, researchers may use that anatomical knowledge and the surgical technique use and the medical imaging and diagnostic procedure, we can use it. For example, that is the 3D printing technology has been approved to create a patient-specific anatomical models for surgical planning. Okay, so for example, that is this 3D imaging of that particular individual who is admitted for a particular disease. Before planning for the surgery, you can work out on that the 3D printing model. That is the same exact ditto of the patient. So we can work out more in the, that is the before surgery. We can it. So that will be that more helpful in the clinical anatomy. Then in the disease research, anatomical studies can contribute to understanding the underlying causes and the mechanism of the disease. This knowledge can lead to the development of the targeted drug therapies and interventions. For example, this is the deep brain, DBS, the deep brain stimulation, implanting the electrode into the brain to treat the condition like Parkinson's disease and the essential tremor. So to keep that is the DBS, that is we must know that is where is the anatomical site, where to stimulate these neurons, especially that is the corpus callosum and agent. So we need that is the anatomical knowledge. Then that is the LVADs, that is left ventricular assist devices, or the surgical devices implanted to assist the failing heart by taking over the pacemaker function. So for that is we need that is the basic anatomy. Now the drug delivery system. Researchers use anatomical data to design the drug delivery system that target the specific anatomical region. This can enhance the effectiveness and reduce the side effect of medication. This we all know that is especially in the radiotherapy or in the brachytherapy, and especially that is the uterine artery embolization, and in the case of fibroid. So many advances are there. So for the drug delivery system, we need that is the typical anatomical course of these vessels, the variation in the blood vessels, such a things will be helping. Then anatomical education. translational research can improve the methods and the tools used for teaching anatomy to the medical students and also the healthcare professionals, ensure that they are up to date and in the relevant anatomical knowledge. That is nowadays that is the lot of softwares, the virtual dissection, anatomage, isn't it? So that is the and then now that is the artificial intelligence. So these all are that is the helping that to educate that anatomic knowledge. Okay. The rehabilitation and in the prosthetics. The translational research in anatomy plays an important crucial role in designing prosthetic limb and the rehabilitation programs. The detailed knowledge of the morphometry of the hip and knee joints help in creating prosthetics with mimicking the natural and that is the movement and the function. For example, most of the that is these devices, say that is the knee implant and the hip implant that is manufactured in either in Germany or in the USA. Their morphology of that is the tibial condyle and the femoral condyle, or say that is the head of the femur and the acetabulum will be different when compared to the Indian Asian population. So we have to work out on that our population to find out the morphometry of the condyle. And if you develop a device prosthesis for the Indian people, that will be the more fittable one. So that is the rehabilitation and prosthetics. Now, come to that one important two examples I will give. One is for that is the research in the research aspect in the histology well-being. Another one is that is the gross anatomy. So I will give that is these two examples that is for in the translation research. When you will start, that is a T0 stage, that translation research in the first step is we have to identify the problem. Where is the problem? Where is the lack? Where is the gap? So in my research, we have identified that there was some contradictory scientific reports on the effect of radio frequency radiation emitted from the cell bone on the biological tissues. We found that problem is there. We identified that problem. The World Health Organization and the International Agency for Research on Cancer, they classified that is the cell phone radiation, the RFR emitted from the cell phone is category 2B. It means it is possibly carcinogenic to the humans. So that is category 2B. So ICMR has asked to conduct a multidisciplinary study in the Delhi to find out the adverse effect of radio frequency radiation emitted from the cell phone on the general population, that is especially on the human volunteers. Okay. The ICMR also has cited that there are no conclusive data or evidence till date so far to show that radiation from the mobile phone or the towers have adverse health effects. So ICMR initiated the study with a statement that emphasizing the need for further investigation. So we took that study and we found out that is they have reviewed that literature and everything, and we found out that is the effect of mobile phone radiation induced single and double stranded DNA breakages. There is a lot of reduction of that is the Purkinje cell number in the rat cerebellum. There are significant reduction in the sperm count, reduction in the serum testosterone level, and also that is the short-term memory affected in the mice. There are short-term exposure induced damages in the kidney. And in the human volunteers, the study says that there is risk of acoustic neuroma. That is a long-term exposure in the human volunteers, 10 years, because keeping the cell phone in the air and hearing, so it will induce that is the acoustic neuroma. There is a reduced, that is the reaction speed to a particular stimulus, both increase in the systolic and the diastolic blood pressure, and the genotoxic effect in human peripheral blood leukocytes, decrease in sperm count, motility, and viability. There was a question once that they asked, that is, sir, where to keep my cell phone safely? Nowhere. That is, we always have a mail, we always have on the left side, that pocket, chat pocket is on the left side, not on the right side, because we always use that is the right hand. So whatever thing we put in the pocket, it will be easy to take out if it is on the left side, but the same thing left side. So in that you will have the heart. So those who are using that is the cell phone, they are having that is the conduction abnormality in the conduction system of the heart, especially that is the brady and the tachyarrhythmia. And when you keep the cell phone near your, that is the inguinal region or the spermatic cord, there also that is the sperm count is reduced. When you keep it in the pocket, pan pocket in front side, Again, that is the sperm count is reduced. Then where to keep? The ideal place is put it over on the back pocket, back one. So that is the gluteal muscle only will be exposed. This problem will not arise for the female because their ovaries are very safe in the abdomen. Okay, so that is all the male has to take care. Okay, so that is. We found there are some contradictory finding also. That is no breakage of the double-stranded DNA of the rat brain, no effect on the mouse embryonic lens development, no effect on the psychomotor performance, and no histological changes in the rat test. Okay, so we made that is the aim and objectives to study the postnatal effect of radio frequency radiation emitted from the both at that time. So our study was conducted from 2010 to 2016. That time, that is the 2G and 3G phone was in use. So we have studied that is the 2G and 3G phone in the mice from 0 to 180 days. And we want to identify the gross anomalies occurred due to the radio frequency exposure in the both 2G and the 3G cell phone on brain, liver, kidney, testis, and ovary of the mice. And we want to establish a severity of the structural damage caused by the radio frequency radiation on the solid organs like especially brain, liver, kidney, testis, and the ovary. Okay, and we want to analyze the structural changes in the quantitative terms using that is the histology and the histomorphometric data to prove the potential damage exposed in comparison to the non-exposure. And we want to evaluate the oxidative stress caused by the radio frequency exposure by measuring the level of the antioxidant, that is the superoxide dismutase and glutathione peroxidase. And we want to assess the changes in the protein expression by single dimensional SDS page. We have done also this study in that is developing chick embryo also. We have done that is the COMET assay, and the study in the MGMCRI that they have conducted the study in also that is the 3G as well as that is the 4G, comparing 3G and 4G. Now that PhD scholars, researchers are now that is because the emergence of that is the 5G. Now they are conducting their study on the that is the 5G cell phone radiation. Okay, that is just I am presenting that is the my study only. Okay, so this is our experimental setup in the cage. That is the mice is there, and we have kept a cell phone that is both 2G as well as 3G cell phone we kept. And what you are seeing in here is this is the RF meter is there to find out that is how much radio frequency radiation has been exposed and how much has been absorbed by the animal. The various levels are there. That is from starting from the green to that is the yellow and the red. When that is the maximum frequency will be the red. The cell phone will be kept here, and we adapted with that is the auto answer mode. That is whenever we are calling, that is we made that is the 48 minutes per day. Actually, every 30 minutes, 2 minutes call we will give. And once when we are giving the ring, this auto answer mode will be enabled either both in the 2G voice call, also in the 3G, that video call will be automatically on, and that radiation will be exposed. And that radio frequency will be measured by the RF meter kept inside here. Remember that animal will be, the mice will be moving here and there in the entire cage. And wherever that mice will go, that is the RF meter will detect that radio frequency radiation, and we calculate the dosimetry. Okay, so this is our setup, and we use that is the standard mobile phone. We don't want to disclose that what type of mobile phone, what company we have used. Similarly, which network provider has been, that is we use that network. That one also is not disclosed. And the frequency range is from that is for the 2G is 900 to 1800 megahertz. And for the 3G is 1900 to 2200 megahertz. That is for the video call. And the power of the cell phone is 2 watts. And the specific SAR, the specific absorption rate of the cell phone is 1. The standard, that 1.6 recommended by WHO, 1.69 watts per kg. That is per 10 gram of tissue. Okay. The duration of the exposure is 48 minutes per day from 8 a.m. to 8 p.m. That is every 2 minutes, every 30, every half an hour, we expose for 2 minutes. Okay. So this is that is the study parameters we have studied in the brain. That is, we focus only on the, of course, we have taken the sample for cerebrum, cerebellum everywhere, but I have concentrated only on the hippocampal region. That is want to know that is what are the changes in the neuron, because it is mainly concerned with that is the memory and everything. So we want to confine to that area only, hippocampal region. Okay, so that is this is a device, that is the capturing device, and we have used J software as well as that is the ocular micrometer to measure the neuronal diameter and everything, and the neural density, the square graticle we have used. This is the hippocampal region, that is this is the CA cornu ammanis 1, and this is the cornu ammanis region 2, and CA3 region, and this is the dentate gyrus dorsal blade, and this is the dentate gyrus ventral blade. We have examined that the neurons on these the 5 regions. Okay. Then in the kidney, that is we have measured the diameter of the glomerulus. Is that is the microoculometer, and the glomerular urinary space also we have measured, and the diameter of the PCT and the DCT, and the height of the cell lining the PCT and DCT we have measured. And in the testis, the seminiferous tubule, that is diameter and the density of the seminiferous tubule, the average number of the Sertoli cells per tubule, and the average number of Leydig cells per unit area also we have measured. Okay, so this is the testis. You can see the microoculometer to measure the diameter. This is to measure the density, the square graticle you have used, and this is all the 5 regions. That is, this is in the CA1 region. You can see here, this is the neurons in the pyramidal neurons in the CA1 region, and we have compared to the sham exposed and the 2G and the 3G exposed hippocampal neurons. So what we found here in the 2G and the 3G, that the interstitium is, you can see here, you can see the control group. So you can see the interstitium is wider. Okay. This is on the CA2 region. Again, that is the pyramidal neurons are less densely packed and the wide interstitium. So this wide interstitium is due to, that is, the cell phone radiation induced endothelial damage, and the more vascular permeability will lead to, that is, the wide interstitium. Okay, in the CA3 region also same, that is, we have found that is the wide interstitium. And this is the dentate gyrus dorsal region, that the granular neurons, you can see that is the 2G exposed and 3G exposed. Here also that is the interstitium will be a little bit wider, and here in that 2G exposed and 3G exposed ventral bed, there is not much. This is a silver stain, that is a sham exposed CA1, CA2, CA3 region, and the dentate gyrus dorsal blade and ventral blade. All the findings we have already, that is this is our publications. We have already, the JCDR journal, we have published the findings. And come to this liver, the histological study, we have found in the 2G and 3G exposed, that is the liver, we found there are many cytoplasmic vacuolation. You can see here, that is a lot of cytoplasmic vacuolations are there. That is the Fomin spaces, and many, that is the nucleus source, that is the pycnotic nucleus, and hepatic sinusoids were congested. Okay, and 3G exposed liver shows the more marked changes, and we have seen that is a lot of total hemorrhages in the, that is the liver parenchyma, and the more marked hemorrhages in the 3G. These all are induced, that is by the radiofrequency radiation induced, that is the endothelial damage, increased more vascular permeability, lead to these hemorrhages. Okay, so this is in the you can see that is in the periportal region. This is a portal, the portal triad. You can see that the periportal region, you can see it is infiltrated with the lymphocytes, a mixed type of both lymphocytes, macrophages, and neutrophils. It is more marked. You can see the lymphocytic infiltration, mixed reaction. You can see on the, that is the 3G exposed liver. Okay, we come to the kidney. That is in the 2G. This is the SAM exposed. This is the 2G and 3G. In the 2G, that is you can see that the glomerular capillaries are dilated and congested, and you can see that the urinary space is increased. But in the same thing in the 3G exposed group, the glomerulus are atrophied due to the radiation, and also the same congested glomerular capillaries. And because of the atrophied, that is the glomerulus, the urinary space is also reduced. Then you will see that is in the parenchyma. We can see that is in the 2G as well as 3G. That is the peritubular lymphocytic infiltration is also there, and also along the pericapillary infiltration is there. This is the capillary. You can see that is along the pericapillary, that is infiltration. This is you can see this in the 3G exposed, that is the pericapillary region, that lymphocytic infiltration here on the peritubular, that is the lymphocytic infiltration. This is more marked in the 3G. Come in the kidney also, you can see there are a lot of hemorrhages, focal hemorrhage in the 2G, more marked on that is the 3G exposed, that is the kidney. So indicating that is the radio frequency induced damages. Coming to the proximal converted tubules, in the proximal converted tubules, see the 2G and the 3G, the discontinuous or that is the loss of brush border lining in the both in the proximal converted tubule of 2G and the 3G. So the lumen looks like a wider one. Okay, and in the that is the this also you can see that is the loss of brush border appearance and the lumen appear more wide. Okay, so this is similarly that is in the peritubular, that is a lymphocytic infiltration in the both 2G and the 3G. And come to the distal converted tubule, the same thing, that is the you can see the cytoplasmic vacuolations, and also that is the pycnotic nuclei. You confirm that is the apoptotic changes in the cells. Okay. And come to the testis, that is this is the 2G exposed and 3G exposed. This is the sham control. In the 2G and 3G, the capsule is thickened because of the radio frequency radiation. Remember that testis is placed in the scrotal region. More the radiation exposure is more, and you see that in the 2G and the 3G, here is the sham exposed. See the interstitium is here; it is compact. So here you can see the interstitium will be very wide, both in the 2G and the 3G. And if you see the seminiferous tubules, here the compactness of this seminiferous epithelium, here the seminiferous epithelium shows the maturation arrest, and in the most of the peripheral tubules, there is no spermatogenic cells. The maturation is arrested here. Okay. Similarly, that is you can see the detachment of that is the Sertoli cells and the spermatogonial cells. You can see here that is detached from the basement membrane. That is the detached. Okay, so the 3G shows that is the extensive changes is there. Okay, similarly that is the vacuolar degeneration and disc formation in the seminiferous epithelium. Okay, so 3G shows that is the more marked change. So there we have conducted that Johnson testicular biopsy score. The normal in the compact that score was 10, and for the 2G it was 8 and 9, and for the 3G radiation exposed group shows that is the 7 and 9 only. So we have published the result. We have published that the effect of cell phone radiation in the paper. And come to the ovary, there is no marked changes. Okay, no significant changes in that is the ovary, because the ovary is kept in the abdomen, and maybe due to the hormonal effect. So the ovary we could not find any significant changes in the ovary. Okay, we have done the SDS page analysis. Okay, we have found that is the intensities of the band significantly higher in 2G and the 3G exposed mice, especially that is the 97 kDa protein expressions are decreased and 29 kDa protein expressions are unregulated in the liver and kidney. Also in the kidney, that 66 kDa protein expresses higher level, which all indicates that is the RFR induced damage, the protein expression. Okay, so we conclude the study with the following recommendation that it is logical to minimize the cell phone use, especially in the children and the teenage. Till further research on long-term exposure to the RFR confirm that is the cell phone radiation induced damage. The hand-free device we can use, especially the Bluetooth device we can use, okay, or the speaker mode we can use. So please never use the cell phone close to the tower, and similarly that is when the signal strength is very low. Children can be allowed to use play the cell phone games and such a thing after putting in that is the airplane mode. Okay, there are some limitations to our studies. We cannot directly extrapolate this outcome of the present study into the human population. That will be limited because of many reasons, due to differences in the species, the volume and size, the lifespan, and the anatomical organization of the tissues, and the duration of the frequency and the intensity of the transmission, and the shape of the exposed organs, and the water and the mineral content of the organ tissues, and the distance from the radiation source. So we recommend that more comparative study is also needed, and now that is already that is the 3G and the 4G studies has been done, and the reversal of the exposure, whether the reversal of the changes has been done, that everything has been studied. Now that option is we can go for the 5G cell phone and study on the animal tissues. Okay. Now come to the gross anatomy. That is just I will present a few thing about the variation in the upper abdominal organs. You all can see that is the celiac trunk. Okay, it gives typically the 3 branches. That is the left gastric artery. This is the splenic artery. This is the common hepatic artery. It will give the gastroduodenal artery and the proper hepatic artery. From there, that is the right gastric artery. So here the right hepatic and the left hepatic. The right hepatic artery gives the cystic artery in the calyx. That is a normal, isn't it? So here that is the knowledge of that is the vascular anatomy in the upper abdomen is very, very essential for the clinicians to perform that is a surgical procedure or the even angiogram and what is that either the gastrectomy or the hepatectomy is very, very important. That is so the success of surgery is solely rests on the vascular anatomy. The first step to that is the encounter in cholecystectomy is the cystic artery. So that is that much important is there for vascular anatomy in the abdominal organ. So just I will present a few that is the variation in the upper abdominal blood organs, that is the blood supply to them. You can see here that is the celiac trunk gives the left gastric artery. See that is the common hepatic artery. This is the proper hepatic artery is going to give only the right hepatic artery a small kick. That is the middle hepatic artery. So the left hepatic artery is absent. So the left hepatic artery is arising from the left gastric artery. Left gastric artery is going to supply stomach, that is the gastric branch, and the main contribution it is going through the hilum to reach to the that is the left lobe of the liver. So this is called that is the replaced left hepatic artery. Okay. So while doing a gastrectomy, please consider these replaced left hepatic artery should be intact. Please don't ligate proximal to that. Okay. To the replaced left hepatic artery. So please preserve that. The soul, that it is a replaced one. It is soul, that is the left lobe will be, that is the based on that blood supply of the replaced left hepatic artery. Okay. So this is similarly, that is, you can see here, that is the celiac trunk giving only 2 branches. This is postmortem specimen. That is the splenic artery and the left hepatic, that is the splenic artery and this left hepatic artery. That's not giving a proper hepatic artery. See, that is the superior mesenteric artery is giving that the right hepatic artery. It is replaced from that is the superior mesenteric artery. So it is the sole artery that is supplying the right lobe of the liver. Okay, so that is. See, this is the another thing, that is the celiac trunk. So the common hepatic artery is going to end as only the left hepatic artery. So the remaining branch is the gastroduodenal artery. So the right hepatic artery is not there. See, the superior mesenteric artery is giving the replaced right hepatic artery arising from the superior mesenteric artery, putting into the porta hepatis and going to supply that, giving the cystic artery and supplying that is the right lobe of the liver. See, the other thing that is the left gastric artery is going to supply that is this is the gastric branch and the esophageal branch. Along with you can see there is another tick that is going to supply the left lobe of the liver. Already that is the common hepatic artery end up with only with that is the left hepatic artery. The another left hepatic artery is arising here from here. So this is called that is the accessory left hepatic artery. Okay. So accessory to the main left hepatic artery. So such a variation will be should end. So here is the another one thing. You can see that it's a normal anatomical pattern only, vascular pattern only. But you can see here that is the right hepatic artery is making here that Moonigan hump or caterpillar hump. From the hump, that is the cystic artery is arising. Sometimes surgeon may mistakenly that is the ligate that is the right hepatic artery as a cystic artery, especially if the caliber is very less. Okay, so this is within the triangle. You have to identify the cystic artery, trace it. If it is that is a caterpillar hump, that is found out that it is the right hepatic artery. Please don't like it. This only artery should supply the right lobe of the liver. This is similarly that is the Moonigan hump or the caterpillar hump, a huge one. Okay, from the hump, the cystic artery is arising. Okay, so this is the angiographic studies. You can see that is the replaced right hepatic arising from the superior mesenteric artery. What we have seen in there, that is the gross anatomy. So this is once again the celiac artery. You can see that is it is giving the left gastric artery, giving the replaced left hepatic artery. Also there is you can that is the replaced left hepatic artery. Okay, so that is the angiographic study. I want to sum up that is the presentation. See that. So this is our research in in the effect of cell phone radiation on the animal tissue. That is we have that is noticed in the public gazette. So that is not public gazette in that is the newspaper, the New Indian Express. Our studies. There is the excessive use of mobile during pregnancy and childbirth well-being. So this is the one message we have to convey to the community. What we observed in the histological lab, what is our findings in the translational research in anatomy in the histological lab, the observations we turned into the intervention. We are giving the conclusion to the community or to the society. Please avoid that cell phone during the pregnancy and also in the early childhood to avoid that is the damage in the brain, kidney, liver. Okay. So we have presented that is the variation in the upper abdominal organ, the blood vessels. We have published that will be a lot of citations are there, and that will be great help to the surgeons, radiologists, and throw their hands. They are helping to the public. The communities are getting that is the benefit. Okay. So translational research in anatomy aims to provide that anatomical knowledge to solve the real world medical problems and enhance the patient care through the improved diagnostics devices and the treatments and the medical procedures. Okay. Thank you. If there is any doubt, I can answer for a few minutes. I think so. Thank you for patiently watching, everyone.
Thank you, sir.
Thank you, sir. Thank you, sir.
That was a wonderful session. We have a query in the chat box, sir. I'll read out the question.
Oh, okay, sir. Okay, sir. Okay, sir. I will answer him, sir.
Yes, sir.
Okay, sir. Okay, thank you.
By Dr. Shahjahan. Since the SIR consistency varies among mobile brands and models, the penetration of radiation, how are we going to measure during the process of experimentation?
Sir, actually, that is that each phone has its own SAR, sir. That is the specific absorption rate. When you are purchasing that mobile, you have to concentrate on that WHO recommendation. The SAR value should be that is the less than 1.69 per 10 grams. So this is the cutoff value. So it indicates that is the specific absorption rate per 10 gram of tissue. When you are using a cell phone, that radiation emitted from the cell phone, it will be absorbed through the tissues. So if it is a more specific absorption rate, say that is more than that is the 2 and 3, it means our body tissue is absorbing more radiation. So that can be measured by that is the radiation frequency meter. That is how much it has been absorbed. That we can that is the measure. Especially that is on the surface. That is in the topography, superficial tissue we can measure, but it is very difficult to measure in the deeper tissue, sir.
Thank you, sir.
Thank you, sir. Thank you, sir.
Query is answered. It's time to appreciate our resource person for today. We are really thankful to you, sir, and we present to you the e-certificate and the memento for the resource person.
Thank you, sir. Thank you, sir. Thank you very much.
It's also time to honor our chairperson for today. We also are happy to honor the e-certificate to Dr. and Dr. Shantini Arulselvi. Thank you, sir. Thank you, ma'am.
Thank you, sir. Thank you very much. Thank you for the opportunity to share my knowledge on the translational anatomy. Thank you, sir. Thank you, one and all. Thank you, all the participants and the organizers. Thank you. Thank you very much, sir.
Thank you.
Thank you.
Am I audible?
Yes, sir.
Yes, sir.
Just a moment, sir.
Can I share my screen or I'll wait?
Sir, your screen is visible, so you are visible, sir. Just be there, just waiting for some technical aspects, sir. Just
okay, fine.
Yes, sir. Good afternoon, all. Good afternoon, all. Once again, good afternoon, all. Today, day one, the fifth scientific session talk today by our eminent speaker, Dr. Nihar Ranjan Jana, who's a professor in the Department of Biosciences. He's going to present on the topic of Huntington's disease. For that, I would like to invite Dr. D. Amudraj to chair the session. Dr. T. N. Satyaprabha, who's also the another chair, due to some unavoidable reasons, she is unable to join us now for this moment. So only Dr. Amudraj will be chairing the session. Now I would like to introduce our chairperson, Dr. D. Amudraj. I am very happy to introduce such a humble person met in my life, and who's one of a senior to me. I'm very happy to introduce sir. He has completed his MBBS and MD from JIPMER in 2010, and he has finished his DNB physiology in 2011. He has completed ACME course, and he is level 2 anthropometrist from the International Society for Advancement of Canthrometry, and he has experience of 13 years of postgraduate training. And then his area of interest is research such as cardiovascular autonomic physiology, medical education, and medical instrumentation. Sir is pioneering in his institute, and he's very much interested in delivering the cardiovascular autonomic function testing as a patient care in his institute. I welcome you, sir. Now I would request.
Thank you.
I would request Dr. Amudraj, sir, to introduce our speaker, sir.
Okay. Thank you, Dr. Frank. Thank you for the very kind words that you have given to me. And at the outset, I would like to thank the organizers for giving me this opportunity to chair a session on a very important topic, that is the Huntington's disease, which is an autosomal dominant neurodegenerative disease that is affecting a sizable amount of population in the world. And unfortunately, we don't have a cure for this disease. And people are affected with motor disabilities, cognitive disabilities, and many of them have got psychiatric illnesses also. And ultimately, we lose them after a long period of battle. We lose these patients either mostly to pneumonia, and sometimes, I'm sorry to say, even to suicide. So it always, cries for attention, and it has been in the attention of the scientific researchers of the medical community for quite a long time. Even till now, we don't have a direct cure for this disease, and mostly, it's all going to be supportive symptomatic treatment of variable success. So really, we are in a junction where we need to look upon this disease, and for that, we are calling a very befitting scientific personality who is an expert in the field. None other than our professor, Dr. Nihar Ranjan Jana from the Department of Bioscience, IIT Kharagpur. I take a great deal of pleasure in introducing sir to the august audience here. Sir has graduated from Bindrapur College, and he got his post-graduations from Calcutta University. He did his PhD from Viswabharati University in the year 1996. Then after that, sir had an international exposure. He went to Japan, that is Riken Brain Science Institute, and he has done his postdoctoral work there. Then after returning to India, he has entered in a very premier institute, that is the premier institute of the country for neurological studies, that is the National Brain Research Center, as a faculty in the year 2001. He served as a faculty there, and up to 2018, he has done a lot of pioneering work there. And then now he has moved to IIT Kharagpur, where he has joined as professor in the School of Bioscience. Sir has received a number of awards for his work in this neurodegenerative diseases, and the list is quite a big number. So, with this introduction, I welcome our speaker, Dr. Nihar Ranjan Jana, to deliver his talk on Huntington's disease from genetics to pathogenic mechanism. So, the stage is yours, sir. Thank you very much.
Thank you very much. So can I share now my slides or yes? Okay.
Yes, sir. You can share the slide now, sir.
So I can share the entire screen or I can share only my, I think I can share my presentation only.
You can share screen, sir.
Yes,
After opening your PPT, you can put it share screen, sir.
All right.
Yes, sir. And
So I hope you can see my presentation, right?
Yes, sir. You can go to slideshow, sir.
All right. I think everything is fine.
Yes, everything.
All right.
is fine,
So
sir. Go ahead.
thank you very much for inviting me to give talk in this forum. So probably all of you are probably hungry at this moment, probably waiting for the lunch. So I will try to as brief as possible. Okay, so I am from IIT Kharagpur. So just a few lines about the institute, our institute. So I think all of you are familiar about IIT Kharagpur. So one of the premier institute and one of the top ranking institute in India, both in education and research. And it has actually more than 60 departments, centers, and schools. So you can understand how big it is. So if you see here my mouse point, so this is our actually newly constructed life science center. So all the people who works on basically biological field, so we all actually housed in here. And just to tell you that the IIT Kharagpur is also planning to start medical education shortly. So our hospital as well as other buildings are almost ready. So we are trying to recruit people to start the program. Anyway, so with all this, let me start my topic. So it's on Huntington's disease, where I am actually working last maybe more than 20 years on this disease on a particular aspect. So before coming to my area of expertise, let me briefly introduce about the disease and then the problem that we particularly focused on, right? So if you look at almost all age-related neurodegenerative disorders, you will find out one thing is very, very common. That's the pathological hallmark. And you can see here, that's the abnormal deposition of proteins. If you look at Alzheimer's disease, I'm sure all of you are very familiar about it. It's the amyloid plaque, which is extracellular. And you can see the neurofibrillary triangles, which is intracellular. Similarly, you can see in case of Parkinson's disease, the Lewy body, which is extracellular, intracellular component, but cytoplasmic. And you can see also in case of ALS and Huntington's disease, right? So there are a lot of debates going on for a long time about whether these abnormal protein aggregates can cause the neurodegeneration or not. There are completely two school of thoughts, and right now most people believes that the aggregates, not the aggregates actually, it's the early intermediates, which are actually kind of amyloid fibril-like structure. You can see here in the center. So those are probably the culprit. Okay, and they probably try to induce the degeneration, neurodegenerations by a number of ways. So the goal,
Sir, excuse me, sir. Sir, your audio feed has been cut.
Is it better?
Yes, sir. Now it's better, sir.
So what the current knowledge in this field is the aggregates, or mostly their early intermediates, are actually confer toxic gain of functions through a number of mechanisms that ultimately leads to neuro dysfunctions or neurodegeneration. So in my lab, what we do actually, we use Huntington's disease as a model system to understand how these abnormal proteins can induce neuronal dysfunction and neurodegeneration. So that's the problem. That's today's topic. So already, I mean, the already earlier person has introduced about the disease. So it is an autosomal dominantly inherited neurodegenerative disorder, and this is actually first discovered by George Huntington. You can understand this is 1872, more than 150 years ago. And that time, if you can read here, he described this is one of the incurable disease. And see, after 150 years, it is still incurable. Although there are a lot of progress have been made, but there is no therapy. I mean, real therapies exist. All right. So now, if you think about the behavioral phenotypes or the typical phenotypes or the problems that clinical problems that if you see, it at least 3 components actually primarily affected. So initial stage is maybe psychiatric problem, followed by cognitive as well as motor problem. But over the time, the motor problems are so severe. It's typically called classically is named is the Huntington's chorea. And it's very difficult for a patient to perform day-to-day activity. And this motor problem that classically in medical jargon it's called the chorea, Huntington's chorea, which actually happens, this kind of involuntary movement, because the specific regions of the brain is actually affected. You can see my mouse point. This is actually called choroid and putamen area. Is you can see in a Huntington's patient, it's almost not there. You can see if you see compared with the control. And because of the loss of that area, or more selectively, it is the loss of medium spine inhibitory neurons. And because of that loss, you see the involuntary movement that's classically called chorea. So now, as I told you, this is an autosomal dominantly inherited disorder. So people are desperately looked for what is the gene that causes this problem. What is the gene or gene mutation that causes this problem? And in 1993, the first disease-causing gene actually discovered by it's called Huntington Disease Collaborative Research Group. It's multiple peoples are involved, and one of the pioneering lady. I'll talk about the next slides how she become so popular and how she explored these things in Venezuelan populations. Let's first try to understand the discovered or the gene is actually called HD genes or Huntington's disease gene. It also another name. It's called IT15 gene. So IT stands for interesting transcript. So that's why I'm telling people it will be easy for maybe person to remember. And this is one of the largest gene in a human genome. There's a multiple exons and introns. Okay. And if you look at the mutation, they're also pretty interesting. So what sometimes we think is a point mutations or deletion, this kind of is not that kind of mutation. It's a mutation of abnormal expansion of a triplet codon. I hope everybody are familiar about what is codon. So a codon here is the CAG repeat, which codes for glutamine. So the mutation here is the abnormal expansion of CAG repeat in a coding region of this Huntington gene. So in normal individuals, the CAG repeats is highly polymorphic. Like in all of our normal individuals, repeat length might vary from 8 to 29. While in case of disease, it must be greater than 40 CAG repeats. So if somebody is having CAG repeats about 40, he or she might get the disease, say about 60, 65 years of age. And there is a direct correlation between the repeat length and the onset of the disease. If you look at the graph here, so greater the repeat length, you can get early age of onset. So in actual genetics, there's a classic problem which is called anticipation, which explains that the successive generation of the affected family is actually experienced early age of onset. Like if I have the CAG repeat 40 and I get the disease say around 65 years of age, my next generation, my next progeny might get the disease much early age, say about 60 years of age, and the repeat length would be high. This is typically called anticipation. All right, so that's what 1993 gene was discovered, and then there is a huge progress in understanding the disease biology. And in 1996, there's a first animal model of Huntington's disease appeared. It's actually made by Gillian Bates from United Kingdom. And the first, actually, this is one of the first, not the transgenic mice model for any neurological disorder you can think about, the first model system. And that mice actually showed a lot of behavioral abnormalities that are very, very similar, like a Huntington patient can exhibit. So I'll just show you the video of Huntington patient. You can just think about how the chorea looks like. It's an advanced stage of a disease. You can see how the abnormal movement can happen. Anyway, so I told you the 1996, there's a first animal model appeared, and which recapitulates most of the clinical problem that a Huntington patient can exhibit. And the subsequent year, in 1997, first report came that the mutant gene, or another way, the mutant protein is actually form aggregates. You can see here, this is the transgenic mice brain. That's the first discovered, first generated transgenic mice brain. If you look at my mouse one, you can see every cell, neurons have aggregates inside the nucleus. So this is 1997, same Bill Gates group. And if you simply introduce this mutant gene inside the mammalian cells, you can see here, this is the normal repeat length, normal CAG repeat length. And you can see here is a mutant protein, which has 150 CAG repeats. And you can see it form aggregates inside the cell. And subsequently, people have looked at patient brain. And actually 2000, much later, people have demonstrated that the mutant Huntington actually form aggregates in Huntington disease patient. All right. So with this, I'll just give you some idea which could be very interesting or could be, for young student. It's very inspiring about the discovery of this gene. I told you that the gene actually is one of the instrumental. The lady who was instrumental in discovering this gene, her name is, you see here, the Nancy Wexler. Her mother was affected with Huntington's disease. So she knew, because it's an autosomal dominant mode of inheritance, she knew she has a 50% chance to get the disease. But still, she didn't lose the hope. She was desperately looking for the cause of the disease, the gene that is involved for the disease. She went to Venezuela. In Venezuela, there's an area called Lake Maracaibo area, where almost every family was affected with Huntington disease. So went there, collected the blood sample, and that actually led to the discovery of the gene. All right, so there is a series of papers our lab published, and also other people have published. But unfortunate story actually came in 2020, not very far away. So the first report came, and the report was that she was affected with the Huntington's disease. So you understand, at the age of 74 years. So you understand, she discovered the gene and did a lot of groundbreaking work. And at the end of the day, she was suffering from the same disease. I wrote a line here that's very, 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 touchy story, of course, if you go and read. She never did take any genetic test that resulted from her own research. Can you believe this? So I hope this could be very, very inspiring for the young students, medical students, to think and to take, research as their integral part, which actually is seriously lacking in our country, in India. Most medical students actually go for their profession. Very few of them take the job as a responsibility as a researcher. Well, so with this, let me try to introduce the work that we try to follow up. So this, after the discovery of the genes, there are n number of models actually generated, including the animal models. So this is all the model that we use in our lab, the cellular model. So where we introduce the gene, normal gene and the mutant gene inside the mammalian cell. The system is called inducible system. The idea is normal conditions cell will not die because you can't express the protein. When you want to express this mutant protein, you can induce with a particular inducer. The gene will be expressed, protein will be aggregated, and cell will die. So this is how we can maintain the cell. So this is a stable cell line. All right. So there's another model. This is the model actually fast generated. So we have these mice. This is often called the Guillain-Barré mice, because she is the person first generated these transgenic mice. So if you look at the milestone of the disease, so you see about, first of all, this mice is generated, the transgenic mice, with 120 CAG repeat. It's a little bit higher than what you usually see in case of patient, right? Human patient. But you see the disease progression actually started, like if you see about 4 to 5 months of age, 5 weeks of age, the first aggregates begins. And the cognitive and motor problem or psychiatric problem begins around 8 weeks of age. And then the progression happens very rapidly. So 8, 10 to 12 weeks, this range, disease become very, very progressively increased, severity. And then by 120, 130 days, animal mostly die. So I'll just show you one of the simple, very simple behavioral parameters that anybody can do without any expensive equipment. You just hold the tail of the mice. So first I'm showing the normal mice. You see how it looks. So all 4 legs almost touching in the 4 different directions. Now, if you look at a Huntington disease mice, you see all 4 legs are coming together. This is a problem, or this is a phenomenon often called clasping. And this happens because of the damage or the dysfunctions of the basal ganglia. Typically, you can correlate this phenotype with a chorea that happens in patient. All right, and you can see the mice. You can see frequently the motor movement, and also sometimes the seizures. You can see the seizures happens in these mice. You see, the seizure happens. So this is the model system that we use to understand how these aggregates that I demonstrated can cause the neurodegeneration. Now let's try to understand the problem that my lab try to investigate. So you have seen these aggregates. Almost every cells or every neurons have these aggregates, and that is in the nucleus. It's a very classic problem. So now the question comes here. We detected this, the immunohistochemical staining of these aggregates by using a general marker, that's ubiquitin. So ubiquitin is a kind of a generic marker for any abnormal proteins aggregates. So now you can think about if any abnormal proteins are detected by this ubiquitin, which simply you can think or indicates the proteins or cells actually trying to degrade these abnormal proteins through a mechanism that is shown in the right side called ubiquitin proteasome system or autophagy. So if that happens, if cells are trying to clear these abnormal proteins, the question comes, then why the form over the time? Is this that the cellular, the control mechanism, the degrading controlling mechanism is failed? So that is the questions that my lab in the last 20 years is trying to explore. So the question that we address, how this abnormal failure or abnormal degradations actually happens, or why cell cannot degrade this abnormal protein inside the cell, in neurons. That's the mechanism that we are trying to explore. I hope you understand what question that we are trying to address. Now, if you understand that, how can we understand or how can we address this problem? So if you look at this ubiquitin proteasome systems, it's a very complicated pathway. I'm not going to describe that. But just look at the first phase, that's called ubiquitination cycle, requires 3 enzymes. There's a single E1 enzyme, but there are multiple E2, you can see here, conjugating enzyme, and E3, which is the ligase. This might be boring for many of you. Okay, so, but just think about the single E1, but there's multiple E2 and E3. This E3 is the ligase, ubiquitin ligase. And in any cell, you might expect 800 to thousands of different E3 ubiquitin ligase exist. It's not a single one. So you have to remember there could be 800 to thousands of ubiquitin ligase in a single cell. So when I joined to National Brain Research Center in 2001, my project was to identify which is this among this 800 or 1000 ligase, which is the ligase, or the multiple ligase could be, which are the ligases that are involved in ubiquitination of this abnormal protein. Okay, and we identified, I'll go forward. So we identified a ligase which named ubiquitin ligase, which is called UBE3A, quite uncommon probably for most of you. It's a ubiquitin ligase. I'll explain one by one what it does. But you look at slide here just to show you here that this ligase is associated with this, abnormal aggregates that are found because of the mutant Huntington, not only in the cellular model that I described, also in the Huntington disease transmitting mice. You can see almost every aggregates are actually recruited with this ligase, which indirectly suggests that this ligase might be involved in the ubiquitination of this abnormal Huntington. So we dissected out, I'm not talking about, we dissected extensively, and we find out that this ligase indeed involved in the clearance or in the degradations of this mutant Huntington. Okay, but because of the length of the mutation or length of the CG repeat, if the length of the CG repeat, or in other way you can think about when it translated into the protein, the length of the glutamine is high, this ligase would find hard to degrade them. If the length of the polyglutamine is shorter, UB3A can degrade it much faster, while if the length is larger, it can take more time. But it does degrades.
So now if you look at the Huntington disease mice sample, you see the different part of the brain. What we noticed is this ligase is dramatically low. Protein level, the active level of this ligase, if you look at here, wild type versus disease mice, wild type versus disease mice, every part of the brain, if you look at the level, the protein level of this ligase is dramatically dropped compared to the RNA level. RNA level is almost nothing all the time. I mean, his expression level is not changed while his protein level is changed, which indirectly indicates the ligase activity is almost gone. And that probably, that time we try to explain because it might be recruited to the aggregates. So going to the insoluble fractions. So most soluble active ubiquitin ligase, UB3A, is probably not accessible to this mutant protein for the clearance. So that's why probably aggregates are forming as you patient goes older and older. All right. So now, so you might be having questions about what is this protein about? What is this UB3A? I told you it's a very unusual name. Well, say of course it's a ubiquitin ligase. And another fascinating aspect of this, because my subsequent slide will explain that, is imprinted in the neuron. What does it mean? So it's the paternal copy in our brain. So if you look at almost all of the genes, it's a maternal copy and a paternal copy. Both are biallelely expressed. But in this case, this UB3A is actually maternally expressed, which means the paternal copy is actually silenced, epigenetically silenced in the neuron. I hope I'm able to explain it. In all of our brains, or in other way, we can think in all of our neurons, if you think about UB3A, it is the mother's copy that is expressed. Father's copy is epigenetically silenced. I hope this problem is clear. And now we can look at if there is a loss or defect or mutation in the mother's copy, there is a developmental disorder which is called Angelman syndrome. I'm sure many of the medical students might have heard this name. It's a developmental disorder which shows learning and memory deficiency. And recently, people have shown if there is a overexpression of UB3A, there could be chance of the child might get autism. So you understand how or the importance of this ligase. Other than neurodegeneration, it has heavily linked, or it is heavily linked with developmental disorder. I hope with this we can go much forward. So our intention, what I told you or what I showed you, this ligase is involved in the degradation of mutant Huntington. So what next we can do? So what next we actually did, this is just to show you the imprinting thing happens. If you look at here, we have another mice that's called UB3A maternal deficient mice or maternally knocked out mice, where you can see there's a clear absence of this ligase. I hope it's clear. That's what I was explaining, the imprinting phenomenon. You see the normal brain, normal mice brain, UB3A is expressed and it's pretty nuclear, localized. But once you maternally knocked out, you only knocked out the maternal copy genetically. Paternal copy is there, right? But that is epigenetically silenced. That makes absolutely absence of expression of UB3A in the neuron. You have to understand it can express everywhere in the body, but you get a restricted knockout in the neuron. I hope that problem is clear. If you are clear of this, then it's easy for you to understand what we did next. What we did actually, our goal is to generate a Huntington disease mice where UB3A is knocked out. Is that clear? That's our idea. Idea is if you remove the UB3A from a Huntington disease mice, what could be the phenotype? And you can expect if it degrades the mutant protein, then the disease progression will be much faster. That's what you can hypothesize. Is that going to happen or not? So we use that typical imprinting strategy. So you see there is a male Huntington disease mice crossed with female this UB3A maternal knockout mice. And then you do all sorts of screening. It's very complicated ways you have to screen. And then you will find a group of mice, group of Huntington disease mice where UB3A is knocked out. You can see this last lane. That's the characterized. Fine if you are not okay, but just simply think we have generated a Huntington disease mice from where UB3A is selectively knocked out in the neuron. I hope that's clear. And you then look at the phenotype. This is, if you look at this back bar, the black line, this is the typical lifespan of a Huntington disease mice. They can survive hardly
120 days maximum, right? But if you knocked out, this is the knocked out, TB3 knocked out Huntington disease mice. If you knocked out from the neuron only, you see the lifespan is almost half. By 60 days, they are all dead, which indirectly suggests that this ligase is playing very, very important role, crucial role, to some extent. And then you can see in body weight, that also dramatically dropped compared to a normal Huntington disease mice. All right. So then you can look at some of the behavioral phenotype, whether the behavior of these Huntington disease mice are really aggravated or not. I'll just show you the same clasping video, which I showed you earlier. You just see, the thing is going to work. All right, never mind. I think there is a problem of link probably. All right. Can you hear me?
Yes, sir, you're audible, sir.
I am audible, but I guess there is some problem of video. Oh, either my I think I lost my link, right?
Yes, sir.
Oh. So what I need to do?
Sir, you can restart your presentation, sir, slide, and then you can share it, sir. Reshare, sir, you can reshare. Yeah.
Can we switch off the timer for time being?
Yes, sir, we will do, sir.
Yeah.
Sorry, due to some technical error, he got logged out, sir. He has joined again. Sir, yeah. Sir,
Is it okay now?
Yes, sir.
Yes, sir.
But I still cannot see the pop-up. Oh, let me try. I cannot see either slide is here, right?
Yes, sir. The screen is already shared, sir. You can.
We are able to see your screen, sir.
Okay. I am not able to.
Yeah.
Yeah.
Yes, sir. Yes, sir. Put in slideshow mode.
So it is now okay, right?
Yes, yes.
Yes, sir. Maximize the screen.
All right. So I stopped it here. So I think I will not show the video. So I think that's the problem. So what I am trying to explain is the motor behavior or motor phenotype of these mice, that UBE3 deficient Huntington disease mice, is actually more and more severe. All right. So if I think this is working now. So if you look at simple water test, this is actually the Huntington disease mice. Sorry, this one. See, it's fall down. It's the UBE3 deficient Huntington disease mice, and this is the only Huntington disease mice which will perform much better than the earlier mice. All right. So I'll not show it. And then we did a lot of molecular studies, which I'm just showing few of them. One of the interesting things that you should be seeing is there is not a single neurons in this mice diet. I mean, died by apoptosis or necrosis, whatever you say. It's very interesting. Not a single neuron. You see all those absurd behavioral problems, which is very severe. And mice dying also, but not a single neuron actually died. So what we noticed, that's such surprising for us. What we noticed is only the neuronal architecture. You can see here, neuronal arborization or the dendritic branchings, those are actually severely affected. And that probably leads to the behavioral abnormalities and probably subsequently die. Many of the mice die with epileptic seizure. And this is what actually we wanted to see. See, look at here. This is a pre-symptomatic HD mice. We just started showing aggregates, very early stage, about 43 days old mice. You can see few of the aggregates in the nucleus, but you see that EB3 deficient mice, where you can see the aggregates number is quite high in the different part of the brain, which indirectly kind of suggests or indicates that this ligase is involved in the clearance or degradations of mutant huntingtin. I hope that's clear for to everyone. So we have an in vivo proof in animal model that this ligase can be able to clear this. Well, so with that, then we again next went forward. So we actually try to screen some of the drugs, some of the molecules, which can actually target EB3. All right, so there are a lot of failures, so that we cannot slow down. We actually screened a lot of library, chemical library. And I'll just show you one of the compounds which somehow it worked. And that's one of the molecules you can see here called azadiradione. Its significance is this molecule is present inside, highly concentrated in the neem seed, in our medicinal plant neem, we all know. And the seed of neem seeds actually have high level of azadiradione. It's very accidental studies, so findings. So you can see here, the EB3 level actually increased in the cell lines once we add these molecules. So then we went ahead to study in animal model, the Huntington disease mice model. And you can see here, the level of EB3A in wild type mice also increased, as well as in Huntington disease mice. You look at, this is the Huntington disease mice, 3 mice, you can see here, the level of EB3A is quite low. That is I showed you earlier. And also some part, it's called some molecule called DAP32 is also significantly low. That's immaterial, but if you look at the azadiradione treated mice, Huntington disease mice, you see the level is rescued quite normal level. And that's the proof. Even you might have doubt whether this molecule can cross the blood-brain barrier or not, but it simply can tell this molecule cross the blood-brain barrier. Well, then we also did some behavioral studies. It's not that dramatic changes, but you see they're easy. If you look at this black bar, which actually shows the body weight gradually decrease in case of Huntington disease mice. But if you see this drug-treated mice, the dose is about 10 milligram per kg body weight. This typically rescued the body weight. And also, that's most interesting thing that you might be looking at is the the lifespan. If you look at the black bar, that's the typical lifespan for a Huntington disease mice. And this drug-treated mice has significant improvement of the lifespan. So this is kind of very, very exciting for us because we have not really optimized much because we only used 2 doses, 10 milligram and 20 milligram. And we could see much better in case of 20 milligram. But we need much more extensive studies to show that there is not much toxicity of this drug in the mice. And that could be much more safer. So those works still we need to conduct much more extensive way. But at least this findings is clearly pointing out with a mechanism that this molecule is increasing the level of EB3A, and that might be involved in the enhanced clearance of the mutant huntingtin, and ultimately leading to extended lifespan. So this is my last slide. This is the future. You can think about this molecule could be a potential therapeutic molecule that we are looking for. Let's see. And this is a summary. I already told you. I think I should not explain, but just briefly tell about that we have identified a EB3A is an important ligase which not only could be useful in case of Huntington's disease, the abnormal protein clearance, this could also be involved in other diseases like Alzheimer's or even Parkinson or ALS. So that's the implications, and then at the end we find out small molecules which have, I mean, for Indian context, has huge importance, could be involved in treating this kind of disease which has almost no therapeutic important drugs that can cure the disease or halt or slow the disease progress. So with this, I'll stop my talk, and this is just to acknowledge my funding agencies and all the collaborators, everything I just mentioned. I think who introduced me. So I was working around 19 years in this institute. It's a beautiful institute, beautiful campus in very close to Delhi, and this is my new building where I am now working. So thank you very much for your attention, and I'm sure all of you probably hungry, so just probably waiting for the lunch. Well, so I'm done.
Thank you, sir. Thank you so much, sir. I think today we have something new take-home message of UB3A, sir. Now I request the chairperson, Dr. Amudraj, sir, to give his comments and queries, sir.
Thank you very much, sir. Thanks for the very enlightening talk. Actually, so far we have been very unidirectionally looking only at the cytoplasmic inclusion, that is the Huntington, and you have taken us from that into a totally new, different this thing. So, sir, as a not as a chairperson alone, but even as a common delegate, what are the different techniques that a person has to upgrade himself to do this kind of studies? So, what are the techniques that he has to learn?
Well, first thing is animal behavior. So you can see that that's not very complicated. I think very simple tools that we use to study the motor deficits, like rotor rot, clasping, then gait analysis. These are very common, routinely used techniques that anybody can use, and it's not so difficult. That's the first part. And second part is the molecular tools. That sometimes may be a little difficult because once you try to maintain the mice, just to maintain the mice without any experiment, you need a genotyping. So you have to cut the tail, extract the DNA, then do a PCR. That's the regular tools you need to think which mice is your transgenic mice, which mice is your old mice, I mean normal mice. Then you have to do a lot of molecular tools that commonly what we use. It's not an expensive things. I mean, there's very routinely used molecular and biochemical techniques that we use, like Western blot, RT-PCR, real-time PCR, immunohistochemical staining. So it's very common. I think everybody is familiar about that.
So usually many of our MBBS students, they used to come and ask, sir, is there a facility in our country or nearby where for a summer vacation we can go and learn few techniques and come? So is there anything like that in NBRC?
I think NBRC has that. NBRC has a program with even doctors. Actually, with the AIMS Delhi, they used to have a program. So MBBS or even the MD students used to come to the NBRC for a few days for their experience, and then they could have experience and then could go back. There was actually formal training. Now I think that's stopped, but certainly MBBS student can even come. Actually, in my lab there are 3 or 4 MBBS students did PhD, and two of them actually from Niemans Bank. They did from there, and they did PhD in our lab. So there are people. Even in MBRC, there are, other than this, we have a imaging facility. We have a fMRI, so that also you can give a different experience. Certainly, you are most welcome to visit MBRC.
Thank you, sir. Thank you. Dr. Frank, if there is any question from the audience, we can ask, sir. Otherwise, we can collect it.
No, sir. There is no questions from the audience, sir. Thank you, Chairperson, sir. Thank you, Speaker, sir. For another thank you for opening the observership doors for the MBBS students to NBRC, sir. I think there were many applications will be waiting to come as an observership, sir. And now so for this beautiful talk, now we would like to appreciate you with an e-certificate and then virtual memento, sir.
Thank you.
Thank you, sir. I would also now like to appreciate our chairperson, Dr. Amudraj, sir, who is a family member of our conference team. Thank you, sir. Thank you, one and all. Now there will be a lunch session, and post lunch we will rejoin the conference with YRS presentation, Young Research Scholar presentations, and the oral free paper presentations today. The links have been dispatched separately into the group. Please kindly join in the specific links. Thank you all.
Thank you. Thank you very much.
Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. Welcome to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education, we established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future. to Vinayaka Missions Group, where education meets healthcare excellence. Our academic landscape spans across 13 colleges and 11 schools, situated in 4 vibrant campuses. We are proud to serve the Union Territory of Puducherry and extend our reach to Salem and Chengalpet districts in Tamil Nadu. Pioneers in private medical education. We established the first private medical college in Puducherry back in 2000. Today, we operate 2 medical colleges in Puducherry, where we provide healthcare services to an astonishing 4.5 lakh outpatients and over 50,000 inpatients annually. We manage 9 rural health centers, where we attend to approximately 1,000 patients every day, ensuring quality healthcare for all. And now, as we embark on our next level in healthcare, we are proud to introduce the Vinayaka Karkinos Oncology Institute, or VCOI, in collaboration with Karkinos Healthcare. Karkinos Healthcare is a purpose-driven, technology-led oncology platform, revolutionizing cancer care in India, with a primary focus on early detection, precise diagnosis, patient care, and groundbreaking research. Through setting up of the Vinayaka Karkinos Oncology Institute, the aim is to bring comprehensive cancer care closer to the communities under the distributed cancer care network. Our mission: early detection programs through community screening programs, identifying high-risk patients and guiding them throughout their entire care journey. The facility will be equipped with state-of-the-art diagnosis and treatment options. And as part of our commitment to the people of Puducherry, Vinayaka Karkinos Oncology Institute aims to screen nearly 1 lakh population within the next few months. This collaboration between Karkinos Healthcare and Vinayaka Missions Group, including Vinayaka Missions Research Foundation, in setting up of the Vinayaka Karkinos Oncology Institute, VCOI, in Puducherry and Salem, shall be a testament where education, research, and affordable and accessible cancer care come together for a brighter, healthier future.
Hello. Hello. Good afternoon.
Good afternoon. We'll start in another 2 minutes.
Okay, fine.
Hello. Hello.
Yes, sir. Hello.
So this is hello. Hello, sir. Can you hear me?
I'm able to hear.
Yeah, actually, I sent my people to your email address. I haven't said from share the screen.
Yeah, we are doing it.
Ganesh Babu 3703.
Yeah. I'll do.
Are you understood? Sir,
Yeah,
got the mail, sir? Yeah.
Welcome you all for this YRS faculty presentation.
Yes, sir.
Hello, sir.
Yeah.
Sir, there is no share the screen option here. So how are we going to share the PPTs?
Your name, ma'am? Yeah, 1 minute.
Driver again.
1 minute, I'll promote it to panelist group so that you can get that option.
Okay, okay. Thank you. So when are the presentations going to get started?
Yeah.
Good afternoon.
Good afternoon, ma'am.
When are the presentations going to get started?
Yeah, we are starting right away. Some technical problem. Yeah, we have with us for YRS faculty presentation 3 judges. Dr. Shivlila C, Professor, Department of Anatomy, Sri Siddhartha Medical College. Welcome, ma'am.
Good afternoon, sir. Thank you.
We have Dr. Ponnudali, Professor and Head, Department of Biochemistry, Vinaya Mission Kribanandavarya Medical College, Salem. Welcome you, ma'am.
Thank you, sir, for the.
And we have third judge, Dr. S. Ravi Kumar, Associate Professor, Head, Department of Biotechnology, AVMC, Pondicherry. Welcome you, sir. Sir, you're there. Yeah, sir is there. And we'll start with the first presentation from anatomy. Shah Sumaya John. Are you ready, ma'am?
Yes, sir.
You have the sharing option here.
Sir, could you please, I don't have the share option, sir. I have already shared my PPT with you through the mail. Could you please share that?
Yeah, then give me some 2 minutes, ma'am.
Yes, sir.
Dr. Priya
And there is
Marina.
no video option as well, where I could turn on my video. If it's not necessary, then it's okay.
No, I'll see to that. We'll call the next participant, Priya Marina.
Yes, sir.
You're there with your PPT, ma'am?
I have also emailed my PPT. If it is to be shared, I can share the screen.
Yeah,
but I cannot see the option.
The same here. I also cannot see the option.
Ma'am, I have given option to promote you as panelist. Please accept that so that you'll come into the panelist list so that you'll get the option also. You'll get a pop-up in your screen itself.
Yes, sir.
Yeah, please accept that. Dr. Ganesh Velluri. For all the presenters, I'm giving that.
Yes, sir. So shall I start? Shall I share my screen?
Yeah, fine. Your BMSECON
Okay,
ID
fine.
is anatomy 271. Am I correct?
Okay, fine, sir. Sir, wait, I'll be sharing. Desktop, can you hear desktop? Desktop, computer. Computer share, share. Computer business save kari. So please give me some time. Fine.
Yeah.
Sir, is it getting shared or not?
No.
Meeting चला share. So can you see my screen?
No, ma'am. Is there any other? Or else the PPT I'll get it from the department. Is there any
So
other
please get
presenters
it.
with the? Yeah. Is there any other presenters with their PPT who can share?
Good afternoon. Am I audible?
Dr. Ganesh Velluri, or you are? Can I present your things? You can do now.
My name is Adi Shankaran. I'm from Department of Biochemistry. Am I
Yes,
audible
sir.
to you?
Yes. Yes, sir. Ganesh, I'm ready to please share the screen.
Yeah, first we'll go with Ganesh Velluri.
Please share the screen.
Yeah, is it visible for you?
Yes.
Is it visible for you, Dr. Ganesh?
Yes, sir. Yes, sir.
Yeah. Please,
Can
judges.
I start?
The participant is Dr. Velluri Ganesh. His ID bio 215.
Yes, sir. Yes.
Can I
It
start?
is,
Yeah.
the name is Rajakumaran T.
No,
Bio ID.
Velluri Ganesh.
Bio
215.
215.
215. Yes.
Yeah.
Can I start?
Yeah, you can proceed, Doctor.
I'm Dr. Velluri in Department of Karnataka. So I'm going to present one of my research topic. So title is correlation with magnesium vitamin levels with microalbumin for oral related property in nutrition diabetes.
Sorry to interrupt. Your voice is not clear.
Velluri, your voice is breaking up.
Your voice is breaking.
it clear now? Sir, this is Ganesh, sir. Is it clear?
Now it's low.
Sir, is it clear?
Yeah.
Sir, is it clear, sir?
Yeah, it is clear now. You can proceed.
Hello.
Yeah, it's
Yeah,
clear.
is it clear, sir?
It's clear now. You can proceed, doctor.
Sir, yeah. So can I start, sir?
Yeah, please start.
Yeah. Good afternoon, all. Here, Dr. I am Veluri Ganesh, Department of Biochemistry, ESIC Medical College and Hospital. So here I'm going to be present one of my research topic, correlation of serum magnesium, vitamin D, and adiponectin levels with microalbumin for early detection of nephropathy in patients with type 2 diabetes. So please change the slide. Coming to the introduction, type 2 diabetes mellitus is a chronic metabolic disorder due to two major abnormalities, including insulin resistance and dysfunction of insulin production, leads to chronic hyperglycemia. The prevalence of diabetes in worldwide population was 366 million by 2011 and expected to reach 550 million by 2035. In Indian scenario, 40.9 people millions were affected with diabetes, and potentially this number will be increased to 85 million by 2030. Apart from hyperglycemia, the other factors like obesity, dyslipidemia also contributes to increased free radical production and decreased antioxidant level, leading to diabetes mellitus and its complications. Please change the slide, sir. So diabetic nephropathy. Diabetic nephropathy is one of the major microvascular complication occurs due to multiple risk factors like hyperglycemia, obesity, smoking, dyslipidemia, and hypertension in patients with type 2 diabetes mellitus. Globally, in 2015, the prevalence of nephropathy in type 2 diabetes mellitus was 20 to 40%. Among in this Indian population had 36.3%. Microalbuminuria is currently considered as gold standard and is the earliest clinical available marker for detection of diabetic nephropathy. Nephropathy starts with normoalbuminuria, microalbuminuria, and progresses with microalbuminuria with kidney failure and multiplication of central diseases. However, it has having low availability are the main albumin in the urine without diabetes mellitus. Hence, the kidney most in the body and it's having essential tyrosine kinase activity and to insulin resistance and poor the type 2
Voice is
diabetes
not clear.
mellitus.
Voice is not
Vitamin
clear.
D is the fat soluble.
Dr. Ganesh, your internet.
Ma'am, now, ma'am. Now,
now
ma'am.
Now it's better.
No, it's okay.
Vitamin D is the fat soluble vitamin and act on the beta cells of pancreas, results insulin production, diabetes mellitus and its complications. Sir, can you change the slide, sir? Adiponectin is the adipocytokine synthesized from adipose tissue, other cells like skeletal muscle cells, hepatocytes, colonic mucosal cells, and myocytes. It has anti-inflammatory, anti-atherogenic, and anti-diabetic properties. The metabolic imbalances of adiponectin cause insulin resistance and poor glycemic control in the type 2 diabetes mellitus patients. We aim that the correlation of serum magnesium, vitamin D, and adiponectin levels with microalbumin for early detection of nephropathy in patients with type 2 diabetes mellitus. Sir, please. Coming to the aim, the present study was taken up to correlation of serum magnesium, vitamin D, and adiponectin levels for microalbumin for early detection of nephropathy in patients with type 2 diabetes mellitus. Objectives: to measure the adiponectin levels in different stages of diabetic nephropathy among type 2 diabetic patients we compare with healthy controls. To measure the serum vitamin D levels in different stages of diabetic nephropathy among type 2 diabetes patients compare with healthy controls. To measure the serum magnesium levels in different stages of diabetic nephropathy among type 2 diabetic patients compare with healthy controls. Correlation of HbA1c, urinary HCl with serum magnesium, vitamin D.
Voice is not audible. Mr. Velluri Ganesh.
please come to the type 2 diabetes status as per American Diabetes
Your internet is dropping in.
No, sir. No.
No, it's not. Am I audible? Dr. Ganesh?
Yes, sir, you are audible. Dr. Ganesh is not audible.
I think he has lost his connectivity. Dr. Ganesh?
Now,
Can you hear me?
sir. Can I start, sir? Can I continue? Yes.
Yeah. Can you go to the
30.
Next slide? Yeah, proceed.
Sir, please change the slide, sir. Sir, please change the slide, sir. Coming to the inclusion criteria of the study. So, age is, all the subjects' age is greater than 30 years. The patient diagnosed with type 2 diabetes mellitus based on American Diabetes Association criteria. American Diabetic Association criteria. So, in different stages of nephropathy. Coming to exclusion criteria, the patients with type 1 diabetes mellitus, non-diabetic renal diseases, so urinary tract infections, individuals on thiazolidine diones, anti-inflammatory and immunosuppressive drugs, thyroid and liver diseases. The patients who have macrovascular complications such as cardiovascular, cerebrovascular, and peripheral vascular diseases were excluded from the study. So I'm coming to the sample collection. Two different blood samples were collected from all the subjects. 5 ml of fasting blood sugar, 3 ml of after food for postprandial blood sugars, and also we collected spot urine samples was collected from all the subjects. All the separated samples were transferred into appropriately labeled liquids and stored at minus 50 degrees centigrade until biochemical analysis was done. Please change the slide, sir. Sir, please change the slide, sir. Coming to the methods, the fasting blood sugar, postprandial blood sugar, urea, creatinine, and HbA1c, magnesium was analyzed by using laboratory standard methods. Serum vitamin D was analyzed by chemiluminescence immunoassay. Microalbuminuria was analyzed by albumin creatinine ratio. So here adiponectin was analyzed by enzyme linked immunosorbent assay method. Sir, please change the slide, sir. Coming to the statistical analysis, data distribution was tested using Kolmogorov test. Continuous variables are expressed as mean plus or minus standard deviation. Comparison between the two groups for continuous variables were assessed using independent sample t-tests. Pearson correlation analysis was done to test correlations among the markers. Statistical analysis was performed using Microsoft Excel spreadsheets, SPSS software for Windows version 16. The p-value is less than 0.05 was considered as statistically significant. Sir, please change the slide, sir. Coming to the results, table 1 shows the comparison of biochemical parameters in patients with type 2 diabetes mellitus and healthy controls. The parameter cholesterol, HDL, so microalbumin, serum adiponectin was significantly elevated in type 2 diabetes mellitus patients when compared to the healthy controls. The magnesium and vitamin D when compared to the healthy controls, the p-value is less than 0.001 is comparison of biochemical parameters in type 2 diabetes mellitus with normal albuminuria and type 2 diabetes with patients with microalbuminuria.
I think he's having a internet problem. So, can we proceed with the candidate now, or like?
Yes, sir.
Or we'll ask him to switch out to discussion or conclusion. Right? I think he has lost his connection.
Yeah.
Sir, could I please present my presentation after this?
Right. What shall we do with this participant?
Sir, can we go to discussion?
Sir, actually we are in ESIS, sir. Here we won't get this proper signal, sir. So, sir, correlation.
Yeah, already we
Sir.
have reached 10 minutes. Just go to
Yes,
the
sir.
slide with the permission.
Okay, sir. Okay, sir. Okay, sir. Please.
Yeah, discussion.
Yes, sir. So, but, can I start?
They are asking to tell the salient points alone and finish.
Okay, sir. Yes, sir. Yes, sir. Please go to the. Can I start, sir?
Yeah, shall I go to conclusion slide, or you want to tell something in discussion?
Yeah. No, sir. No, sir. Go to conclusion, sir. No problem, sir. Because I have issued step of the sir.
Yeah, this is.
Thank you, sir. In the present study, were included that the reduced levels of serum magnesium, vitamin D, and increased levels of serum adiponectin levels was observed in patients with type 2 diabetes when compared to the three groups of studies. Increased serum adiponectin because of tubular injury, and also it is useful for preventing magnesium and vitamin factors like type 2 diabetes mellitus and its complications. Thank you.
Dr. Velluri Ganesh,
Yes, ma'am.
how did you calculate the sample size?
Sample size, ma'am. According to the previous studies, R values, ma'am. We calculated, ma'am, in online by using software.
Okay. It came out to be 90?
Ma'am, by using software, ma'am, online software, ma'am, we calculated by using previous studies R values, ma'am.
It was a cross-sectional study, no?
Yes, ma'am. By mistake, actually, in the materials and material we mentioned case control study. It was my mistake from my side.
Okay. And what do you say about the adiponectin levels? Adiponectin levels are increased, no?
Yes, ma'am. Adiponectin levels are increased in both the groups of type 2 diabetes mellitus. The normal.
Is there any difference between type 2 diabetes and nephropathy? Adiponectin levels?
Yes, ma'am. Actually, it is having renoprotective effects, ma'am. It is prevent the damage of kidneys by activation of AMP kinase pathway.
Renoprotective effect, but it has been increased, no? But it has been increased in nephropathy. That's what you're telling, no?
Yeah. It is increasing maybe so to protect damage of the kidney. To prevent the damage of the kidney.
To prevent the damage of kidney, it is increased in nephropathy.
Yeah. For prevention purpose, maybe. Not only for our study, even we have references, other studies also. It is maximum studies which is mentioning and reported increased serum adiponectin levels were observed in nephropathy conditions.
Okay, thank you, doctor.
Thank you.
Sir, could I please present my PPT now? I can share the screen.
Please wait a moment.
Okay, sir.
Any other questions from the other judges?
Okay, sir.
Dr. Ganesh, your presentation is good, but because of the interruption, maybe it was not very clear.
Yes.
I'm not a subject expert, but slides were good.
for the interruption from my side, ma'am. Actually, we are bound to get the signals from here. So we are in the forest area, ma'am. That's why.
Okay. No problem. Good presentation.
Thank you, ma'am. Sorry, ma'am. Sorry for the interruption from my side. For all the judges.
Ravi sir, is there any questions from your side?
No, sir.
Fine.
It's okay.
Then we'll go with the next presenter. We'll
Yes.
finish off the biochemistry. Dr. Kirtika, are you ready with your presentation?
Yes, sir.
Yeah. Can you share it from your side?
1 minute, sir.
Yeah.
Could I please present because I have a doctor's appointment for my child?
Yeah, the screen is visible. You can proceed.
Yes, sir. Thank you. Good afternoon, all. I'm Kirtika from tutor in Biochemistry Department in Savita Medical College. I presented potential antimicrobial activity of green synthesis copper nanoparticle and characterization. So the introduction, the copper nanoparticles are one of the most significant transition metal oxide in nanotechnology because it is simplicity and eco-friendly and potential as next generation antibiotics. Then its synthesis using green chemistry principle in gaining transaction. At the same time, I use coriander leaf has accounted the numerous pharmacological exercise like cell reinforcement and antimicrobial because its cost effectiveness and lower toxicity and remarkable broad spectrum antibacterial activity against the range of bacteria through the generation of reactive oxygen species and release copper ions. To assess antimicrobial activity, green synthesis copper nanoparticles, coriander leaf is the aim, in the objective to determine the physicochemical characterization of copper nanoparticles and to assess in vitro antimicrobial activity of copper oxide nanoparticle against multi-drug resistant bacterial strains. Materials, before we know about the preparation of coriander leaf extract, so 20 grams of dried leaves of coriander taken into 250 ml beaker with 100 ml of distilled water and then boiled for 20 minutes at 80 degrees Celsius. So copper nanoparticle synthesis to depend on the copper sulfate or use starting material and the reduction is carried by coriander leaf extract from copric ion to copper oxide. So in the synthesis of copper nanoparticles is based on 10 ml of coriander leaf extract was added to 100 ml of aqueous copper sulfate solution. It kept this flask at room temperature as overnight and the copper nanoparticles were separated and settled at the bottom of the solution. So finally, the copper nanoparticles obtained purified for the repeated centrifugation method at 5000 RPM for 15 minutes, followed by the derepression of the pellet in deionized water. So copper nanoparticles were dried into oven at 80 degrees Celsius. Methodology, physicochemical characterization of copper nanoparticles. So first in spectroscopy in UV visible range, I use double beam spectrophotometer in the range of 400 nanometer to 700 nanometer. So it is examining the optical characteristics. And infrared spectroscopy using Fourier transform, it is examining the Perkin-Elmer with a spectrometer recorded in 400 to 4000 centimeter. It generally identifies the capping and efficient stabilization of metal nanoparticles. So next in diffraction of X-rays, so copper potassium radiation in two ranges from 20 degree to 80 degree. So this X-ray diffract method was used as the crystallized size, structure and crystallinity of nanoparticles, followed by the data analysis in PowerX software. Then one I used for transmission electron microscope because the size of produced particles were determined with the help of TEM. At the same time, we analyzed for various magnification and screening of copper nanoparticles. In vitro antimicrobial activity of copper nanoparticles in multi-drug resistant bacterial strain, I follow clinical laboratory standard institute guidelines. This is under two method is kit by disc Bureau diffusion and agar-well diffusion method. In the gram positive is Staphylococcus aureus, Staphylococcus mutans, Staphylococcus pyogen and Staphylococcus epidermidis used. Then gram negative is Escherichia coli and Pseudomonas. Each strain was tested around 50 liters of the test sample. So based on the concentration of copper nanoparticles is 1.25 milligram per 50 liter. It is kept for 37 degrees Celsius and standard positive controls is 50 microliter aqueous extract of coriander was used as positive control and 10 microspectromycin disc for gram positive and 10 microgram norfloxacin disc for gram negative strain. So antibacterial activity was assessed using the zone of inhibition measure after the incubation period against each tested or microorganism. Time kill determination, the mixture of nanoparticles were also tested in investigate possible antibacterial activity and this minimize the potential toxicity and resistant problems. So the copper oxide and sub-minimum bacterial concentration was used to killing assay against following strains for S. aureus strain, MRSA and S. aureus Oxford NCTC 6571 and S. termitis SE51 and E. coli SCTC. So all the nanoparticles were prepared in phosphate buffer saline with sonication. At the zero time, each microorganism was added to the nanoparticle suspension and dilution of 1:80. So inhibition was carried in shaking inhibitor in 200 RPM at 37 degrees Celsius in the air for 4 hours. After that, the plate were then incubated at 37 degrees Celsius in the air with carbon dioxide for 24 hours. It is statistical analysis are using one-way ANOVA. Result, the pH of the copper sulfate solution is 2.16 and the coriander leaf extract is 6.78. When I added the coriander leaf extract to this solution, the pH will be changed to 2.16 to 2.64. So I confirmed that the capping between the copper and coriander leaf extract was taken places. Next one, the UV visible, the reduction of copper sulfate to pure copper nanoparticles for monitoring with ultraviolet visible spectrophotometer. So UV visual spectrum of copper nanoparticle disappears is deionized water at room temperature is peak at 560 nanometer. And the diffraction of X-rays, the sample demonstrated at high crystallinity level. The diffraction level is 22.74 degree to 33.12 degree. The size of copper nanoparticles was found in the range of 21.66 nanometer to 41.65 nanometer. The FTIR, the biosynthesis of copper nanoparticles were recorded in identify the capping and efficient stabilization of metal nanoparticles by biomolecules present in coriander leaf extract. So band at the 3,500 centimeter to the correspondent of alcohol and phenol. So in the band at the 1,575 centimeter is correspond to the primary amine. So 3,500 to 3,000 to 25 bands are responsible for the presence of phenol, flavonoids, aromatic amino acid. So the band at 600 centimeter is corresponded to presence of copper. So synthesis of copper nanoparticles was surrounded by the protein and metabolites such as phenolic acid, carboxylic acid, and flavonoids, etc. So I confirm the phenolic compounds have stronger ability to bind metal, indicating that phenol could form the metal nanoparticles to prevent agglomeration and thereby stabilize in the medium. Transmission electron microscope to demonstrate that the nano copper particles is exhibited for axis shape with no sharp edges is observed. So the particle size was determined to be the range 22.4 to 94.n nanometer. Minimum bacterial concentration at kill time is using this kill assay, the population of gram-positive and gram-negative organisms tested were reduced by 68% to 65% is presence of 1000 microgram ml nano copper oxide within the 2 hours. The population of the S. aureus and the MRSA, the epidermidis were reduced to 0 by 4 hours in the presence of 1000 microgram ml nanogram of copper oxide. The conclusion, copper nanoparticles appear the joint to microbial cell surface and enter inside and were intracellular target and including the respiratory catalyst and disturb. So the copper nanoparticles suggest to release of iron may be required for optimum killing. Future plan to evaluate effect of copper nanoparticles on bacterial genome and their genotoxicity. These are the reference. Thank you.
Yes.
Yes, ma'am. Over to you, ma'am.
Shall I give my opinion? Yes.
Yes.
Kritika, the study is very well conducted and a very detailed explanation you have given. But I have some doubts regarding this. Can only coriander leaves can be used for this nanoparticle synthesis, or any green leaf extract can be used?
Any green leaf extract, we're using the copper nanoparticle extract, ma'am. But very cheaply, and at the same time, we're using the coriander is every day our food. So only I choose the coriander leaf. Other leaf is also contain nanoparticle, copper nanoparticles is presented in other studies.
No, in what way this coriander leaves in our food is related to your study?
Every day we taking the coriander, ma'am. Some bacterial resistant coriander is copper oxide is highly presented in the coriander. So it is also helping to prevent some bacterial infection, other disease.
Okay. But with your with your study, what is the scope? How it can be applied in medical treatments? Because nanoparticles have some adverse effects also, no?
Yes, ma'am. Nowadays we
and how far it is superior to? Is it comparable to the antibiotics? The zone of inhibition you talked about, no? Have you compared it with regularly used antibiotics, or only you're using it as isolated assays with copper nanoparticles?
It is isolated part, ma'am. It is a suitable replacement for antibiotics. Otherwise, the disinfected part. Nowadays, more studies are going on the copper oxide nanoparticles. It is compared to other drug. It is very fast moving to target the particular cells and phagocytosis action.
But it has some adverse effects also, no?
Yes,
immunotoxicity, genotoxicity.
Yes, ma'am. Yes, ma'am. So only I plan that future plan. We evaluate the copper nanoparticles on bacterial genome and their genotoxicity. So it is very suitable replacement of antibiotics and disinfectant of the medical clinical side.
All this you expect, but we do not know how
Yes,
the
ma'am. Yes, ma'am.
Okay, that's all from my side, sir.
Thank you. Dr. Ravi, sir, is there any questions?
Hello. Kritika. So my question is that what do you think, what is the significance and the composition of coriander leaves in your study? Did you check what are the compositions on the coriander leaves?
Flavonoids, phenol, primary amines, aromatic amino groups. So these are all generally presented, and the flavonoids, phenol.
The flavonoids and other things have been present in all other plant materials.
Yes, sir.
The reason for choosing this coriander for capping agent, no?
Yes, sir. Because
why did you choose this one?
Because in the band at the in the FTIR test, the copper it will be presented, sir, highly presented, indicate the band at 600 centimeter. So it's correspond to presence of copper in the coriander leaves. So some other studies or coriander leaves highly content in the copper. So only I checked that. After that, the nanoparticles are presented or not. The copper nanoparticles are presented or not. So only I do this test.
No, in your FTIR analysis, you have done only once, it seems, and the peak which you got, it also doesn't have any functional group explanation has not been provided in that in the one run actually. How many times did you run the FTIR?
Sir, only one time I ran this part.
Okay. No, the thing is that no, you have done only once, it seems, right? Okay. You have checked the same also, that is also showing 28 micrometer.
Yes.
Okay. When you prepare other time, the size will be varied.
Okay, sir.
So what kind of optimization is needed for your study actually? Did you done any optimization study in developing the nanoparticles? And another question is that what is your actual application of your study? So you have developed the nanoparticles. What will be the application of your study? As the madam said that already the antibiotics are available. So what your newly developed nanoparticles will do actually?
It's a suitable replacement of antibiotics. So it's compelling bacterial microorganisms.
See,
It's easily
antibiotics will target on the specific microbes. Okay.
Yes, sir.
How does your antibiotics? Did you done any kind of targeting studies for a particular bacterial species? If you use these nanoparticles for all microbes, the good bacteria also will die, right?
Yes, sir. Yes. Yes.
Then how can it can be a replacement for antibiotics?
Yes, sir. The next study only the future plan is particular practical bacterial genome, the effect of copper nanoparticles in bacterial genome and genotoxicity.
See, actually, if you take any heavy metals like zinc,
Okay.
copper, that will be a toxicity to any of the species. It is toxic to ours also. Okay. How you are going to encapsulate it? The capping is very, very important. That's why you are using different type of biological material for capping. For that also you need a proper characterization is needed for your studies. Actually, you have to do FTR. You need to do Raman spec. So there are several characterization, and you have to optimize the single size or shape. It should be a single size or it should be a single shape. But whatever you do will have what do you say? It's kind of different size. You will be getting different shape. You will be getting those things cannot be taken to the market actually. So that's you need to understand it. Okay.
Yes, sir. Yes, sir.
Thank you from my side.
Thank you, sir. Thank you.
Thank you, sir. Is there any questions from Dr. Shivrila, ma'am?
No, sir. Yes, they can proceed. Okay.
Due to medical emergency, like one of our participants, like Dr. Sumaya, she wants to present next. With the permission of judge, can we go ahead?
Yes, sir. Let her present.
Yes.
Shah Sumaya Jan, anatomy 271.
So is it visible?
Yes, it's visible. You can put in slideshow mode.
Okay. Slideshow, fine.
Yeah.
Is it fine, sir?
Fine.
Okay. Good afternoon, everyone. I'm Dr. Shah Sumaya from Government Medical College, Srinagar. from the Department of Anatomy, working as a demonstrator. Today, the topic of my presentation is anatomic differences in patellar dimensions. It's a comparative study of left and right sides. Coming to the introduction, the patella is a sesamoid bone that is formed within the tendon of quadriceps femoris. And coming to the contribution towards the knee joint, it forms the femoropatellar component. It has various variations in the position. We can call it patella alta when it's higher in position. It's attenuated patella alta when it's abnormally small above the knee joint. And patella baja, which is positioned very low and may hinder the knee extension. Coming to the structural features, if we see it from the front, it's rough with vertical ridges from the tendon of the quadriceps femoris, the fibers that are coming from the tendon of the quadriceps. The articular portion, the facets for the articulation with the femur's patellar surface. And in the medial border, it presents an odd facet in contact with the femoral condyle during the extreme flexion. And coming to the apex, it's lower and the non-articular part, and it attaches to the patellar ligament. Now, what was the need for the study? There was an existing gap. There was lack of data on patellar morphology in the Kashmiri population. So Kashmiri population is a unique population with distinct features, ethnic group, and the genetic and environmental factors are there. While coming to the geographic context, the Kashmir is located in the northern India that is bordered by Pakistan and China. Now, relevance, we have to understand the patellar morphometry that is crucial for the diagnosis and treating the knee pathologies. Now, what was the aim of the study? This study will involve the collection of dry human patella from the individuals of the Kashmiri origin and the measurement of the various morphometric parameters, including length, width, thickness, and the angles of inclination and rotation of both the left and the right sides. Coming to the materials and methods, it was a cross-sectional study that was done in September 2022 to December 2022 in Government Medical College, Srinagar, on the ethnic Kashmiri population. We arbitrarily took 42 patellas. Among them, we had 22 right and 20 left. We included the patellas that were from the ethnic Kashmiri population, and we excluded all those patellas that had any congenital abnormality or that had any fractured bone. And before doing the study, we sought this ethical clearance from the ethical committee. Then Coincue's classification, it provides the foundation for the patella's morphological classification that's based on the measurements of the lateral and the medial patellar facets. The patella were divided into groups A, B, and C. The parameters used in the study were patella's height, width, and thickness, the length and the width of the medial and the lateral articular facets, as well as their analysis were measured. A measurement of the central ridge length was also made. The digital vernier caliper were used to take the measurements. The length of the patella was measured from the superior point as the uppermost point to the inferior point, whereas the width was measured at the broadest point that is perpendicular to the length axis. Regarding the thickness of the patella, it was measured at the mid-patellar point, which is the thickest point of the bone. Now coming to table one. Table 1 shows the descriptive statistical analysis, including the mean, maximum, median, and standard deviation. As shown in this table, no parameters showed a significant statistical difference between the right and the left patellas, except for the slightly higher mean patellar height on the left side. Coming to the table 2. Table 2 shows a statistical difference while comparison of the medial and the lateral joint facets of the right side. Table 3, it shows a statistical difference while comparison of the medial and the lateral joint facets of the left side. The lateral facets were found to be wider than the medial facets in both Table 2 and Table 3 on both the right and the left sides. Now coming to Table 4, the Coincue's classification, according to that, here the majority of our patellas, they were falling into the group B. That means the articular facets on the lateral aspect were larger than the medial aspect, which is also supported by Table 2 and Table 3. Now coming to our conclusion, The results of this study show that the average height, width, and thickness are 4.10 centimeter, 2.01 centimeter, and 4.1 centimeters respectively. The information provided in this study may be useful for the forensic evaluation and the ethnic morphometry of patellar specimens. This study determined the important dimensions of the patellar bone in a North Indian population, which may provide guidelines for the fabrication of the patellar prosthesis implants. Thank you so much. Thank you. Shall I stop sharing?
Yeah, you can stop sharing. The presentation.
Okay, fine. Fine.
Yes, Dr. Sumaya.
Yes, ma'am.
All these were available in your department, 42 patellar?
Yes, sir. We have. Actually, we had 46 patellas. Among them, I discarded 4 because they had some, they were chips of bones and they were fractured. So I didn't include them in my study.
And one of the inclusion criteria you mentioned, all were taken from ethnic Kashmiri population.
Yes, ma'am.
How was that actually?
Ma'am, actually, we have 2 lots of bone sets. We have dead bodies and we have the bones that we extract from the population of Kashmir, because we know that we are going to conduct the studies. So that represents our Kashmiri population. So we keep them grouped together and we mark them, we label them. And we have other bones that we get from outside. So we keep them aside. We don't take them because it will fabricate the data.
Okay. Then one more question from my side.
Yes, ma'am.
So you had taken articles for reference, right? So
Yes,
any differences are there in your region dimensions and from other studies which were done in other parts of India?
Ma'am, actually, there was no such study was done in Kashmir. It was done in outside in other parts of India. So I thought that there is a lack of data that is in Kashmiri population. So I thought that it would provide some data to the Kashmiri population. That's why.
Okay. So what is the significance of this?
Ma'am, actually, if we get to know, because from our statistical analysis, we have seen that we got to know that according to the classification, the articular facets that are on the lateral aspect, they are larger than the medial aspect. So according to that, first of all, these bone studies, they help in the forensic evaluation. And number 2, because patella is the most important bone in the knee injuries, or like in the old age, they get injured. So for the prosthesis, for the implants, we can get a clearer view so that we can plant them or we can manufacture them according to that. That may be of some help to that.
Yeah, you can try to sexing also in these.
Yes, ma'am.
So it can
Yes,
add
ma'am.
some more
I can get that.
information.
Yes,
Okay.
ma'am. Yes, ma'am. I can
Thank
do
you.
that.
Good
I'll do
presentation.
that. Thank you,
Good
ma'am. Thank you so
presentation.
much. Thank you, ma'am.
Thank you, ma'am. Is there any questions from other users?
Dr. Sumaya, I have one
Yes,
question.
sir.
You have a very good presentation.
Thank you so much, sir.
So how it will be useful in forensic?
Sir, because here we can, if we have like unknown bodies, that can be like there is some encounter anywhere or some accident emergency in the forensic. Like if we have a clearer idea of what our ethnic Kashmiri population, we got a data. According to that, we got a data. So we can frame out like in the forensic, we have a data like this particular bone, or because something is like that has a connection with the ethnicity and a particular group of population. So this way we can like.
So
it will contribute to that.
We need a earlier data, right?
Yes,
to
sir.
understand. So you said
yes,
that's
sir.
the first of its kind in India, right?
Yes, sir.
Okay. Did you publish this paper?
Sir, not yet.
Okay, good.
Okay. Thank you so much, sir.
Good presentation, Dr. Sumaya. But I have
Thank
certain
you, ma'am.
doubts. I have certain
Yes.
doubts in your study.
Yes, yes, ma'am.
Did you compare the patella morphology with other ethnic groups, so that you can
Yes,
say
ma'am,
it
I
is very specific to your group, to your Kashmiri population?
Yes, ma'am. We did because as I have already shown that in table 1 there was no significant statistical difference, and while in table 2 and table 3 there was a slight statistical difference. There was like the lateral facets were found to be more wider than the medial facets. That was shown in table 2 and table 3. And according to table 4 in our study, most of our patellas, they fell into the group B. That showed that the lateral aspect is larger than the medial aspect. So that had a slight significant difference was with the other ethnic population of other parts of the zone.
So actually you didn't compare, you just took the data, like how it differs from the other populations, no?
Yeah, yes, ma'am.
And do you have the history of the petala? Just doing a study with the petala without any prior history, how do you correlate? The age, sex, does it matter or not? Like Madam was asking, we should have noted down the sex of the individuals also, no?
Yes, ma'am. We can add that in the, I can further extend my study and I can see it in the further study. I will extend it.
Any
Sure.
bone or any structural component without any history of who it belongs to, how can you tell the validity of your results, no? It should be comparable.
Yes. Yes, ma'am.
And only you have done in your population. You don't know how it, based on studies you say, but it is not a part of your study, no?
Yes, ma'am.
All that should have been, if you do that, it will become, your study will become more valid.
Yes, ma'am. Surely I'll do that in future. Thank you so much. Thank you so much for the insight. Thank you, ma'am.
We'll call the next participant, Priya Marina. You are ready?
Yes, sir.
You'll share from your end, or I should?
I can share from my end.
Yeah, I have a presentation also. If you want, I can. Yeah, it's visible.
Is it visible?
It's visible. It put in full screen mode.
Is it fine?
Please put in slideshow mode.
It's on slideshow. Can you see?
No, madam, it's not reflected here.
Where is that option, sir? Like I can see
top ribbon, home, insert, design, animation, slideshow, or at the bottom you can see here. You have to zoom. Yeah, next to that, slideshow, design, animations, and slideshow.
Priya, you stop sharing. You stop sharing, and then you can share it in the without screen mode, actually, without slideshow mode.
Or shall I do it from my end?
Is it visible now?
Now it's okay.
Yeah. Very good afternoon to the respected judges and all the delegates present for the international conference. It's my pleasure to present part of my study at the Young Research Scholar presentation among the co-presenters. So thank you for the opportunity. My study is on determination of sex by hand dimension and finding index and ring finger length among the students of Kerala origin. I'm hailing from Anapoya Medical College, Mangalore, from the Department of Anatomy. Next slide. Hello.
You have shared from your end, I think so.
Not able to change. Yes. The aim of the study is to identify the sexual dimorphism by studying hand length, hand breadth, and hand index and ratio of the index ring finger length. And the second objective is to study among the variables which can better predict sex. What is the need for the study? This study will be helpful in establishing the biological profile of individuals in forensic investigation. This study has implication in mass disaster, criminal cases, wars, and other disasters which are happening where an isolated hand is recovered and needs forensic identification. An individual's identification is the most important part of the forensic investigation. It is prevalent to discover the dismembered human remains and peripheral sections of the body with growing frequency of disasters. A person's features will restrict to a geography, and an individual's hand can probably provide useful data about the stature, sex, and age when retrieved and taken for examination. So sex determination from hand dimensions can greatly assist forensic scientists in identifying human remains. These are some of the literature reviews which shows the earlier studies which are done in different parts of the world. GSE et al. In Korea, he has done a study of 321 measurements of males and females and analyzed to investigate the sustainability of detailed hand dimensions as discriminators of sex. Around a total of 29 variables were included in his study, including length, breadth, thickness, circumference of fingers, palm, wrist were measured. And according to his study, the result showed a highest accuracy of 88.6% for predicting sex for males and 89.6% for females. Kanchan et al. Also did a study in North and South India using 500 females and did a similar calculation as well, where the variation is seen in different percentages. Ishaq et al. Conducted a study to determine the sex using dimension of various hand parts and handprints collected from 200 Australian subjects, wherein there is a similar measurements which are done of hand breadth and length were reported to offer highest identification accuracy. As well as another study was done by Abol Hagag et al. Attempted sex determination by using variables such as hand length, hand breadth, and index finger versus ring finger length ratio. And in this study, he predicted the hand index, hand breadth, and length, which showed 80% of accuracy for males and 78% for females. Now, I have concentrated to study the same in the region of Kerala population because as per the study, as per my review, there are a lot of students which are located around this region of Venapoya Medical College, and I thought it would be a great contribution towards the forensic medicine if we can have something done for an age group of 18 to 20 years. So we have taken the study group which is conducted in Venapoya Medical College and with the 50 undergraduate students of Kerala origin, including both male and female, age group of 18 to 20 years. Hand length and breadth, index finger and ring finger measurements are done using digital vernier calipers. And these are the parameters which are used. The length of the hand will be assessed. Now, how do we assess the length of the hand? It is a straight line between the distal crease of the wrist joint and the tip of the middle finger. Second dimension is the hand width. It will be assessed as a straight spectrum from the second metacarpal sides, most laterally placed point, the most medially placed point of the fifth metacarpal surface. Third one is the hand index, where we do a hand width versus hand length into 100. That is a measurement we get for the hand index. Fourth one, the index finger measurement, where the index finger length will be measured, tip of the index finger to the metacarpophalangeal crease. And the ring finger measurement, the tip of the ring finger to the distal metacarpophalangeal crease. So this figure shows how the measurement goes about. For the calculation, all the measurements were statistically analyzed using software and Microsoft Excel. A t-test was used to compare the hand length, breadth, and hand index, index finger length, and ring finger length. Descriptive functional analysis was done. These are my results, which you can see. This is the table 1, which shows measurements and calculation of right hand. There you can see the length, width, index length, and ring finger length, which is measured. Mean and standard deviation is calculated. And you can see that p-value is less than 0.001, which shows it is statistically significant. This is the measurement for left hand. You can see the length of female and male, width of female and male, index finger length measurements of female and male, and ring finger measurements of female and male. The statistically significant differences in right and left hand measurements of male and females are evident in the above table. We can see that females have a very particular measurement in all the calculations, whereas male measurements are little higher compared to the female. This indicates the hand dimensions are can be better used as a predictor of sex. In case we find an unidentified hand, in case of any disasters, this would be a great contribution for them to compare the lengths and to determine which geography the person may belong. The following table gives a canonical correlation coefficients for the right hand. You can see the correlation for width, length, ring finger and index fingers. This is for the right hand, and this is a correlation coefficient for the left hand. So we compare the same as a standard values, and we have used a formula for this. This is the formula Fisher linear discriminant to calculate. How do you calculate the left hand measurement? This is a LF is for left hand of female, and this is a calculation where after discussion with my statistician, this is the calculation we have come up and the formula we have come up with. And this is the left length measurement in case of females. LF is for female and M is in males. So we replace the exact values, length of the right hand, width of the right hand, index finger, length and ring finger measurements, and according to this calculation we get a coefficient which is compared with the standard values, and if these values are correlating with if it is greater or lesser than if it is a female or male otherwise. And I come to a conclusion that right hand, left hand dimensions, when they are used separately or together, they give almost same accurate predictions. So either if we have one hand or two hands, either with one hand we can also predict what this particular hand may belong to which sex, if it is a male or female. That is what the sentence says. Evidence favors to right hand dimensions than left hand dimensions for prediction. That more right hand dimensions are more predicted well than left hand dimensions. When both hand dimensions are used together, the accuracy improves only by 2%. So there is only difference of 2% when you calculate together and separately. So the accuracy is just a difference of 2%. So the results may be skewed because the male-female ratio is not 1:1 here in my study. So it may vary accordingly. So in summary, the developed equations offer a reliable method for predicting sex based on right hand dimensions, and the identified predictors contribute significantly to the accuracy of the predictions. These results may have a greater implication in various fields such as forensic anthropology or medicine, where sex prediction based on physical characters is relevant. So that's my contribution to this study. These are my references. Thank you so much.
Thank you. Move to judges.
Yes, Dr. Priya,
Yes,
you are working in Mangalore, right?
Yes.
So you have taken hands of Kerala origin students, right?
Yes.
So what was the sample size?
We have taken 50 students, 50 undergraduate students.
So 50 students. So how many were male? How many were female among the 50?
We have 37 male and 13 females.
13 females.
Yeah.
So both the hands. So same right and left
Yes,
hands
yes.
of these people only.
Yeah, yes.
Okay. Sample size is comparatively less, I think. No, you could have taken.
Yeah. It's the 37 females and 13 males.
It is a cross-sectional study, right? So how did you calculate the sample size?
We took it from the other studies, ma'am, but due to the time constraints, actually, we couldn't do more of it. We are extending the study to further 200 samples.
Okay.
Yeah, yes.
One more thing you could have done. You could have taken other students, like you had students from Karnataka also and other places also.
Yes.
And you have compared the dimensions of different states.
Yeah,
so
okay.
it would have been a good study. Or
Okay,
you have taken references. You have taken references from the studies where they have done from only Kerala. So you could have compared that.
Okay.
Like you have taken the references in common, right? Not only from the students from the Kerala. You have taken
No.
students, right?
We also have, yes.
Yes. So I think you cannot compare once you take the different places references. So when you are taking dimensions from only Kerala students, so you can do a comparative study in your place only with other people, and then you can compare. That will be a better study, I think so.
Yeah, yes, ma'am. We are planning on to continue it further in like different groups and different age groups as well.
Yeah, increase the
contribute
sample
for-
size also. Increase the
Okay.
sample size. Yeah, that's from my side.
Thank you.
Thank you. Is there any other questions? From other judges?
What is your future plan from this?
Sir,
What will be your future plan from here?
future plan is to publish the results so that it would be of some help
I'm
in
just talking about how you going to extend your project from here onwards.
extend, you mean?
Hmm.
We are planning to take up, as I said, more sample size, up to 250 students, including equal number of male and females, so that results will be more accurate, as well as compare it with our region as well, because I've just restricted it to Kerala origin. I can compare it with the Mangalorean origin as well, and because if there are any other regional people there of the same age group, we can compare with different regions.
So how long it will take for a person to measure? I mean, whatever you are measuring, the length or height or weight now. So
per person, yeah, per person it would take maximum of 15 minutes for all these measurements.
Okay, then you do it everything manually, right?
Yes,
so
it's by using digital vernacular.
Okay, my suggestion is that why don't you implement artificial intelligence in your so that you can able to collect a lot of data and analyze that your data, so that it will give more direction. I mean, positive direction, where you can take it further to a much larger sample size, more than 2,000 or 3,000, where you can able to reduce your statistical deviations. Okay.
Yes,
so this
sir.
is my suggestion. So you can take it forward by using, now a lot of softwares are available and a lot of algorithms are available. You can choose any, you can talk to any person who is expertise in artificial intelligence, and then you can take your project towards that direction. That is my
Yes,
suggestion.
sir. I can try to collaborate somewhere with our research center. We have the people working for artificial intelligence.
Thank you.
Thank you, sir.
Dr.
Yeah. Yes,
I
ma'am.
have two basic doubts in your study.
Okay.
First of all, the statistical relevance. With 37 males and 13 females, I think it is quite difficult to come to a conclusion like what you have given. No?
Yeah. Yes, ma'am. Actually,
minimum. See, you can. How can you defend your sample size? Because it's sort of a declaration, or you're trying to say something new, no? That
yes,
females index finger, ring finger, that palm width are less comparable compared to males.
Yes.
In that case, your female sample size should have been equal to males or even a little bit higher than males. No?
Yes, that is what I mentioned.
And
that the
I
ratio
think
is
it is very difficult to even come to a conclusion with this sample size. Such a major difference.
Okay, ma'am. We are, yeah.
Yes. See, at least it should have been equal. No? To come to any conclusive evidence, and you cannot say it as a general. Is it generalizable with this sample size? First of all, variation in sample size. And how did you calculate the sample size?
Sorry, ma'am?
How did you calculate the sample size? This 37, 13, or just like that you took 50 students and you came to your
we actually had taken a group of students that available for particular batch.
Actually any research should not be conducted that way, to my knowledge. First of all, you should fix a statistical number, sample size, and then only you should go about with the study.
We wanted to take around 250 students, and but just because of the time constraints, like for this presentation, I couldn't finish all of it. So I'm extending it to around 250 students again.
And one more basic doubt I have. Did you take the height and weight of the individuals into consideration? Did you standardize the students or the individuals before you say that the finger size is different from females and males? Some males can be males and females
the
is
tallest. Yeah, ma'am. That is also one of the parameters which has not been mentioned, I believe. Yeah, sorry for that. We have taken that into consideration because the taller the person is, the dimensions will change accordingly.
No, I don't want to know whether it is taken into consideration. The research means you should have definitive data, no?
Yes.
What sort of standardization did you do? It's all evidence based, and you cannot just say that we have to take it into consideration. What? Just my doubts.
Yeah, it is not mentioned in the calculation.
How did you standardize? How did you standardize the height and weight?
We
of
no, I mean,
individuals?
we are just referring it, but not mentioned anywhere in the calculation.
It's just we're just taking up a hand, which can be male or female. The person can be bulky, tall, short. Based on that,
yeah, we are just trying to take up the parameter of determining the sex itself, because according to those calculations, if we can determine if it's a male or female, that was the focus of the study.
Without standardizing, I think it's very difficult to come to a conclusive evidence. You please concentrate in those areas if you want to publish it or continue with this research.
Okay,
thank you.
Thank you so much.
Thank you. We'll go with the next presenter, Dr. Adi Shankaran, Bio 414.
Sir, am I audible to you?
Yeah.
Yes, sir. Good afternoon to the organization. First, let me share the screen, sir. Is the screen visible, sir? Is the screen visible to you?
Yeah, it is visible.
Okay, sir. Okay, sir. Okay. So shall I start?
Yeah, proceed. We can start.
Okay. Good afternoon to all. Good afternoon to the organizing secretary, organizing chairpersons, and the judges and delegates. I am Dr. Adi Shankaran to present my work on the effect of particulate matter PM2.5 in tuberculous granuloma formation and reactivation of latent tuberculosis. As we all know that after inhalation of mycobacterial tuberculosis, that bacilli, only 10% of the individual general population develop active tuberculosis, whereas in the rest, the bacilli resides in the dormant state in the form of granuloma. And WHO and some other studies also says that prolonged exposure of PM2.5 is associated with more TB fatality, as well as exposure of PBMCs and airway epithelial cells to the pollutant affects host innate immune response. But no study have observed the direct impact of PM2.5 in granuloma formation in vitro, as well as reactivation of latent bacilli, which is contained in the granuloma. So with this, we had two objectives for the study. These two objectives, if we see the clinical relevance, it is like two clinical scenarios. The first subjective, the first scenario is like there is a person who is exposed first time to the tubercle bacilli. Simultaneously, that person is exposed to the PM2.5 also. So the objective one was to evaluate the effect of pollutant PM2.5 on the formation of PBMC's granuloma using MBOV's BCG. And the second objective is like another scenario in which there is an individual who is already having the latent tuberculosis in the form of granuloma. But if this individual is exposed to PM2.5, what will happen? So the objective 2 was to assess whether PM2.5 can reactivate the latent mycobacterial bacilli contained in the preformed PBMC granuloma. So in order to execute these objectives, we had to standardize few things. First, we had collected PM2.5 in Teflon filter, and that was extracted by sonication, filtration, and lyophilization. This stock concentration was made. And PBMCs were isolated by using density gradient centrifugation. Now, these isolated PBMCs were subjected to different concentrations of PM2.5 to see whether this PM2.5 is itself causing any cytotoxic effect to the PBMCs, and that this was done by using MTTSA. And simultaneously, MBOV's BCG culture was done, and the granuloma was induced with this PBMC and MBOV's BCG in the extracellular matrix with MOI, that is multiplicity of infection 0.1, which means one bacilli is required. One bacilli is required for 10 PBMCs. This is the minimum criteria to form a granuloma. And these granulomas are then harvested in the actual experiments and were subjected for human cytokinesis, histopathology examination, gene expression studies, and colony forming unit enumeration. So this slide shows how I induced the granuloma and how I harvested. So first the PBMCs were isolated from the study subjects and the viable cells were counted. These viable cells are then mixed with the extracellular matrix made up of predominantly collagen and fibronectin. And the PBMCs were added at a ratio of 5 lakh PBMCs per 50 microliter of the extracellular matrix, and they were dispensed in the 96-well tissue culture plates. Simultaneously, when the MOV's BCG attained the log phase, they were added over the extracellular matrix in a ratio of 50,000 bacilli for 5 lakh PBMCs so that it will attain a MOI of 0.1. Now, these culture plates were maintained 37 degrees Celsius for 7 days. Here, I did not add any pollution, so this is going to act as a control group. But for the objective 1, I had to add the pollutant at the day 0 itself. So I used two concentrations of PM2.5, 0.1 and 10 microgram per ml. These were added at day 0 itself to the tissue culture plates. And the granuloma for the objective 1 were harvested on day 10. Whereas for the objective 2, the granuloma was allowed to form first. So we waited for 10 days to the formation of granuloma, and a different concentration of PM2.5 were added after day, that is after the formation of granuloma, and they were maintained for 3 days. They were harvested on day 13. So I had different sets of experimental wells. So from few wells, I have taken the supernatant and subjected for human cytokine assay by using flow cytometry. So these are the 7 human cytokines of T helper cell 1, 2, and 17, including TNF alpha and interferon gamma. And the remaining of the extracellular matrix were treated with formalin, and they were scooped out and made for paraffin blocking and subjected for histopathological examination like hematoxin eosin staining and Zeal-Nielsen staining. And some other wells were treated with collagenase type 4 to lyse the extracellular matrix, and then they were treated with Triton X to lyse the PBMCs, and they were centrifuged to collect the pellet, which are predominantly rich in the bacilli. So now this pellet were suspended in trizol and subjected for RNA isolation by bead beating method and followed by DNase treatment. And then complementary DNA synthesis was done, and the primers are designed for quantitative real-time PCR to see the expression of dormancy associated genes like triglyceride synthase 1, isocitrate lyase, heat shock protein, and reactivation associated genes like resuscitation promotion factor B. So dormant associated genes are the one which should be upregulated whenever there is latency, whereas reactivation associated genes will be upregulated whenever there is reactivation, and gene expression is calculated by using Levac method. The other set of pellet were suspended in 7H9 media, and they were plated on 7H11 agar plates and maintained for 21 days without any contamination, and CFU counts were enumerated. So first we will see the results of standardization. So this is the MTT assay. Here we can able to see the PBMCs before adding the MTT, and these are the PBMCs after adding the MTT. So MTT is the one which will taken up by only by the live cells because of the oxidoreductase activity in the cytoplasm that will be converted into crystals. So here we can well appreciate the crystals in the cytoplasmic area. So based on this data, we have plotted this bar graph in which it is clearly shown that whenever higher concentrations of PM2.5 is used, it is itself is creating cytotoxic effect on the viability of the PBMCs. So we thought we will go with only these two concentrations, one with the lower and one with acceptable higher concentrations of PM2.5 for the following experiments. So this is the control granuloma, which is formed in the extracellular matrix phase contrast images. Here we can able to see the well-developed granulomas where the PBMCs are clustered together. And this is again the images in 80x magnification. Here we can see all the PBMCs were clumping around something, forming the granuloma. So this is going to act as a control group. So this is going to act as a control group. So next we will move on to the results of objectives, for which the actual experiments are carried with the study subjects more than 18 years working in indoor environment so that they are least exposed to the outdoor pollution. And the chronic lung disease individuals, HIV patients and immunosuppressed patients were excluded from the study. And for each study group, we have used 20 in vitro granulomas for each study groups. And the one-way ANOVA test was done for these two groups by using SPSS software, and p-value 0.05 is considered as significant. So coming to the gene expression studies, in both the objectives, we can able to see with respect to the dormancy associated genes, the TGS1 and the HSPX, except this ICL, everything is down-regulated whenever we are treating with either lower concentration or higher concentrations of PM 2.5. Whereas the reactivation associated genes were upregulated, and also it is very much upregulated in the higher PM 2.5 group. The same thing is observed in the objective 2. The dominant associated genes are down-regulated and the reactivation genes with higher concentrations are upregulated. So we can infer that in both conditions, with higher concentrations of pollutant, the bacilli either survives in its active state or gets reactivated. So these are the human cytokine assays. Here we can see with respect to TNF alpha, when it is compared with the control group, that is no pollutant group, with higher concentrations of PM 2.5, it is significantly upregulated. And the same thing with the interferon gamma, we can see when compared with the control group, here also there is increased levels of interferon gamma. So we can infer that higher concentrations of PM 2.5 promotes inflammation in the granulomatous microenvironment, suggests the active status of the mycobacterial bacilli. And these are the histopathological examinations. We can see with lower concentrations of PM 2.5, here there is clumping of PBMCs here and there, suggestive of the granuloma, and these are their respective AFB staining. Here in the presence of bacilli, we can able to.
Dr. Adishankaran?
Yes, sir.
Yeah, already 7 minutes over. Just indicate.
Okay, so just 2 slides completing. So here in the presence of bacilli, we can able to see the clusters of mononuclear cells surrounding the bacilli, indicating granuloma, whereas with higher concentrations of PM 2.5, there is no any granuloma. And even though there bacilli is there here and there, but there is nothing suggestive of granuloma, even though mononuclear cells are spread apart. So we can infer that even in the presence of mycobacteria, the granuloma architecture was disrupted if treated with higher concentrations of pollutants. And these are the CF enumeration images with in both objectives. When we compare with the control as well as with the PM 2.5 pollutant treated group, pollutant may not significantly affect the multiplicity of tubercle bacilli. So summarizing the study findings, gene expression studies showed with a higher concentration of PM 2.5, it prevents the granuloma formation at the first place itself, as well as reactivates the latent mycobacterial bacilli, which is supported histologically, where it converts well-defined granuloma to a ill-defined form. And increased levels of cytokines in PM with higher concentration of PM 2.5 suggest the active status of the mycobacterial bacilli. So concluding the session, when due to increased interstitialization, there is more PM 2.5 produced, which if inhaled by the person who is already having granuloma, that is latent tuberculosis, will get reactivated, leading to active tuberculosis. Thank you all for this wonderful opportunity.
Very well done study.
Yes,
Mr. Adishankaran.
Yes, ma'am.
It's an in vitro study, or what are the study subjects? Who are the study subjects here? In vitro, or you took it in some 18 number you had given?
No, ma'am. Actually, we collected samples from the study subjects and we isolated PBMCs from those study subjects. And these PBMCs were then mixed with the extracellular matrix, subjected with this Embovis BCG strain, and granuloma was developed, which is subjected to further experiments, ma'am.
Very thoroughly conducted study.
Thank you, ma'am.
Very good study. That's all. But still, it is still an in vitro study, which is to be further carried out in human population. You have to.
Yes, ma'am. Yes, ma'am. Actually, this is a baseline study. It has its own future perspective also, ma'am.
Who were your study subjects? Who were your study subjects?
Ma'am, my study subjects were healthy individuals who are working indoor environment. That's all. That's all inclusion criteria.
So purely an in vitro study, no?
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Please extend it further, and I think it's a new study.
Yes, ma'am. Yes, ma'am. Actually, this study should be proceeded with even higher sample size, and we instead of using PBMCs, which is collected from the peripheral blood, we should have in the future we should use directly the airway epithelial cells, and instead of using BCG, we should use this virulent strain itself. So by that way, we can get better results.
Maybe you can conduct it in people who maybe you can conduct the study in a population who are exposed to this sort of pollution.
Yes, yes, ma'am.
Factories
Yes, ma'am. Yes, ma'am. So we have in future I have to use the study subjects from the people who are actually exposed.
Exposed.
Yes, ma'am.
Okay. Thank you.
Hello, Dr.
Yes, sir.
I just want to
Yes.
have a few questions. Why
Yes,
did
sir.
you
Yes, sir.
choose this 4 gene for your expression studies? Do you have any specific reason for this one?
No, sir. Actually, there is no any exact specific reason, but the thing is, just a second, sir. I will go to that slide. The reason is in the early literatures they have used only these genes, sir, and these genes are also actually, even though they have used many genes for dormancy or for, as well as reactivation, these genes give better results, sir. So I want to concise the study only to these genes. That is the reason I have chosen these genes.
No, in what way? In what way it's correlating with your outcome of your project actually?
Sir, as we see the outcome, it is clearly shown with even with treated with pollution, this dormancy genes are, as well as the reactivation genes, either it is upregulated or downregulated depending upon the conditions. So it clearly shows whenever if some individual is treated with or exposed to pollution, definitely the dormancy genes are going to downregulate, leading to the reactivation of the latent basalizer. So this will be the study outcome based with respect to the genes.
Okay. So my suggestion is that better you can send the sample for transcriptomic, where you can get that what are the upregulated genes and what are the downregulated gene. You will get a complete set out of it, so that
Yes,
you can take it for further for the biomarker development.
Yes,
for particular, I mean, for this disease actually.
Okay,
So
okay, sir.
you're just constrained to only for 4 genes actually. That's
Yes, sir. Yes, sir.
You might have done transcriptomic profiling so that you get more input. Maybe in your further extension of your studies, you can do this one actually.
Sure, sir. Sure, sir. I will keep this, sir.
Okay. Then the first is that you told that you are using a density gradient centrifugation, right?
Yes, sir. Yes.
Why did you choose this one? Because of your particle size. Did you use sucrose gradient or what type of tube did you
Sir,
use?
actually, the gradient that we used is this phycol hypax, sir. So this is the material which will act as a it is having specific density which will separate the RBCs at the lower layer and the PBMCs at the upper layer of that solution, sir. So we have used phycol histopack for that.
Okay. Then I have a certain thing regarding your concentration for study. You used 0.1 and 10 microgram
Yes,
per
sir.
liter, right?
Yes, sir.
Why don't you try with 1 as well as a little higher value? Like why you are stopped with these 2 concentration? You told that you got a better reading, but the thing is that 1 and 10 you have a large variation, right?
Yes, sir. Yes.
And 10 and 50 you'll have a large variation.
Yes,
Why
sir.
don't
There is a
you
gap also in between.
There
Yes.
is a bigger gap actually.
Yes, sir.
Why don't you choose that? So that your transcriptomic profile will be entirely different, right?
Yes, sir.
Better you might have done it with the 4 concentration so that you get an wholesome data actually. So
yes.
So are you doing a PhD or a faculty member?
No, sir. I'm senior resident, sir. This is my MD work, sir. Thesis work.
So maybe you just think over of it to have a wide concentration so that you get a much more insight about your transcriptomic profile.
Sure, sir. Sure, sir.
Okay. Thank you. Well done.
Thank you, sir.
Thank you, Doctor.
Thank you,
There is no other questions. Right. We'll move to the next presenter. Dr. Neeraksha, BMR 400.
So is my screen visible?
Yeah, it's visible. You can proceed.
Yeah.
Put in
So
slideshow
shall I
mode.
start? I'll start?
Please change to slideshow mode.
Yeah, I did.
Yeah, no, fine. You can.
Good afternoon. I'm Dr. Niriksha, Assistant Professor at Department of Conservative Dentistry, ABC Memorial Institute of Dental Sciences. So my work is on potential role of antimicrobial peptide, that is LL37 and vitamin D, and its role in dental caries. Dental caries is one of the most commonly prevalent diseases in global condition, and it's not only mediated by the dietary factors or any other factor. Now, because there is a lot of modification in the definition, it also says it can be a biofilm mediated and also a dynamic disease which can have various other underlying factors. So before it was only those factors like the dental flora, the pH, and the diet. But right now we also look into genetic factors, because one of the study like Weipel-Humud study which said that it's even though a group of people in an institute were given with the regular similar diet, there were certain even under uniform conditions there were a lot of people who were resistant to caries. And why these people who were resistant, the major reason was their genetic makeup or the underlying causes. So that's why in this present study, we had focused more on the vitamin D and its association with peptides, which can play a possible role, and maybe one of the underlying factors in various cases where dental caries is not because of the presence of any etiological factors. Even then, they have a lot of prevalence of caries. So vitamin D, we know we speak when it comes to dental or tooth related factors, we speak more about calcium deposition, calcium mineralization and all that. But the other factor, that is once the tooth matures, the more important factor for vitamin D is definitely the innate immunity system, where the genomic effects of vitamin D and its effect on various peptides, how it decreases the inflammation and infection through the expression of some of the peptides. Now we know there is active and inactive form of vitamin D. The active form, that is once it's converted into 125 hydroxy vitamin D, it binds with vitamin D binding protein and gets into the target cell to perform its genomic functions. That is along with the vitamin D receptor and a complex that forms. Though they go and attach to particular areas of these target areas in particular genes. Now here we are looking into LL37. The target gene here is CAM gene. So there is vitamin D receptor element on this particular gene, and where it attaches and it produces LL37. So the LL37 that is produced, it acts on the pathogens that's engulfed by the bacteria. As you can see in the image, there is a CAM gene promoter region to which the whole complex of vitamin D receptor and the other the complex that comes and attaches. So now the vitamin D receptor element polymorphisms affect various affects the LL37 production. So these are the four or four detected single nucleotide polymorphisms, and we have seen in the study what is the prevalence of this vitamin D receptor polymorphism and its effect on how the prevalence of dental caries changes. So these are the four detected single nucleotide polymorphisms, and I've mentioned the chain in the acid and the location, and why we have chosen, what is the reason for rationale for selection of these SNPs. So coming to the peptide which this polymorphism might affect, because that's a way how it is produced. The LL37 is a human antibacterial peptide, and it has all the properties of wound healing and etc. And it is proven in a lot of studies how it affects the oral flora. Now, the objective or the study question is here, that the single nucleotide polymorphism, does it affect the dental caries? Also, is there an association between salivary vitamin D and antimicrobial peptide with dental caries? And there goes the aim and objective, that is to evaluate the association between salivary vitamin D, LL37, and the vitamin D receptor gene polymorphism in dental caries. 377 samples were chosen based on the inclusion criteria. Here we had excluded all the poor oral hygiene patients with periodontal diseases. We had only chosen patient with the good hygiene habits and all the history related to the food consumption and nutritional supplements were taken, and the age group was 18 to 40. So here we have done ELISA estimation of saliva vitamin D and LL37. Also salivary DNA isolation was done, and vitamin D receptor gene polymorphism was evaluated. This is the ELISA method, and how the salivary collection was done. DNA isolation for saliva. And these are the primers and the PCR cycles for the same single nucleotide polymorphisms. The fragment size of the particular single nucleotide polymorphisms is mentioned here. Statistical analysis. Coming to the results, The vitamin D was significantly, it was higher in caries free individuals compared to the caries active individuals. Also, when we saw with the severity of caries, because we had not just seen whether there is decay or not, we had also seen the number of caries and the number of and the vitamin D prevalence. Along with that, we are also seeing the PUFA scores, that is a pulpal ulcerative fistula abscess score. Says if one tooth is there which is pulpally involved, it is equivalent to almost 2 or 3 initial caries. So we had seen if the individual had more PUFA scores, then he would also show a low vitamin D. Therefore, vitamin D level of 28.33 picogram per ml had 71% specificity with 57 sensitivity with 57% specificity. So the optimal cutoff can be in saliva, that is 28.33 picogram per ml. So vitamin D stabilizes the demineralization and disintegration of the tooth surface, maintains adequate surface proteins by upregulating various proteins. Coming to LL37, we did not find a significant difference in the salivary levels of LL37. However, a few studies mentioned that because of the capacity of these LL37s to reduce the thickness of the biofilm and decreasing the adherence of these biofilm of the tooth surface, so due to this, maybe we are getting a lower level of LL37, or we might have to do in a bigger population, that is more than 400, to justify the levels. Coming to the vitamin D receptor gene polymorphisms, among the 4 single nucleotide polymorphism, the fork one was the most important, because the change in the amino acid that happens, which I'll explain why. But the difference in the prevalence of the genotypes between the caries free and the caries active group was highly significant. And as you can see, the genotype CC is higher or it is more associated with caries free, and comparatively, C allele here is associated with caries free group, and allele T is associated with caries active group. Coming to TAC1, TAC1 also there is a amino acid change, but the amino acid change is similar. So that's why there is not much association for TAC, BSM and APPA. But FOC1 gives a very significant association between the caries free and caries active group. So this is all the genotype prevalence of TAC1, APPA1 and BSM1. I would like to skip this, but the vitamin D receptor gene polymorphism, that is FOC1. Why does FOC1 make such a big deal here? Because the mutation is from methionine to threonine at the position 1, and the mutant size is smaller than the wild type. The amino acid chain decreases from larger to smaller. That's why the activity of this amino acid increases 2 to 3 times more. So the downstream regulation of all the antimicrobial peptides after this is bound to increase because of the this change in the amino acid. That's why FOC1 can be one of the most important prevalent factor, or can be one of the underlying factor in prevalence of dental caries in cases where there is no chances that the person can get caries with other etiological factors. This can be one of the factors. So the salivary vitamin D statistically decreased with increasing severity of caries, which showed that vitamin D plays an important role. And when pathogens are detected by these toll-like receptors, there is an activation of enzymes that is converted from 25-hydroxy vitamin D to 125. Then it binds to this complex, binds to this receptor element, hence production of LL37. So this is a protective role how vitamin D works. But among the 3 significant SNPs, RS-2228570, that is a single nucleotide polymorphism for which the RFL restriction enzyme is FOC1, showed a better correlation due to its amino acid change that occurs at position 1 from methionine to threonine. The amino acid changes from longer to shorter chain. Thereafter, the vitamin D produced may be 1.5 to 2 times more efficient. Therefore, downstream regulation of peptides may also enhance. So, the RS-2228570 has a significant association with dental caries progression and occurrence. So the present study the significance of the present study would be that the optimal presence of salivary vitamin D is very important for the protection of the tooth surface through these peptides and antimicrobial peptides. And RS-2228570 in BDR, the single nucleotide polymorphism, makes individuals lesser susceptible, and therefore can be one of the underlying factor. Antimicrobial LL37 peptide is protective and prevents the bacterial colonization of the tooth surface, and hence can be a possible candidate for exogenous administration. Thank you.
Over to judges.
This is Niriksha, right?
Yes, sir.
So you have what is your sample size actually for your study?
377, sir. 100 was control. I mean, in the sense the care is free people, and rest was the care is active.
Okay. So what is the homozygous mutant? What is
TT, sir?
No, I'm asking about the. Can you go back to the slides, please?
Yeah, sure. Yeah.
So what is the percentage of homozygous mutant?
Percentage. Among the care is free, sir. CC is more because the sample size is 100 there. So among that, 88 people have CC.
Okay. So you have mentioned that RS228570.
Yes.
So it can be developed as a biomarker.
Sir, actually, with this sample size, we found it is quite evident compared to all the SNPs. So with further more number of samples, we might be able to say it can be a biomarker.
Okay. What kind of evidence you have done for vitamin D actually?
No, vitamin D actually for salivary also for all the 400 samples we have done, sir, the ELISA. So with that we are getting that vitamin D levels are higher when there is not much, like in a sense it's associated care is free. But with a bigger sample we can justify more, but that's what we have found that it is
why
significantly.
I'm asking is that you have done it with ELISA. Why don't you do this same experiment in the expression study of that particular gene? Because vitamin D will be produced one genetic polymorphism. The amino acid, only that particular amino acid will be there. Okay, it can be visualized in your what do you say in your ELISA. But if you look into the genes response, I mean the enzymes responsible for it is doing its actual function or not. Okay. So if you want to compare it, you need to do the expression studies along with the
biochemical levels.
Yeah, biochemical evidence.
Yes,
that will give you a clear idea about
its function.
So did you collect the saliva or did you take the
saliva, sir? Actually, initially when we wanted, we have mentioned about blood, but the ethical committee was like because my inclusion is only purely healthy, only dental. They're coming for dental treatment. They did not want because sometimes patients do not want to give blood samples, so they said you have to start with saliva. Then we started with saliva. Even the DNA isolation was done with saliva itself.
Okay. Okay, that's it from my side.
Okay, sir.
Thank you, doctor.
Yes.
I think madam, the other judges, due to technical things, they have left out. I think so. They are rejoining back. We'll call the next participant.
Okay, sir. Thank you.
Dr. Shilpa Shetty,
Yes,
BMR
sir.
403. So you are there?
Yeah. Hello, sir. Is the screen visible?
Yes, you can proceed.
Very good evening to all. My topic is fatty acid desaturates to gene polymorphism and susceptibility to type 2 diabetes mellitus. I'm working as an assistant professor grade 2 at Central Laboratory at KSLT Medical Academy.
Coming to the introduction part, type 2 diabetes mellitus is a metabolic disorder characterized by the presence of chronic hypoglycemia, resulting in the impairment of metabolism of carbohydrates, lipids, and proteins. When we talk about type 2 diabetes, the Indian phenotype is usually said to be more susceptible to type 2 diabetes mellitus, and it is mainly, according to the literature, it is said to be mainly due to the Asian Indian phenotype. That is, we have more abdominal fat, usually referred to as the thin fat Indian, higher levels of C-reactive proteins, lower levels of adiponectin, and a characteristic dyslipidemia, which shows low HDL levels, high triglycerides, and high levels of small, dense, low-density lipoproteins. Now, why is this fatty acid desaturase gene polymorphism is selected for this study? Free fatty acids or even complex lipids and their metabolites impact a wide range of physiologic and pathologic processes, and polyunsaturated fatty acids, mainly omega-3 and omega-6 polyunsaturated fatty acids, are synthesized by the intake of these essential fatty acids, ALA and LA, which go through a series of enzymatic desaturation and elongation steps to produce their primary biological components like EPA, DPA, DH, and ARA, and fatty acid desaturase gene is the one that is involved in this process. This is the representation of the PUFA pathway, and these are the two enzymes that are the desaturation enzymes that are mainly involved in the process, delta-5 desaturase and delta-6 desaturase, where delta-5 desaturase is encoded by the gene fatty acid desaturase 1, and delta-6 is encoded by fatty acid desaturase 2, which is also said to be the rate-limiting enzyme. In the present study, we have selected one SNP, that is RS174575, which belongs to the FADS2 gene and is present clustered on the chromosome 11. This SNP was mainly selected based on previous studies because it provided a best combination of two desirable factors as per the literature. It had known linkage disequilibrium throughout the region, the promoter and the intergenic region of FADS2, and it had a minor allele frequency sufficiently prevalent to permit their use in the test of gene environment and gene-gene interaction. Why is this study needed? So recent study evident that there is an association between SNPs in FADS1 and FADS2 gene and disease susceptibility, but our current knowledge on the impact of these genetic polymorphisms on long-chain polyunsaturated fatty acid metabolism and their potential role in the development of disease is limited. And most of the studies is done on the Western population, whereas the studies on the SNPs from the fatty acid desaturase gene is very much limited in the Indian scenario. With this, the study mainly aims to study the fatty acid desaturase 2 gene polymorphism, that is mainly RS174575, and its role in the susceptibility to type 2 diabetes mellitus, with the following objective, that is firstly to analyze this polymorphism, and secondly to find an association between the glycemic control and the gene polymorphism in type 2 diabetes mellitus subjects. Coming to the methodology, this is the complete flowchart of how the study was approached, with the procurement of institutional ethical clearance, recruitment of subject, followed by anthropometric body measurements, fasting glucose sample collection, glycemic profile, fatty acid composition, and FADS2 gene polymorphism. And the data was analyzed for comparison of the study parameters between the group, correlation of the study parameters within the group, and the association study, finally building a predictive model for type 2 diabetes mellitus using this genotype. This was how the sample size was calculated, and the calculated sample size was 196 subjects in each group. This is the study setup. We had total of 429 subjects which were recruited, with the non-diabetic 216 and diabetic of 213 based on the following inclusion and exclusion criteria. PCR amplification and gene sequencing. DNA isolation was performed using our lab-grown method, which is a modified method from the Chiltern Lab, Wake Forest University. Quantification was performed, a PCR was done, and electrophoresis. And finally, the PCR product was subjected to Sanger sequencing. Statistical analysis was performed using SPSS software. Log transformation was performed for skewed variables, and odds ratio was with the 95% confidence interval for logistic regression analysis. And the discriminatory of the substantial model with the highest odds ratio was done using receiver operating characteristic analysis. Coming to the results and discussion. First, we compared the glycemic profile, lipid profile between the non-diabetic and diabetic group. And here we found a statistically significant difference between the two groups, except for HDLC. Then we analyzed a single nucleotide polymorphism of RS174575. The SNP was selected based on the NCBI database, and these are the primers that were utilized for the study. This is the agarose gel electrophoresis of the PCR product that we have found, that is around 329 base pair is the size of the product, and this is the electropherogram result that was generated from Sanger sequencing of the PCR product. Genotypic distribution between the non-diabetic and diabetic group. In the diabetic group, 14% showed recessive phenotype was higher when compared to that of the diabetic group. On comparing the glycemic profile and lipid profile in diabetic individuals based on the genotype, we developed 3 models: additive model, dominant model, and recessive model. So first we compared the genotype, that is additive model, with that of the glycemic profile as well as the lipid profile, and we found a statistically significant difference between the 2 groups. Even in the dominant model, we found a significant difference. But in the recessive model, we found significant difference between the insulin resistance, insulin sensitivity, and the lipid profile parameters. From this, we took up the association studies to develop the predictive models involving the genetic marker. And we predicted 3 univariate, 2 bivariate, and 1 multivariate model. And as per the results of the ROC, we found the multivariate model to be the better one because it showed a 3-fold association with diabetes. So with this, we can say that our suggestive discussion on this is that when we increase saturated fatty acid content of the membrane, it will render it rigid, which leads to the decrease in the number of insulin receptor and affinity of the insulin to its receptor, which in turn causes insulin resistance and associated hyperinsulinemia. Therefore, in the heterozygous recessive, it might be resulted in reduced D6D activity, thereby reduced PUFA metabolism because it is one of the rate limiting enzyme there, reduced membrane fluidity, thereby decreasing the insulin receptor, which might result in insulin resistance. This is a suggestive mechanism of how the homozygous and the heterozygous interaction may result in insulin resistance, because PPAR gamma, it is one of the its activation mainly is involved, plays a major role in insulin sensing, and these PUFAs are known to be their biological ligands. So decrease in the ligand formation may thereby result in inactivation of PPAR gamma, thereby resulting in insulin insensitivity, glucose uptake, reduced glucose uptake, reduced lipid lowering, and thereby causing insulin resistance. So with this, we can conclude that the distribution of this RS17457 genotype differed between type 2 diabetes mellitus patient and controls in the present study, and it does appear that having the minor allele genotype G alone would significantly increase the risk of incidence of type 2 DM. The study also revealed that genetic variation in FADS2 could be an additional facet to consider when studying the risk factors of type 2 diabetes mellitus, and this reinforces the value of incorporating a fatty acid-based approach in future clinical studies to investigate the contribution of specific fatty acids to the development of insulin resistance. These are my references. This is the paper that is published based on the present data. Thank you. Hello.
Yeah, thank you. Over to the next, sir.
So Dr. Shilpa, it's a good study.
Thank you, sir.
I have 2 questions. One is that why did you choose particularly this SNPs?
Sir, as I have showed in one of the slides, based on the literature we had surveyed, as this SNP had a LD with other 5 SNPs, sir. So based on that, and it was near the promoter region, that is how it was selected, sir.
So could you, tell me what is the percentage of mutant did you receive in your study?
In the percentage, sir.
Can you go back to the slides, please?
1 second. Among the diabetic group, it was 14% were having the CG, 2% were having GG, and 84% were CC.
So you have done some grouping, right?
Yes, sir.
Based on your dominant recessive.
Yes.
Can I see that table? Can you share that once again? You stopped sharing. Okay.
Yeah, I'm doing it. You can see, sir?
Yeah.
This is the additive model, sir.
Okay.
This is the dominant model, but here in the recessive group, it was very less. That is only n is equal to 4. So we have taken the recessive model because we had 180 that is compared with 33. So we have picked the recessive model for the association studies.
So what is the diabetic, I mean, in what stage the diabetic patient you have chosen?
Early diagnosis.
All you have chosen is early
Yes.
diagnosis. So there is no differences in the age.
1 second.
Do you have a age match controls or?
Age match control, sir. It was 45 to 60.
Okay.
30 to 60, sir.
So the 60 years, it was the first diagnosed with diabetes.
No, it actually started with the 45 was 45 was the age that started up to 50 was in what we had recruited, we had got, sir.
No, why I'm asking is that you have just compared with the biochemical parameters with the
Yeah,
genetic.
because the thing is that, sir, this data, what is presented now, it is a part of the data. So the complete data is not given here. That is why that age one is the table is missing.
Actually, you have shown this flow chart, right? That is not a Sanger sequencing. You just go back. That is a. It is not a 6 studio actually.
Pardon? This one, sir?
The picture you have showed as a Sanger sequencer, it is not a Sanger sequencer. Okay, it is a real-time PCR. Yeah, it is a real-time PCR. Please try to change the photos.
Yes.
You done the sequencing in your lab or
No,
you
we
just
had outsourced it to Eurofin, sir.
Okay. You just go to your mechanism part.
This is just a suggestive mechanism, sir. We are doing in vitro studies on this particular thing.
In vitro means
using the cell line.
Using the cell lines.
Using knockout models. Knocking out the FADS2 gene, and it is an ongoing work, sir.
You're knocking out the FADS gene, or you're just creating a single mutation there?
No, we are knocking out the FADS2 gene, and then we are checking with the expression of PPAR. And we are supplementing it with the fatty acids, like long chain, omega-3 and omega-6 in different ratios. Because another part of the study was we had checked the ratio of omega-6 by 3 fatty acids, and we found it to be.
How can you relate this this FDS mutated and knocked out on? Hello. Hello.
Hello, sir.
Shilpa, can you hear me?
Yes, sir.
So how do you relate in vitro? I mean, this is not a part of your study, even though I was a little curious.
Sir, actually, I'm not supposed to discuss it online, sir. It's a part of, it's still unpublished and it is going on.
So in that case, you should not show the slides, right?
No, this was actually the suggestive mechanism that we had put up, and based on this, we developed the studies. This was on the study which led to like a future direction. What was there from this study is that what we have taken.
Okay, interesting. Thank you.
Thank you all. At the outset, I would like to thank our judges, Dr. Shivlila ma'am, Dr. Ponadali ma'am, and Dr. Ravikumar sir, for spending their valuable time in judging this Young Research Scholar Award under faculty presentations. Thanks and congratulations to all the participants who have done their participation today. And the result will be announced in the last day during our valedictory function. And please do join us for knowing the results and also for our valedictory function. Thank you all.
Thank you, sir.
Thank you, sir.
Thank you.